Prova, prova. 15-minute talk. That's the one I gave you for Michael McIntosh.
Michael. Okay. Good. Okay.
So the third one in that session. Just make sure it's right.
Oh, yeah, I got a bunch of talks on that.
Macintosh L121 Siena.
Yeah,
This.
yeah.
One of them. I should not.
Yeah. Well, what do you do? If she leaves the country, she might lose her status. Yeah. Where do you find the restaurant? Downstairs?
No, upstairs.
Straight back?
Yeah, straight back there. There's little outflows here and there.
This is here by gate.
Siena talk. Siena talk. You are. This is the first. Okay. Siena for the second session. That's the second session. the first video. Second video. Okay. This is you stay in this point or the big button for the next slide or
But you probably should, you have to stand right here because it's screening. You can't move.
yes.
You know how you like to talk around? No, you stay there.
Because you see I can stand this slide, but I can stand. Okay. There is another.
Now, do we have a pointer or
Yes.
is there an online? So are there people online?
Or
They
the-
see your pointer?
So I should do this. When speech is here.
Got it.
Okay.
So talk behind the
This
computer.
is the room, the
No.
school room,
Okay,
and no auditorium. Big
understood.
auditorium.
So that folks online can see what we're pointing at. Is there Wi-Fi access? Oh, I see desk. Yeah. Desk. You guys need to. You've done that? No.
Per la prima sessione, giusto? No, non la vedo ancora. Professor Piacini, ma me lo può fare avere un programma per cortesia? Vado un po' alla cena. Seconda sessione, inizio della quando parlo? Parla del cambiamento. sposa sia questo che sta pensando, non lo so, consiglio di usare il mouse come puntatore.
Comunque questo è il proprio stesso.
This is the screen of.
You can use it also, but it's
It's very complicated. Okay.
How are you?
Siena meeting final. When you speak.
No. Forgot my card.
You're in Utah, right?
This is for it.
Io ho fatto quello il primo che ho fatto. C'era un video che era audio, che si fatto la audio.
Drives on Mac. Non si la vedo. No. Solo il Mac lo legge. Siena. Prova, prova.
Red light.
Okay, and then. Yeah, perfect.
All right, thank you.
My talk today.
No, e lo speaker chairman sempre di là perché almeno
Il chairman vanno di là e lo speaker ma questo è tutto a disposizione. Sono pendenti.
il link, Roberta l'hai mandato un link nuovo. Ah,
Vuole
ok.
sapere lui? Sì, sto per mandare
Fai link.
Yes, yes. Maybe you could send it. It took a few minutes. 2 minutes. All the people who are yes, siamo qui e deve far partire la streaming. Allora, where are you? Cioè, se ne avevo 10 erano 2. O se no, il professor Pattari. Solo 2, il professor Pattari. Da tutto allora.
Arrivando altri, parlo. Almeno parlo.
No.
You're the first speaker, right? Yeah, that's what they tell me. Okay, perfect.
Siena, l'Italia. Posso suggerire, ragazzi?
Pronto?
of the presentation. Finally.
Good morning. If there is anyone who has not uploaded the slides, people who are speaking this morning, please come forward and do it. Thank you. Francesco Baccianti? Francesco Baccianti? Puoi venire un secondo giù, per favore, per caricare la tua presentazione.
Okay, good morning to everybody, and I'm pleased to welcome you to this 20th meeting of the on EBV. This is one of the first meetings that we are organizing here in Siena in present after 2 years of pandemic. So there is a particular thank to all the organizing committee and to the staff of the University of Siena, Roberta, Giuliana and Elisa, and also to the Department of Medical Biotechnology, Sandra Maggi, who help in starting again all the Congress machinery. But we are not yet out of the pandemic, and so I kindly ask you to wear the mask during this meeting because there are 170 participants from all over the world. There are many from Europe, many from United States, but also from South America, also from Japan and Australia. So I want all of you. Who? At the mask. Okay, we'll do. Okay. Okay. And anyway, and I want, so there are all these participants, and I want that all of you go back safely at home. As you have seen in the web page, there is a lab. We have an agreement with the lab to do the test if you need the test before going home. So now we start to go to the scientific program. Today will be based mainly focused on the biology, on basic biology, on EBV infection and mechanism of oncogenesis. Tomorrow will be more focused on pathology, on the EBV-associated malignancies, in particular lymphomas, and apologize if I put too much emphasis on lymphomas, but I hope that you can forgive me. And then, but there is not only lymphoma, we have also put a session on multiple sclerosis because we have received many abstracts on this. So we thought that we could dedicate a session to this new topic on EBV pathology. And finally, then we will finish with therapy and vaccine development. And I really hope that you will enjoy the scientific program of this meeting, but that you can also enjoy the atmosphere of the city in these days, special days for the city, when Siena, that is usually a small provincial town, in this day became again the capital of its republic, ancient republic. So welcome to all of you, and if Pankaj want to give, that is helping in the organizing of the meeting with me, want to give a few words. Thank you.
Thank you, Lorenzo. Buongiorno. Good morning, everybody. My name is Pankaj Trivedi, and I am the co-organizer of this symposium. I am at the La Sapienza University in Rome, and it is a great pleasure and honor for me, and also a personal delight, to welcome you all to this 20th International Symposium on EBV and Associated Diseases in this fantastic, beautiful medieval town, Siena, in Tuscany, the land of some of the most revered giants of human conquest, like Leonardo da Vinci, Michelangelo, and last but not least, Brunello di Montalcino. It is a particular pleasure to see so many young faces today, and to me that is an indication of the attractiveness of our field of research and also a hopeful sign for future. Now the Italian connection with the International EBV Symposium started back in 1988 when Alberto Fagioni at the Sapienza University organized the third EBV symposium there. And I was a graduate student at that time in George Klein's lab at the Karolinska Institute in Stockholm. So I had absolutely no idea that I would be settling in this country. And after 34 years of gap, I will be part of the organizing committee. So it's it's kind of nice to see the closing of the circle. So organizing conferences is not an easy task, and even more so here in this country, full of beautiful red tapes and the pandemic and all the restrictions that followed. But as you know, the saying goes, when the going gets tough, the tough get going. And Lorenzo and I, we strongly believe in that motto. And in order to explain you better what I mean, I have this
what the fellow said. Mentally, for 30 years under the Borgias, they had warfare, terror, murder, bloodshed, but they produced Michelangelo, Leonardo da Vinci, and the Renaissance. In Switzerland, they had brotherly love. They had 500 years of democracy and peace. And what did that produce? The cuckoo clock. So long, Howard.
So that was from the 1949 classic, The Third Man. Is Christian Munz here? Christian, my apologies. And please forgive Orson Welles, because I'm sure that when it will be your turn on Wednesday, you will prove him wrong with your data. The final word of advice. It's very, very warm outside, so please hydrate yourself. And by hydrating, I mean with water. Thank you very much again for surviving the pandemic and making it all the way to Siena. And I hope you will have an enjoyable meeting. The meeting is, of course, we have organized it in a very informal and a relaxed way so that many of you can interact with each other freely. The new postdoc, if they're seeking for new opportunities, you'll have plenty of time to interact with PIs and also start new collaborations. So thank you very much again, and let's have a great meeting. Thank you.
Okay, so I think we're going to begin the session now, and I think we might actually begin with a quick round of applause for Lorenzo and Pankaj for getting us here. And I've just got one very quick announcement before we start the session, which is this is really just for people who are interested in diagnostics for EBV. And there's an initiative from Argentina, actually, from Buenos Aires, to try to get a group together who would like to discuss common standards and practices in EBV diagnostics. And this is being promoted by Dolores Fellner in there. And I'm going to ask Dolores to stand up so you'll actually know who she is. So if you're interested in diagnostics and would like to talk to Laura, she's very nice, please do that. And she's going to be at the poster sessions, I think, for a start. Or you can just find her in the coffee break. So diagnostics, Dolores. Okay, thank you very much. So now I'd like to invite Earl Robertson to give the first talk and to remind all the speakers that within their allocated time, we're hoping that they would leave a couple of minutes or 3 minutes for questions. And Earl, you're going to sit there and talk to the screen. It's Zoom all over again. But we're here. Okay, take it away, Earl.
Thank you very much. Thanks to Lorenzo and Pankaj and the organizing committee for providing the opportunity for this presentation. And today, what I'm hoping to do is provide you with some new information that we have identified in the lab looking at M6A methyl 6 adenosine modification of the host epitranscriptome and how it might actually be involved in interference signaling and promoting EBV lytic reactivation. So can't really have an EBV talk without giving you some background, although everyone here knows about it, but I figure as the first presentation, I should provide some background. And as we well know, this is a picture of Tony Epstein and Dennis Burkitt. And with Epstein-Barr discovered EBV in the early 1960s, examining tumor material from lymphoma patients by Dennis Burkitt. The map here of equatorial Africa, the malarial belt is seen here, and the 814 translocation, which is quite common in Burkitt's lymphoma. And here's a classic photograph of a young child in equatorial Africa with Burkitt's lymphoma. As you know, primary infection, greater than 95% of the population is seropositive. 50 to 75% of young adults are positive in developed countries, and about 100% after leaving college are seroconverted. It's referred to as the kissing disease of glandular fever. Yeah, we have EBV-associated disease and mononucleosis, incorrectly spelled and missing an L. And then two photographs here provided by Dr. Walder. One that Rosemary provided those to me some years ago, as well as more recently multiple sclerosis. Some of the information that most of you know, the different types of latency program, the type 1 default, the growth program, and true latency. Lawrence and Alan Rickinson did a nice job in a review looking at the primary infection of epithelial cells going on to infect naive and memory B cells to see the memory B cell reservoir of EBV infected B cells. On the right, you see a map, a circular map of the Epstein-Barr virus episome, with 184 kilobases in size, with a number of the transcripts that listed, as well as the Barr 0 transcripts. And a lot of the work here was set up in terms of the sequencing and all of that by an annotation, a lot of the work by Paul sitting to my left here. And a lot of other people contributed to this as well. So to move on to what I wanted to talk to you today about is RNA modification. So what's known about this? It's the most abundant modification in eukaryotic mRNA discovered in the 1970s. It's about 10,000 M6A sites in over 25% of human transcripts. Plays a role in splicing, transport, stability, protein synthesis. A lot of the molecules involved referred to as writers, readers, and erasers. The writers MET L314 WTAP. There's a new one, MET L16, that's recently identified. There's a number of readers as seen here, and the erasers ALKBH5. The M6A methylation is recognized by a motif referred to as the DRASH motif, or the consensus sequence, as seen here. This is, again, extremely important in regulating a lot of cellular processes, and more to come, I'm sure, in the future. Over the last few years, there's a number of recognition of M6A modification in host pathogen interaction in HIV, flaviviruses, Zika. S.J. Gao published a nice paper looking at KSHV, and we did an initial observation looking at the identification of MET L14 critical for viral-associated tumorigenesis and modification of the EBV epitranscriptome, and critical for the tumorigenic process. So just giving you where we started from in this questions that we tried to ask, and the reactivation of the virus, whether or not there's something that we need to understand a little bit more in how M6A modification might be important. This is the schematic to the left here, where you reactivate cells, collect the mRNA, do the fragmentation and the immunoprecipitation with the anti-M6A antibody, and do next generation sequencing. And the analysis led to identification of a number of different peaks for the latent ACATA and the reactivated ACATA and reactivated LCLs. So we did it in two different cell lines, one, the ACATA cell line that we're quite familiar with, and as well as a recently derived lymphoblastoid cell line that was infected. And as you can see to the right, you have most of the modification occurring in the exonic regions, as well as the TTS, and some others in the 3' UTR and as indicated here. In the latent type identification of peaks, was identified, but we focused on the reactivation, and there were 4 molecules that was identified that we went on and validated. 2 of those are identified here, the DTX4 and the TYK2. TYK2 is involved in antivirus interference signaling, and the DTX4 involved in Notch pathway and central pathogen-associated diseases. So what's known? We have the DTX4 is the delta-X E3 ubiquitin ligase involved in signals nucleocytosolic sensors. The NLRP4 DTX axis is a key suppressor in the tank binding kinase 1, or the kappa B kinase, related in innate antiviral signaling on type 1 interference signaling. The type 2 tyrosine kinase 2 is a key intermediate in interference signaling pathways. Interferon and gamma, as you well know, inhibits the lytic reactivation, and LMP1 was previously known to interact with type 2 in dampening its activity and anti-proliferative selective pressure imposed by interferon alpha. If you overexpress MET-L14, you can alter the levels of type 2 and DTX4 as seen here. If you knock down MET-14, you can see type 2 levels going up and DTX going down. And if you overexpress MET-L14, you see the reverse. So that's the relationship that we first saw in terms of how M6A methylation might be affecting the expression of these 2 key genes involved in interference signaling. If we look at the results of the methyl rib sequencing for DTX4 and type 2 on the left and DTX4 on the right, the clear indication I just want you to look at here is that in the color sequence in both panels, you can see increased M6A methylation on both the type 2 and DTX4 across the board, and more so in the reactivated LCL and reactivated ACATA seen in the purple and green. You go on to validate this and look at this more closely, you can see that in the latent infection and reactivated, you can see clearly that you have induction of M6A modification of the RNA for LCLs and ACATA for both type 2 and DTX4. In normal messenger RNA, you can see that you do see a type 2, but not that much difference in the DTX, in the LCL here. But in the ACATA, you do see a drop in DTX mRNA. If you look at the Western blots to the right, the GP350 and BZLF1 is just to show you that we do have reactivation happening. And if we do FACS analysis, we have over 85–90% of the cells induced for reactivation as determined by FACS and GP350 staining, as well as BZLF1. And you can see again, if you look at the tank binding kinase, DTX4 is going up and type 2 is going down in your reactivated cells. Is it independent? Is it dependent on the cell types? We looked at BL41, B95, and the method of reactivation. The answer is no. You can see here again inducing reactivation by TPA butyrate and cross-linking as shown here. We also did it by hypoxia induction for reactivation. And all of these show that you do have reactivation, and you can have induction of DTX4 and type 2 methyl 6 adenosine incorporation of the transcripts. What's the effect on mRNA transport and stability? Type 2 mRNA in the cytoplasmic fraction, as seen on the left, you can see that you have significant less type 2 in the reactivated cytoplasmic fraction as seen here. And DTX4 not so much as seen here, but clearly with type 2 there's a significant loss in type 2 methyl 6A incorporation. If you treat the cells with actinomycin D for 12 hours and then look at this, you can see by 24 hours there's a significant loss in type 2 signal in both the LCLs and the CADA as seen here for the latent as well as the reactivated population of transcripts. With the DTX4, there wasn't much of a significant difference in any of those. If we look at polysome profiling as seen here, we can clearly see that with the DTX4, you have a dramatic increase in the polysome compartment in both what we see here for DTX4 and during reactivation, there's clearly less of the DTX mRNA in the mRNP fraction, but not much change in the TYK. So in the polysome fraction, there's clearly significant increase in DTX4 mRNA during reactivation. So that increase in mRNA polysome fraction explain why there's more DTX protein during reactivation, as we showed earlier. What is known about the reader YTHDF2, we know that there's an enrichment with TYK2 during reactivation. If we look to the right here, we see a fold TYK enrichment in reactivation. Here, if we use RNA-IP, we're using anti-reader antibody, and clearly there's a greater enrichment here. And if you silence YTHDF4, you have an increase in the expression of TYK2, as seen to the right in the Western blots. So what is known about the reader? One of the things that we showed was that this affects the interferon alpha and beta expression. And we know that the YTDF2 leads to higher enrichment of the M6A methylation of the TYK2 mRNA compared to controls, as seen to the right. But when we look at the interferon alpha and beta, you can clearly see that you have an induction in interferon alpha and beta levels in reactivated as well as latent infection. And if you knock down YTHDF2, you see clearly viral copy number also reduced. That's clearly demonstrated that there's a clear effect on the reactivation and production of viral progeny on a knockdown of this reader. If you mutate the DRAC motif in the DTX4, as shown here, you mutate those, you can clearly see you have reactivation of the enrichments as seen here on the left. But in the mutated, you see not so much of that enrichment. And if you look at the polysome profiling, again, you can see that you have a loss of the polysome compartment off to the right, as seen here. And clearly in the mutated protein levels, you see a loss of that. We tried to mutate the TYK as well, weren't so successful in actually obtaining the clones for that. So we ended up doing the knockdown of the YTD, and that gave us a similar result. We just couldn't recover the mutants for the TYK2. So what's the involvement? Silencing of this resulted in increased amount of interferon alpha and beta during reactivation, and silencing of TYK2 significantly lowered interferon alpha and beta. And clearly the results point to a role for both the TYK2 and DTX4 in interferon response during reactivation. What we knew previously is that we demonstrated reactivation in both the writer molecule, that MET-L3 and L14, where this was downregulated. We wondered how some of the host mRNAs were hypermethylated during reactivation. So there's two possibilities. There might be another methyltransferase. We thought maybe L16. This is something we're looking at a little bit more closely, but also the fact that the eraser may be selectively removing the methyl marks on these transcripts during latency and high levels, resulting in higher levels of methylation for reactivation. And here we see that the eraser ALKBH5, you have a loss of that in the reactivation in LCLs as well as SOV1 and 3. And here again, we're showing that off to the right by Western blots. What is we were able to demonstrate again was that the role of the eraser on TYK2 and DTX4 gene expression, if you silence it, you see downregulation of TYK2 and upregulation of DTX4. Then if you do the RIP followed by PCR, you can see that silencing of ALKBH5 increases methylation, and also viral copy numbers increase significantly on the knockdown of the eraser, as seen in the graphs at the low end of the screen. So clearly, there's a direct link to the control of TYK2 and DTX4 and how that impacts the transcripts here, but also in the viral copy number as seen to the right of the screen for both the ACATA and the LCLs. If you knock down the eraser, you can see an induction of the viral copy number as well. So this is also demonstrating that there's a link between the control of the eraser in that process and the viral copy number as seen here. What is known in the antiviral response for type 1 is that you have the STAT1 and 2 and IRF9 homo and heterodimer. It forms the ISGF3. This gets nuclear translocated, and this results in activation of the ISGs. Here is just the Western blot showing that in the knockdown of the eraser, you see a loss of phosphorylation of the cytosolic and nuclear stat. Here, just by immunofluorescence, you see that loss of the eraser. You have lower levels of phosphorylation for STAT1 and 2. And if you look at the ISG mRNAs, pretty much you see a loss in signal for the mRNAs for all the ISGs that we looked at. The only one we really didn't see much of an effect was in the IFI27, well, in the LCLs, but in ACATA, clearly we saw a loss of that. So to just summarize here quickly, we think that what we have here is a feedback mechanism where in latency you have regulation of interferon alpha and beta through the interferon receptor, activating type 2 and DTX, then regulating the ISGs, so driving the immune response. In reactivation, you block in that through type 2 and DTX through M6A methylation, and this leads to loss of the regulation of the signal through the ISG compartment and blocking the antiviral response, and that leading to degradation, to proteasomal degradation of the TANG binding kinase, as well as the loss of the transcripts that we see here. But clearly, we think that there's a clear link to the epitranscriptome activation, methylation, and adenosine of the TYK2 and DTX4, and regulating the response. We don't know a number of things yet. We are still working on that, is which of the EBV antigens are involved, is it lytic or latent. There's some things we still need to iron out with this, but hopefully there's more to come in the future. And thank you, and I hope I was on time.
Thank you very much, Earl. So we're time for one or two quick questions on this. You'll just have to shout. Oh, no, there is a microphone here. I actually have a question.
Yeah.
Just, you used the CARTA cells, right, to study the... I just wondered if you had thought about using one of the EBV negative clones of a CARTA to see if there's any influence of EBV on these cell processes, which is what you're studying.
Yeah, we did some experiments with the EBV Akata negative cells, we saw a similar type of response. There's clearly EBV involvement in this direct, but in the Akata negative cells, we didn't see much of an effect. So clearly there is something EBV is doing here with the cell types. Yeah.
One question here. Oh, two.
Yes, there is.
Can you repeat the question?
There's a question was whether or not any of the EBV transcripts were M6A modified. Yes, the answer is yes, and there are clearly regulatory functions related to that as well. And earlier paper we published actually showed some of that work, and we are following up on some of that as well.
Okay. Thank you very much indeed. I think we have to, do you mind? We're over time. So.
It's okay?
Thank you. Thank you.
All good.
So in fact, the next speaker is Michael. I just stopped from asking a question, but very pleased to have you here. And Michael is talking about LMP1 forming a potassium selective oncochannel supports lytic virus release.
Can't stand and do this, right? Must sit. Okay. Buongiorno. Gracias, Lorenzo and Pankaj and the committee for inviting me to speak today. Thank you for the introduction. Where is my talk? No, that's the other talk. Macintosh got it. Excellent. Okay. So my laboratory at the University of Florida studies viruses in the context of immunocompromise and cancer. And while collaborating with the Padre Lab investigating how SARS-CoV-2, so the SARS-CoV-2, activates the inflammasome, we actually became interested in how viruses in general regulate ion channels and ion homeostasis in order to facilitate their own replication and pathogenesis. So first, a little bit about ion homeostasis in viral infections. The sodium-potassium gradient at the plasma membrane, maintaining a high sodium concentration outside the cell compared to a high potassium concentration inside the cell, represents one of the most profound biophysical properties of living cells. It shapes membranes, drives numerous molecular reactions, and in the case of neurons, it drives action potentials. So it's no surprise that viruses have evolved mechanisms to exploit ion homeostasis for their own purposes. In fact, many viruses will encode their own ion channels, and these are collectively known as viroporins. So, and as we found with the beta coronaviruses like SARS and SARS-CoV-2, they actually encode 3 ion channels, at least 3. One of these is ORF3 or ORF3A in the case of CoV-2, and that causes potassium efflux, which contributes to activation of the inflammasome. So we asked the question, might Epstein-Barr virus also regulate potassium efflux in facilitating its own virus release, similar to the beta coronaviruses? So to do this, we first established a FACS-based assay to measure potassium transport at the plasma membrane. That's shown up here, where increased potassium transport gives us an increase in fluorescence or a shift to the right, shown in blue, when we're using a potassium-specific ionophore, in this case, valinomycin. So when we try this assay on EBV-infected Burkitt lymphoma cells, and which are a great model system because they're in latency and they can be activated to go lytic. So we can activate our cells with sodium butyrate, which is a histone deacetylase inhibitor. And over the course of the lytic phase from 24 to 72 hours, we see an increase in potassium transport, culminating in the maximum potassium transport at 72 hours, which is coincident with virus release. So this was intriguing. So we wanted to see if how this potassium transport that we're measuring is really affecting, or if it affects replication of the virus at all. So we can actually block potassium efflux by simply raising the concentration of potassium in the extracellular medium from 5 millimolar to 50 millimolar. And when we do that, we actually see, when we look at intracellular EBV genomes after lytic activation with sodium butyrate, we actually don't see any change in viral genome replication at all. However, when we look at DNase-resistant viruses that are in the medium at 72 hours, we see a dramatic drop when we block potassium efflux. So blocking potassium efflux seems to inhibit virus release. So this was really intriguing. And so we decided we would look to see perhaps, or ask the question, maybe EBV encodes its own virus channel or its own ion channel. So we did an informatic search of the EBV membrane proteins against known ion channels. And we actually came up with hidden Markov model similarity to HIV-1's cationic channel viral protein U, which is a known viral porin, and it's just cationic specific, so sodium and potassium. And that had similarity to EBV's latent membrane protein 1. So latent membrane protein 1 is perhaps one of the most well-studied proteins in EBV. It's obviously associated with the latency. So it's one of the 9 or so latency proteins. And it's got this configuration of 6 transmembrane spans, a cytoplasmic amino terminus and carboxy terminus. It's known to function as a sort of a mimic of the B cell co-stimulatory molecule CD40, except that it's constitutively on. This is through these CTAR elements in its cytoplasmic tail. This, of course, is the motif that we found similar to HIV's viral protein U. So, but these CTAR elements actually signal through TRAFs and TRAD to activate multiple signal transduction pathways that drive cellular gene expression, namely pro-proliferative and pro-survival genes in the host, which contribute to it being actually one of the most potent oncogenes or oncoproteins encoded by EBV. So it contributes to both transformation and tumorigenesis. So we wanted to understand its structure a little more to see if it actually looked like an ion channel. So there's no real crystal structure available for LMP1. So we ran it through a neural network-based modeling algorithm. This is called AlphaFold2. It's a recent algorithm, I think, developed by Google. It's extremely efficient in predicting 3-dimensional structure of proteins just based on their primary sequence, which is amazing. AlphaFold2 is unable to resolve the structure, however, of the amino and carboxy terminal tails, but it gave excellent model agreement between different models of the transmembrane domains, which if we look at them sort of end on from the cell out of the cell, so this would be looking inside the cell out, we see that they're not a linear configuration. They're actually a semicircular configuration, almost pore-like, or a portion of a pore. So it is, of course, the latent membrane protein 1. So, and we're looking at lytic activation in the lytic phase and how, potassium efflux may contribute to lytic replication of EBV. So the nice thing about the Burkitt lymphoma system is that because of the type of latency that it's in, LMP1 is not expressed at baseline. And so when it gives us the opportunity when we induce it to look at the expression and functions of LMP1. However, when we look at the literature, the known functions of LMP1, despite its long history of being studied, is quite controversial within the literature, even to the extent is it really expressed at full length or not. So we induced Burkitt lymphoma cells in the lytic stage and FACS sorted them. So these are latent or refractory cells that did not lytically induce versus cells that did lytically induce. And when we look, we see that we do find the 63 kilodalton protein, which is consistent with full-length LMP1 expression in lytic Burkitt lymphoma cells. And when we look at its localization early in the lytic cycle, it appears to be more perinuclear, but then late at 72 hours, it appears to be aggregated at the periphery or the plasma membrane. So this was all very intriguing. It sort of fit our hypothesis. So we turned to a potassium channel expert. This is Professor Harpreet Singh at Ohio State University, to see if he could help us demonstrate potassium conductance by LMP1. So when we transfect LMP1 into 293 cells, now these are human embryonic kidney cells, but when we transfect them with LMP1, we actually do see potassium-specific conductance of 105 picosiemens, which is the unit of conductance. And from these tracings on the right, you can see that the conductance is actually at negative, when negative membrane potentials are applied, so that means it's voltage gated. And when we use different ions, it appears that it's only potassium that's being transported. So it's potassium specific and it's voltage gated. Now, of course, as I mentioned earlier, all of the functions of LMP1 to date have been ascribed to this cytoplasmic tail, and it functions to turn on cellular gene expression. So it could certainly be turning on cellular ion channels, and that's been hypothesized in the past. So we figure that if we remove the cytoplasmic tail, if that's the case, we should lose potassium transport. However, when we remove the tail, we actually augment potassium transport. And moreover, we see potassium conductance not just at negative membrane potentials, but also at positive membrane potentials. So it's lost its voltage gating, and the channel appears to be constitutively open. So in summary, EBV positive Burkitt lymphoma cells demonstrate increased potassium transport as the EBV lytic phase progresses to the stage of virus release. Blocking potassium efflux prevents virus release. Full-length LMP1 is expressed in lytic Burkitt lymphoma cells and localizes to the plasma membrane, and LMP1 forms a selective voltage-gated potassium channel, and its regulation is in its cytoplasmic tail. So these are the members of my laboratory. I just want to highlight these three who did most of the work on this project. This is Dr. Ibukun Akinyemi, postdoctoral fellow, Tiffany Fry, now Dr. Tiffany Fry, who just recently defended her thesis and actually has a poster here this week. Jennifer Mitchell, a talented undergraduate student. We collaborate on EBV projects very closely with the Paduri Lab. Harpreet Singh's our electrophysiologist at Ohio State. Jensen Jeng at NHLBI is helping us with looking at the higher order structure of LMP1. And these are our funding sources. Thank you very much.
Thank you, Michael. A question over here, please.
Thank you for this really thrilling talk. So my question is, LMP1 oligomerizes. Did you have a chance to look at mutations that are known to mediate inter and intramolecular interactions, whether they affect the channel function of LMP1?
Yes, there is a cysteine. I believe the cysteine is within that cytoplasmic tail or close to the motif that we identified, and I believe our mutation removed that cysteine. That's known to be involved. I think it was Jennifer Marshall's work who had shown that was involved in higher order structure, maybe tetramers of LMP1, which is known to aggregate at lipid rafts in the plasma membrane. So, but if it was critical, and if that aggregation was critical for the channel function, we would expect it would diminish it. But when we deleted that entire tail, it opened it up. So I think we won't know the answer until we actually understand more carefully what the higher order structure of LMP1 is as a multimer.
Okay, thank you. So we've got Pankaj and also Shankar on the top. There's another question, please.
Great talk. Thanks. I have a question about that Western blot that you showed, where the lytic LMP1, what we know as lytic LMP1 is the lower band.
Yes.
And that was almost insignificant expression there. So do we need to change now the nomenclature? I mean, because I would have expected that the lower LMP would be
more. So the literature is very confusing, basically because it's such a long history. I think if we look chronologically, there are several papers out there that now identify full-length LMP1 being expressed in Burkitt lymphoma cells of different types. And the lytic LMP1 that is referred to as sort of an earlier finding within the research of LMP1, and that appears, that I believe comes from a promoter, like an internal promoter element. So it's actually, the deletion would be on the amino terminal side. And for all the Western blots, at least our antibodies are to the carboxy terminus. So, so maybe using different antibodies that recognize different size may answer some of those questions. But I mean, I think it's quite possible that lytic LMP1 coming off of a different promoter may actually have independent function during the lytic phase.
Last question up there.
Yeah, it does retain the last 3 transmembrane spans. At least the last transmembrane span is retained. So it's possible that it might. That's one of the things we're really interested in, is can we express the lytic LMP1 specifically and see, but it only loses, I believe, the first 2 transmembrane spans. So, but it's also quite interesting. The size of LMP1 is a bit confusing because based on its primary amino acid sequence, you would expect it to only be about 42.5 kilodaltons. Yet its full length is recorded in the literature as 63 kilodaltons. So there's clearly a lot of modification to LMP1 that has yet to be elucidated.
Thank you very much. That's great. Nice talk as well. Thank you. So I understand that Francesco Baccianti is not here. So we're going to go straight on, keeping it in the family. And Samita Badhuri McIntosh is the next speaker.
Good morning. Thank you for that introduction. And I'd like to start with by congratulating the local organizing committee for pulling off this feat, and also thanking them for this opportunity to share some of our new work, which was all done by Huanzhou Xu, shown here and here. He's a postdoctoral fellow in my lab. And this project has to do with the lytic phase of Epstein-Barr virus. As we know, the herpes virus lytic phase is characterized by an ordered cascade of events, which begins with transcription of immediate early and early genes, followed by genome replication, and then post-replication events. Now, the first 2 of these events use the circular viral genome or episomal genome as template. We also know that as replication of the viral genome ramps up, immediate early and early gene expression reduces. So the question then was, how does the virus transition from transcribing these immediate early genes to replicating the circular genome when it's using the same circular genome? So in order to address this question, Huanzhou did what's called an iPond assay, isolation of proteins on nascent DNA, where he pulls down replicating genomes. So these are viral genomes that he's pulling down during the lytic phase. And then we looked at all of the proteins that were pulled down along with those replicating genomes by mass spectrometry, and identified a number of known candidates or known proteins, viral proteins, that are supposed to be at these replicating genomes and functioning in replication. as well as a couple of unexpected proteins as well. But one of the biggest surprises was this protein, a cellular protein called CAP1 or TRIM28. This is a protein that my lab has been studying for the last number of years in the context of heterochromatic silencing of viral lytic genes in order to maintain latency. This is a protein that also silences the pericentromeric regions and transposable elements, as well as endogenous retroviral elements of our chromosomes through the use of the constitutive heterochromatin machinery. However, CAP1 has not really been reported to be at the replication fork. So what is it doing at viral replication forks was our question. So I'll give you the punchline first in the form of a model, and then show you some select pieces of evidence to support this model. And what we think might be going on is that just about when transcription has peaked and replication begins, you have both these machineries. You've got the replication machinery, transcription machinery, all in the circular genome. Quite possibly some of these collide initially, but then what we think is happening is that these two machineries recognize the presence of the other. So there is some sort of sensing of the proximity of these machineries by each other, which then causes this protein called RECQ5, which typically travels with RNA polymerase II. So RECQ5 is a cellular protein. It's a DNA helicase, which causes recruitment of CAP1. And then CAP1 is phosphorylated by ATM, which is a cellular kinase. We then think this phosphorylated CAP1 kind of moves over to the replication side of the story, and then of the model, and then it causes simulation of early antigen diffuse. So EAD is a virolytic protein, and it also functions as a DNA polymerase processivity factor. So once EAD is simulated, it then recruits CAF1A, which is a histone chaperone, commonly associated with silencing histone marks, and causes recruitment of silencing marks, thereby silencing transcription from that genome, and as a result, prioritizing replication over transcription. So in order to help understand what's going on, I have put this little cartoon in the corner of every slide as we go through some of the data. So this is an IPOND experiment. In this case, we're doing a Western blot instead of a mass spectrometry, of course, and we're looking at CAP1 as well as phosphorylated CAP1 at serine 824 on replication forks shown here in Burkitt lymphoma cells as well as lymphoblastoid cells, both lytically activated. We do not see this protein, either phosphorylated or total protein, when we look at genomes where we block replication using phosphonolacetic acid, or when we look at newly replicated mature chromatin by chasing with thymidine, or when we block IPOND, the IPOND step itself. So CAP1 is on actively replicating EBV DNA. Is there an effect on replication? It turns out there is. Here we're looking at a plasmid-based replication assay, which uses one of the origins, lytic origins of replication. And if we deplete CAP1, we see less replication. And if we overexpress CAP1, we see the opposite. It turns out then that CAP1 is a SUMO2 ligase. And knowing that, we looked informatically at the proteins, the viral proteins that are known to be at replication forks. And EAD, early antigen diffuse, emerged as a top candidate as a SUMO target, and that it has two putative residues that could be simulated. Does it indeed get simulated? It turns out yes. So we introduced tagged SUMO2, EAD, and flag-tagged CAP1 into these 293 cells, and we found that EAD was simulated. If we also introduced a SUMO peptidase, we lost the simulation. What about the residue at which EAD is simulated? If we, instead of introducing the wild-type EAD, if we introduce the first mutation of the first residue, lysine 135, we saw substantial loss in the simulation, but not so when we use the other mutant. So CAP1 conjugates SUMO2 to EAD at lysine 135. CAP1 also interacts with EAD And this is looking at proximity ligation assay, and this essentially demonstrates that these two proteins, CAP1 and EAD, almost next to each other, so they're adjacent, and they're adjacent in lytic cells as marked by zebra expression. Interestingly, if we block replication using PAA, we lose this proximity of these two molecules, so CAP1 and EAD do not appear to interact when PA is used, and we see the same thing with co-IP experiments. If we pull down CAP1, we lose EAD pull down simultaneously in the presence of PAA. So interaction between CAP1 and EAD is dependent on DNA replication. What is the effect of simulation of EAD at lysine 135 on DNA replication? So this is an experiment where we used a BMRF1 or EAD knockout 293 BACMID, which was obtained from Henri-Jacques Delacluse, and these were introduced into 293 cells. If we activate the BACMID into the lytic phase using zebra and RTA, and of course wild-type EAD, we see plenty of replication of the viral genomes. But that is substantially impaired when we use the lysine 135 mutant of BMRF1, but not so with the other mutant. And this part simply shows that pCNA, which is a cellular homologue of EAD, depleting pCNA has really no effect on DNA replication. So simulation of EAD at lysine 135 ensures maximal DNA replication, so we're still looking at this part of the model. What is the effect on replication forks themselves? So here is a DNA fiber fluorography experiment where we're essentially pulling down single molecules of DNA. These are stretched out and they're fluorescently labeled. And so you can see actively replicating DNA, not all DNA. And you'll observe that when CAP1 is depleted, we have fewer fibers, so fewer DNA fibers shown here. This is aggregate data for multiple fields. We also observed that when we depleted CAP1, these fibers were shorter in length. So in other words, these DNA forks or replication forks were collapsing and DNA was breaking. And this is supported by these Western blots here, where we have depletion of CAP1 showing increased phosphorylation of H2AX. So we see the same thing when we have the lysine 135 mutant of EAD. So CAP1 and EAD maintain integrity of replication forks, and CAP1 prevents DNA replication at replication forks by simulating EAD at lysine 135. Now, in interactome studies, CAP1 has been found to interact with this DNA helicase called RECQ5, which typically travels with the RNA polymerase 2. Does RECQ5 have an effect on CAP1 phosphorylation or EAD simulation? And the answer is yes, it does. If we deplete RECQ5, we see less phosphorylation of CAP1. We also see, in this longer exposure, we see less simulation of EAD, lanes 2 versus 4 when RECQ5 is depleted. What we also observed was that there was less replication of the viral genome, as well as fewer transcripts of early and immediate early genes coming off of the circular genome. You will recall that knockdown of CAP1 and loss of EAD simulation results in DNA breaks, and we believe it's likely due to fork collapse caused by collisions with the transcription machinery, and I'll get back to that in a moment. So how is CAP1 phosphorylated? So a number of years ago, my lab showed that CAP1 is phosphorylated by ATM, a cellular kinase, and ATM, so if we inhibit ATM activity using this compound, we see less phosphorylation of CAP1. We also see less simulation of EAD, shown again lanes 2 and 4. If we deplete ATM, again we see reduced phosphorylation of CAP1, and we also pull down less phospho-CAP1 if we precipitate RECQ5. So if you're focusing on this area of the model, you'll see what I'm talking about. If we deplete ATM, we similarly see less interaction between CAP1 and the transcription machinery, specifically RNA polymerase 2 and RECQ5. So ATM causes phosphorylation of CAP1 at the RNA polymerase 2 RECQ5 complex, and it also seems to impact the recruitment of ATM itself, recruitment of CAP1, sorry. possibly through ATM, not sure how that happens. Now, you'll also recall that CAP1 directly interacts with EAD and is that replication fork. So what's potentially happening is that this phosphorylation of CAP1 causes it to migrate over to the replication complex, causing it to then simulate EAD and the rest of the story. Lastly, then, what are the effects of RECU5 facilitating CAP1 phosphorylation and simulation of EAD on transcription on these genes, on these early genes? To look at that, we did a CAF1A, so we did qPCR experiments, CHIP qPCR experiments, where we pulled down CAF1A and H3.1. So we're looking at enrichment of CAF1A and H3.1. CAF1A is a histone chaperone. H3.1 is a histone molecule. Both of these are recruited to DNA following DNA replication. So these are DNA replication dependent recruitment, and they're often associated with silencing marks on histone silencing marks. So we're looking here at enrichment of H3K9 trimethyl as well. And if we deplete RECU5, we find that there is less recruitment of these molecules, or enrichment of these molecules at immediate early and early gene bodies, specifically on the circular genome, because we look at the genomes after digestion with MBO1, which chews away the linear genomes, the unmethylated genomes. So what we believe happens then is RECU5 is needed to stop the transcription machinery and prevent collisions with the replication machinery. To summarize, EBV uses mechanisms that avert transcription and replication collisions to orchestrate the handover from transcription to replication of the circular or episomal genome, ensuring an ordered cascade of events during the lytic phase, and heterochromatinization of the circular genome during the lytic phase is a pretty dynamic process, in that while heterochromatinization is important to maintain latency and that those heterochromatin marks have to be removed in order to begin early and immediate early gene transcription, silencing marks have to be reinstated in order to transition from transcription to replication. With that, I will stop, and I thank you for your attention. Happy to take questions.
Thank you. And there's two questions at the back.
Hi, Sumita. Very nice talk. Just a quick question about simulation of EAD. Have you seen any interactions or lack of interactions as a result of simulation with other viral components of the replication machinery?
We haven't looked yet. We've been focusing mostly on EAD at this point, and also, primarily because EAD was the top candidate. I suspect, and we know that there are other candidates that are also simulated, and how EAD simulation might interact or prevent interaction with other molecules is clearly an important question. Haven't looked at those yet.
Okay, thank you.
Hi, Sumita. That was a really nice talk, really interesting. I'm just wondering, the CAP1 is known to form a ternary complex with TRIM24 and TRIM33, at least some of it does. So I'm wondering if you found these other TRIMs in your iPod data, and whether you think CAP1's acting independently or as a complex with these other TRIMs in your studies.
That's a good question. Trying to think, I don't recall seeing other TRIMs. I will go back and get back to you on that. But I really don't recall any other TRIMs, really.
Okay, no problem. Maybe we can chat about it later.
Yeah, we can chat about it. Yes, Eric. Yes, great question.
Repeat the question. Yeah.
So the question is, how does the homologue for EAD, cellular homologue for EAD, pCNA, and its own simulation, which is known to have similar kinds of functions, how do those functions compare with the functions of EAD? So there are two papers thus far out from a group in California that it almost sounds like I planted the question. So, but I didn't. So that group showed that in the context of transcription replication complexes or collisions, in particularly the fragile areas of the cellular genome, pCNA gets simulated, and that simulation happens via CAP1, and it then causes dissolution of the RNA polymerase complex. And so this is similar, very similar. That said, in those papers, they haven't yet looked at how it is that CAP1 accomplishes it. Does CAP1 need to be modified? How does it get modified? Does it actually travel to the replication complexes, or is it able to mediate its function? Or how does RECQ5 even recruit CAP1? Or how does it communicate with CAP1 in order for it to talk to pCNA? So, and does CAP1 need to be phosphorylated in that context? None of that is known. How the RNA polymerase complex is dissolved is also not understood. In this case, we find that the silencing marks and silencing histones, histone chaperones, appear on the viral genome, but we interestingly notice that also the activating marks on the viral genomes remain constant. They don't go away. So yes, I don't know if that answers your question. Somewhere up there, I have no idea where. Great.
Okay, it's working now. So thank you very much, Sumita. I was wondering whether...
Just shout it out, please. So, yes, it's a regulator, but CAF1A is thought to be the central player of CAF1. That said, CAF1B may play a role. We did not pull down CAF1B in our IPON mass spec, but that does not preclude its presence because the sensitivity is only as good as how good your pull down is and how good your mass spec is. So it's something that we need to look at. Thank you.
Thank you very much. It's very nice talk. So,
one last question.
I'm just going to slightly correct what I said earlier. So Francesco Baccianti will present his talk tomorrow. In fact, it's not that he's...
Later on, in the next session.
Michelle knows what's happening. She's taking over anyway.
Yeah, he's giving his talk in place of Gillian Bristol, who will be talking later on. So we move on now to our next speaker, who is Ana Catalina Blázquez, who is going to be telling us about could EBV type 2 asymmetric circulation be explained with an immunoinformatic approach.
Okay, thank you for the introduction. EBV1 and EBV2, which mainly differ in the IBNA2 gene and IBNA3 family of the gene, vary among geographic regions. EBV1 were cosmopolitan, and EBV2, and EBV2 male prevails in Africa and Papua New Guinea. Moreover, the incidence of EBV-related malignancies and their association with EBV vary among geographic regions. Over the last few years, and with the advent of next-generation sequencing, more than 1,000 EBV complete genomes have been sequenced. In our previous work, we showed that EBV type could be discriminated using the first component of principal component analysis. However, what happens when we color each of these sequences in accordance to the geographic origin of the isolate? We can confirm EBV1 comes from all geographic regions, while EBV2 mainly comes from Africa, Argentina, and Asia. When we analyze the second component in an independent way, we could see that this component clearly segregates Asian sequences from the rest of the world. In parallel, worldwide variation of human leukocyte antigen was also being described. Some alleles show a marked geographic distribution, and others are cosmopolitan, like HLA A0201. Given this geographic difference in the circulation of EBV type, we wonder if this could be due to a combination of viral immune escape based on variable epitope and immune surveillance based on different HLA population frequency. So our aim was to study the variability of epitopes based on viral type, geographic segregation, and pathology, and to relate their variability with HLA class 1 population frequency. In order to answer this question, we constructed a well-balanced based on geographic origin and EBV type dataset. We selected 40 samples from Argentinian pediatric patients with high viral load for next-generation sequencing. Sequences libraries were prepared using Schwartz select target enrichment, and then their sequencing using NextSeq 500 equipment. In parallel, 246 FASTQ raw data were downloaded from NCBI database. These FASTQ represented isolates from different geographic regions. All FASTQ were analyzed with the same informatic pipeline based on a mapping strategy. And finally, we construct a MSA composed by 211 EBV1 sequences and 75 EBV2 sequences. In particular, considering the new isolates from Argentina, 29 were EBV1 and 11 EBV2. For the immune informatic analysis, we selected 4 EBV proteins. We used immune epitope database, and we selected EBV lineal epitopes with restriction. The search was also limited to epitopes recognized by T-cell MHC class 1 molecules with documented positive laboratory assays. The resolution of the recognizing was to restrict to 210, and after that, the conservation status was assessed on our entire data set. Only epitopes with less than 90% conservation were considering for the HLA coverage analysis. Okay, we found 5 epitopes for GP350, but only 2 were variable, so we carried on our HLA coverage analysis with these 2 epitopes. The phylogenetic tree for GP350 depicts EBV type segregation, EBV1 and EBV2. The color of the sequence name reflects the geographic origin as shown on the map. The first layer of black and white squares refer malignant status of clinical isolates. Second red and third blue layers highlight the sequence with variable epitopes. Okay, we can see epitope in red is variable in all EBV2 sequences, but in non-EBV1 sequences, this variability is not random. This epitope was recognized by HLA A2402. This allele has the highest population frequency in Asia, Australia, and South America. So the coexistence of variable epitope and high population coverage of this HLA allele may facilitate immune evasion and could provide a hint for the higher circulation of EBV2 in Argentina and Papua New Guinea. Blue epitope was variable in all EBV2 sequences and some EBV1 sequences from Indonesia. This epitope was recognized by HLA V1401, and this allele has a high population frequency in Asia and Australia. So here again, the coexistence of variable epitope and high population coverage of this HLA allele may facilitate immune evasion for EBV2 in Asia and Papua New Guinea. We know CP350 epitope variability was related to EBV type, but is not related to malignancy. 9 epitopes were found in IBNA1, but only 6 were variable. On the phylogenetic tree, we can see that epitope variability was the higher in African and Argentinian sequences and lower in all other regions. All of these epitopes were recognized by HLA A0201. This allele has a worldwide higher population frequency. IBNA1 epitope variability was not related to EBV type and malignancy. 11 epitopes were found in LMP1, but only 4 were variable on the phylogenetic tree. Okay. In the phylogenetic tree, we can see that epitope variability was high. In particular, epitope in red is variable in EBV2 and EBV1 sequences from Papua New Guinea, Indonesia, and Argentina, and EBV2 sequences from Asia. This epitope was recognized by HLA A2402. So here the coexistence of variable epitope and high population frequency of this HLA allele may facilitate viral immune escape in Argentina, Indonesia, and Papua New Guinea. But on the other hand, the lower frequency of this allele in Africa could not explain this viral immune escape. Here again, LMP1 epitope variability was not related to EBV type and malignancy. Finally, we analyzed CETA. 18 epitopes were found in CETA, but only 6 were variable. On the phylogenetic tree, we can see that epitope variability was higher in EBV2 sequences and EBV1 sequences from Asia. For example, epitope in red is variable in all EBV2 sequences, and purple epitope is variable in EBV2 sequences from Africa and Argentina, and EBV1 sequences from Asia. Most of these epitopes were recognized by HLA A0201. In particular, yellow epitope is variable in Africa, Argentinian, and East Asian sequences independent EBV type. This epitope was recognized by HLA V1501, and this allele has a high population frequency in East Asia. Regarding disease, CETA is the only protein to display statistical difference in epitope variability between malignant and non-malignant samples, where variability is higher in non-malignant samples. In conclusion, among the studied viral proteins, epitope variation in CETA and CP350 was more related to EBV type and geographic origin, but not in IBNA1 and LMP1. Epitope variability within CETA protein in non-malignant samples could favor lytic cycle reactivation through viral immune escape. Although these are still preliminary results limited to 4 EBV proteins, epitope variation in CP350 and CETA in EBV2 from Argentina, together with the loss in recognition by high-frequency HLA alleles, could explain, at least in part, the higher prevalence of EBV2 in our country. As future perspective, given that the notion of immune escape of EBV2 depends on several antigens in relation to HLA frequencies, we are currently enlarging the set of studied proteins to elucidate the observed high EBV2 circulation in Argentina. Thank you very much.
Thank you. Do we have any? Okay, we have a question over here.
So, thank you very much. It was a great talk. I have a question about, you said just 4 proteins. So
Yes.
EBNA4, that was one of the first proteins to be recognized having mutations in, I think, HLA-A2 predominant countries. So have you looked at EBNA4? Are you planning to investigate other proteins than those 4 that you have shown?
Yes, now we are studying other proteins like IMNA2, IMNA3A, IMNA3B, C, LMP2, and some lytic proteins.
Could you just repeat that with the microphone, please?
I'm confused about how you define an epitope for this particular study. How are you determining that the particular peptide sequence? What assay are you using?
Sorry, can you repeat?
How was the epitopes defined? I think you used a database of epitopes, did you?
I don't understand the question.
Yes, please do.
Te pregunta cómo definiste el epitope.
Okay. We use immune epitope database. Here, different groups study different epitopes, and you can see different positive and negative epitopes to MHC ligand, T cell recognition.
Okay, thank you. So, Christian, we have a question here.
Very interesting. I was wondering, it seemed like you were arguing that there is mainly immune escape from T cell recognition of the lytic cycle, maybe, or better correlation, right?
Yes.
Than from the latent. What is your hypothesis? Is there less immune control required for latent infection?
Yes, probably is for a better immune escape, but now we see other lytic proteins are very, or have epitopes very conserved. So I don't know. It's a good question.
Okay, thank you. Thank you very much. So we move on to our next speaker, who is Shankar Swaminathan, who is going to tell us about effects of RAD21 and CTCF on EBV reactivation from latency.
Thank you for giving me the opportunity to speak here today. That is a picture of our campus in the University of Utah. It's actually not quite that green right now. All right. So as you probably know, CTCF and the cohesin complex are host cellular proteins that affect the conformation of chromatin and its 3-dimensional structure. And both of these proteins are known to also regulate herpes virus transcription, and this has been shown for HSV and KSHV and EBV by a number of groups. We had previously shown that both CTCF and RAD21, which is a cohesin component, act as potent restriction factors for KSHV reactivation and lytic replication. And this is at the level of transcriptional control. So in this study, we did a similar investigation of the role of these factors in reactivation of EBV from latency. So this is just a recap of some of the relevant data from a previously published paper of ours where we did depletions of RAD21 and CTCF and looked at what happens to KSHV lytic gene expression and replication. On the left here, this is DNA replication, and this is done after induction of the lytic cycle by exogenous expression of the OR50 RTA gene of KSHV. And you can see here that under those conditions, depletion of CTCF and particularly of RAD21 lead to clearly discernible increases in the amount of DNA replication, and similarly on the amount of infectious virions that are produced and released into the supernatant. And over here is just a Western blot that shows the independent and efficient depletion of these two proteins. So when we did this similar experiment in EBV, we set up this system where we used AGS cells, which are a gastric carcinoma-derived cell line that is stably infected with a recombinant GFP-expressing Akata virus from Lindsay Hutt Fletcher. And in this cell line, we stably introduced an inducible ZTA-expressing lentivirus and selected clones that would efficiently and robustly enter lytic cycle on the addition of doxycycline. So we took those cells and we either induced them or didn't induce them by addition of dox, and depleted, we actually first depleted either mock depleted them or depleted CTCF and RAD21 specifically, and then harvested DNA, RNA, and viral supernatant to examine the effect of these knockdowns on DNA replication, on the viral transcriptome, and on infectious virus production. And somewhat to our surprise, we found that although there was good induction of lytic replication and infectious virus production, knockdown of these proteins or simultaneous knockdown of them did not really appreciably affect virus production. In fact, it might even have decreased it a little bit. And again, here's just the Western blot that shows the depletions. So we were somewhat surprised at that, but we asked whether there was a similar increase in lytic gene expression when we had done these depletions as we'd seen with KSHV. And in fact, there was. You can see early genes here that go up more when you deplete RAD21 particularly. And this is an early gene transcript. This is a late gene transcript. And you can see in all these cases that the expression of these lytic genes at the RNA and protein level is in fact increased by the knockdown of RAD21. So we asked where this inability to produce more virus might derive from. And when we looked at DNA replication, you can see that while it's not dramatic, if you deplete RAD21, you do get a significant increase over just inducing with RTA in the amount of viral DNA that's produced. However, the amount of extracellular DNA representing virion particles was not increased. So this suggested that there was some block subsequent to DNA replication in either encapsulation or egress that might explain this bottleneck, if you will. So we did, I don't know if you can see them up there, but there are capsids here. And we looked at the number of capsids per cell and the number of capsid positive cells. And both of these were in fact increased by RAD21 knockdown. And so we concluded from this that the most likely explanation is that even though RAD21 particularly does repress maximal KSHV transcription and DNA replication, that removal of this repression does not lead to increased extracellular virus, possibly due to the fact that we've maxed out the ability of the cell to make some late gene products that are necessary for virion production. So we asked whether we could see an effect on virus production if we didn't supply this overexpression of ZTA by from the inducible lentivirus. So, and I should mention that when we did these experiments in KSHV, just depleting RAD21 in the absence of exogenous transactivator did not lead to spontaneous induction of the lytic cycle. So, however, when we did that experiment with uninduced cells, that is, cells not provided the lentiviral ZTA, we did see induction, particularly again with RAD21, of the lytic cycle, increased virion production, DNA replication, extracellular viral DNA. And we did see that here, even though the lentivirus is not turned on, that we get ZTA expression by knocking down RAD21. And maybe a little bit with CTCF, but primarily with RAD21. So we went on to investigate this further. And you can see here, now these heat maps show in the leftmost column, cells induced with doxycycline, i.e. lentiviral ZTA, and then here, uninduced cells that are either depleted of CTCF or RAD21 or both. And you can see here that if you remove RAD21, you get this really almost as good induction as you do with the overexpression of ZTA. And you can see here that this correlates with an induction of the endogenous. This is measured by primers specific for either the viral BZLF1 gene or the lentiviral ZTA. So knocking down RAD21 leads to de-repression of the viral ZTA, and at least in part explains this very global induction of lytic gene expression in viruses where the RAD21 is knocked down. And it doesn't, as you might expect, have that effect on the lentivirus. So we did ChIP-seq, and this is a zoom in of the region of the BZLF1 gene, and you can see here that there is occupancy of this region, promoter and gene body, by RAD21. And while knockdown of CTCF may reduce this somewhat, it doesn't eliminate it as does the specific targeting of RAD21. And shown here is by qPCR, a measurement of the occupancy in this region of RAD21 here. And you can, as you might expect, with the knockdown of RAD21 using siRNA, that occupancy is reduced to background. And concurrently, the occupancy of this region by RNA pol2 increases, indicating that the presence of RAD21 prevents transcription either directly by promoter occlusion or indirectly by modifying chromatin conformation. So we wanted to see whether we could demonstrate this effect of RAD21 on in a more physiological system. And Cornelia-de-Lange syndrome is a so-called cohesinopathy, which is caused by mutations in a variety of genes that either comprise the cohesin complex or in genes encoding proteins that modulate these components, such as HDAC8 and so on. So lymphoblastoid cell lines have been obtained from these CDLS patients by in vitro transformation with EBV. And we obtained some of these cell lines and examined their patterns of lytic EBV gene expression. And what you can see here is 3 different patient cell lines compared to normal patients or normal volunteers, LCLs. And as is typical of most normal LCLs, they're fairly tightly latent, and there's little spontaneous expression of immediate early and early proteins on this Western blot, but there is spontaneous expression of ZTA, RTA, and EAD in these three different cell lines that have different mutations in that affect cohesin function. And over here, this is sorry, this should say RNA here. This is an early gene and a late gene, and we look at spontaneous expression of these transcripts, again, in these LCLs from normal people and people afflicted with Cornelia-de-Lange syndrome. So to look at whether, to confirm that this is really due to the defect in cohesin function and not due to chance, we rescued RAD21 function in this cell line with a specific mutation that results in hypomorphic RAD21 function. And what we did here was we made a lentivirus that will inducibly express wild-type RAD21 and transduce these cells with that lentiviral construct. And what's shown here is two polyclonal pools of LCLs that was one LCL, but it's a pool of transductants that were selected for wild-type RAD21 expression. And you can see here that a variety of early and late genes, their expression goes down when we turn on RAD21 expression by inducing the lentiviral construct with doxycycline treatment. And over here is a single clone and these pools again with proteins, Western blots examining some of these proteins that we have antibodies for. And you can see here the effect is consistent that expression of RAD21 rescues this phenotype. So I just wanted to say that Wenmin Fu here was a postdoc in the lab who did most of this work with assistance from Dinesh Verma. And thank you for your attention.
Thank you. Do we have any questions? So we have a question here.
Very nice. I was wondering, is there anything known about deregulation of infection in the patients that suffer from this Cornelia-de-Lange syndrome?
Is there anything known that there might be a consequence of having inefficient establishment of latency? No. And we are trying to look at what viral loads are in these patients at, under normal conditions, which shouldn't be detectable in most normal people.
We just have a question there, Paul.
Yeah, hi, Sankar. Very nice talk. Couple of questions, but so RAD21 cohesins can be very positive for transcription. They can support enhancer function, and you're showing them functioning as repressors. And I'm just wondering what you think the mechanism of repression is at the BZ, and only at the BZLF1 locus, or is it throughout the genome? And how do you think it's working?
So that's a very difficult question, but I think, the reason we went to look at its effect on ZTA expression is because we saw that in just depleting RAD21 led to increased ZTA. And really the effect that we see on the lytic transcriptome is extremely broad. And so at least part of it would be due, I think, to the fact that you de-repress ZTA, and that leads to increased DNA replication and fairly widespread activation of the lytic promoters. As to whether there's also a repressive effect on specific other lytic promoters, I just don't know, because I think this one is proximate to the others, right? So one could probably, I guess, investigate that question with a ZTA knockout and with RTA induction. You know, that depends on RTA induction only. And as to whether this is promoter occlusion of ZTA or whether it's secondary to some of the looping that you and others have shown occurs in latency, I don't know.
Thank you. So we have a question here, Sumita.
Great talk, Shankar. Two related questions. Are these, so do Cornelia de Lange patients, are they more susceptible to EBV-related diseases? Is it known? And the second thing is, how difficult or easy is it to establish LCLs from these patients?
So I'm sorry, how easy is it to transform?
You think if the virus is going spontaneously lytic, presumably even upon infection, new infections, it may take longer or it may be difficult to establish LCLs.
These cells aren't the most robust in the world because, you know, of their, I think, DNA repair defects and I don't know what else, but, you know, transcriptional problems. But so, yeah, they're not that easy to obtain, but you do get them. I couldn't really understand the first part.
The other question has to do with whether these patients have an increased risk of EBV-related diseases.
Increased risk of EBV-related diseases.
Yeah. So it's a good question. You know, their lifespan is shorter.
Right.
So I don't know if there might be an increased risk of lymphoma, for example, that has not been detected. But I'm not aware of any correlation with either B cell or epithelial malignancies in these patients.
Okay. Thank you very much.
You.
So we move on to our last speaker of the session, who is David Maestri, who's going to be telling us about Epstein-Barr virus rewiring the host genome through CTCF and POP1 relocation.
Okay. Good morning, everyone. Thank you to the committee for giving me this opportunity to present you my project that I recently started working on. So upon EBV infection of B cells in vitro, basically there is a change in a phenotype of the cells to a cancer-like phenotype that results in a alteration of metabolism and signaling pathways that leads to abnormal gene expression. So we were interested in basically if this abnormal gene expression is due to a different reorganization of the 3D structure of the genome, of the host genome, upon EBV infection. We focused on two proteins. One is PARP1, which has multiple functions in DNA damage repair, and it's also involved in regulation of transcription and DNA methylation. And as we recently demonstrated, it's also involved in maintaining latent transcriptional active program of EBV. And CTCF, which is a well-known architectural protein, and together with cohesin is involved in the organization of the 3D structure of the genome, both the human and the EBV one. So the experimental design is basically, we started with purified B cells from PBMCs, and then we basically infected them in vitro with EBV, and then we analyzed basically the transcription factor profiling with cut and run, the 3D structure of the genome with the in situ high-C and high-CHIP. We used also mass spectrometry to analyze basically the proteomic landscape of the cells, and we used also one from Ben Gavor's lab, available data set. we integrated these data with the RNA-seq data from the Hammerschmidt lab. And we were also interested in dynamic methylation using RRBS. Today, I will just focus on the transcription factor profiling and the 3D structure of the genome together with mass spectrometry and RNA-seq analysis. So basically, as you can see from these heat maps, we profiled the, I don't know why the images do not show up, but both basically CTCF and PARP1 localized at the transcription start site. It should be present here in the average plot, but I don't know why it doesn't show up. I'm just focusing on the day 0, so basically naive B cells, previous infection, and day 21 post-infection. So we focused then on those peaks who were basically common, both for CTCF and PARP1, and those peaks, we got almost 5,000 peaks that were lost upon EBV infection at day 21, and almost 2,000 peaks that were gained day 21 post-infection. We then analyzed, basically, we merged these data from cut and run experiments with the RNA-seq experiments and from the Hammerschmidt lab. And basically, we ran a differential expression analysis on taking day 14 as an example, basically comparing day 14 to our day 21. And we suppose basically that there is not a great difference in CTCF and PARP1 binding at these two different time points. And we ran an ingenuity pathway analysis of these data. And we basically found out some pathways that are known, for example, an FKB and HIF1 alpha. And also MYC and XBP1 pathways. So basically, these are upstream, obviously are the upstream regulators, so transcription factors that bind to these genes where also CTCF and PARP1 bind. And we did the same thing also for those peaks that were gained on day 21. And we found out that some of the, was interesting to see that, for example, MAX and IF1 alpha showed up both in the gain peaks and loss peaks. And it seems that these transcription factors were were helped in binding on their target genes by the rearrangement of CTCF and PARP1 binding throughout the genome. Again, I don't know why the images don't show up here, but we basically wanted to see whether the binding of these transcription factors is changed upon infection. Here, basically, we ran an experiment on chromatin-bound proteins, thanks to our collaboration with Simone Sidoli at the Albert Einstein Institute in New York. And basically, the proteins here should be ranked by their score, which gives an idea of how the magnitude of the change is and how significant that is. And basically, all the proteins are near 0, so that means that they do not change. They are binding to the genome at these two different time points, or it's the significance of this change is not high. So we then wanted to see basically if these genes are inside some of the CTCF loops. So we ran an iChip experiment, and here I'll just show you two different genes that are both present basically where CTCF basically binds both at day 21, so in the cut-and-run experiments, and also here The chip obviously was done on LCLs due to problems with cell numbers, but these cells come from the same donor that I use for the cut and run experiment. So basically, even though these two genes are basically bound by CTCF and are present in one loop formed by CTCF, ABCA2 results in an upregulation, while NDUFA4 result in is downregulated. And these data are both corroborated by the RNA-seq analysis and our proteomics that we run between B cells and LCLs. These led basically us to think about a model where basically the binding of CTCF and PARP1, the gain binding of CTCF and PARP1, probably due to proteins such as LMPs and DABNAs, determines a rearrangement of the structure of the genome forming loops, and that results both in a activation of some in an activation gene expression for some genes, and that basically here I just put, for example, an FKB, or otherwise it results on the other end in a gene repression, probably due to a trapping of an enhancer or a promoter inside one of these loops. And that is obviously true also the other way around. So for those peaks that are lost after infection, probably these determines basically determines the expression of these genes or not, thanks to the rearrangement of the 3D structure of the chromatin. So I want to thank my lab, my supervisor Italo, and in particular Georgia and Sarah, who helped me with the extraction of the proteins for the mass spec and the RRBS analysis that I wasn't able to show you today, but Sarah is still working on it. I want to thank also the Sidoli lab for their work on the mass spec analysis, in particular Sarah Graf, who ran our experiments, the genomic core and bioinformatic facilities from the Wistar Institute, and also my supervisor at the University of Bologna, Giovanni Perini. Thank you for your attention.
Thank you. Do we have any questions? So I guess I could ask you a question. So you did your experimental system was EBV infection of primary B cells, and then you looked at kind of 3D changes using Hi-C. Have you looked at kind of B cells, a control for B cells that are activated through some kind of physiological stimulus like IL-4, CD40 ligand?
Not yet. We were thinking about that, doing these, also expanding like the time points, so basically all the time course. But yes, we are thinking about it.
Because you might expect that a number of these structural changes would be the result of perhaps just B cell activation. So you can kind of try and pull out the EBV specific events. Do we have any other questions? Okay, so we have one here.
So CTCF can be either associated with TAD boundaries, cohesin complex, sometimes though just as a transcription factor and promoters regulating gene expression. So what proportion of those changes that you're observing are really truly associated with TADs and cohesin versus just more of a transcriptional regulation role?
Yeah, we are basically running also in parallel Hi-C experiments just to make sure that basically these changes are exactly on basically the TADS boundaries. But yet we don't know obviously if this is actually just a CTCF acting as a transcription factor or acting as a modulator of chromatin. We are just proposing this, but yeah, we have to look in deep of that.
Very nice work. Thank you.
Okay, thank you very much. So Thank you to all the speakers for that first session. So we now have a slightly extended coffee break, but if you could be back here prompt so that we can start the next session at 11:20. And the next set of speakers, please make sure they've uploaded their talks and check them. Can you please leave from the door at the bottom here?
Io sui grafici vorrei che sia troppo pesante. Tempera. La sezione dopo. L'ultima, quella prima del... Ah. Perfetto. Non lo so perché le tue, forse era pesante il file.
Le tue che problema avevano?
I grafici.
C'era dei problemi nella slide?
Sì, non si sono caricati i grafici. Ha caricato le coordinate ma non ha fatto vedere i figli.
Hai fatto un errore nel caricamento del grafico. Fai riferimento a qualcosa che è nel tuo computer che quando la esporti lui non vede.
Tu l'hai legata all'extra file? No?
No, c'è probabilmente un errore di collegamento.
L'abbiamo copiata come fatto
No.
lo screenshot.
Te sei? Sì, scusa.
All right. So welcome back. Please take your seats. We can start the next session. Please take your seats. Thank you. Right. Yeah. So the next session is on EBV latency program and reactivation. And I'll be chairing it with Lorenzo Leoncini. Unfortunately, Alberto Fagioni could not make it. So the topic lecture of this session will be given by Paul Lieberman from the Visitor Institute in Philadelphia. Control of EBV latency by EBNO1 in cancer and multiple sclerosis. Paul, thank you very much.
Hello, welcome back. I want to thank the organizers for putting together this really exciting opportunity to be together again in person, and thanks for the opportunity to speak this morning. So I'm going to talk about some work that we have not published yet, and it's 3 vignettes revolving around EBV latency and the control by EBNO1, but also extending into some new directions in multiple sclerosis. So hopefully you can bear with me through that. I do have some financial conflict of interest involving an EBNO1 inhibitor, and I will be talking today a little bit about EBNO1, but not about small molecules. So EBNO1 needs no introduction to this audience. Nevertheless, it is a multifunctional protein that is essential for EBV latency in proliferating cells. Maintaining the EBV genome is its critical function. This has really been well characterized by many laboratories represented here today. Bill Sugden showing it's required for OREP function, both for replication initiation and plasmid maintenance, and Laurie Frappier's structural studies on EBNO1 structural domain, the DNA binding domain. We've contributed a little bit to some of those studies. We've shown EBNO1 is involved in forming these recombinational structures at the family of repeats, and that's important for maintaining the episomes during cell division. We've also shown EBNO1 can bind to many host cellular binding sites through its DNA binding domain, and that's important for tethering episomes to specific loci, and that contributes to some of the chromosomal domains. But one of the striking features of EBNO1 is that the genetic context of EBNO1 binding sites is important for conferring function. And this is probably best characterized at OREP, where the dyad symmetry element, the arrangement of binding sites, determines that it can very efficiently recruit ORC and MCMs to be a site of replication initiation, while at the family of repeats, the positioning of the DNA recognition sites make them very efficient for episome maintenance, chromosome tethering, transcriptional enhancer function. So how are these different? We wanted to explore that a little bit more in detail biochemically. So we initiated some structural studies. We tried to do cryo-EM on the full-length EBNO1, and we have not yet succeeded with the full-length protein. But I'm going to tell you a little bit about work on an extended C-terminal domain that goes from amino acid 400 to 641. And we have solved the cryo-EM structures of that smaller domain, and then some biochemical studies on the N-terminal domain and how we think these two may be interacting. And we're going to look at 2 different elements, the dyad symmetry element, 2 binding sites there, which we call half DS, and at the family of repeats, 2 binding sites called 2XFR that are separated by this AT-rich 12 base pair spacer element. So the cryo-EM studies were done in collaboration with Kenji Murakami at the University of Pennsylvania. We were able to solve EBNO1 binding to these structures to either 3.6 or 3.2 angstrom resolution. Not as good as what one gets with X-ray, but pretty good for cryo. The main difference in the 2 structures is that the DS has a significant bend in the DNA, and that's mediated mostly through the contacts between the dimer-dimer interface for EBNA1 at the DS, and that's been shown by John Yates to be critical in terms of the spacing between the two elements. And at the FR, there are no contacts between the monomers, the EBNA1 dimers, and so there's a much less DNA bend, only about 155 degrees instead of the 120. But what I do want to talk about first is that we notice in the FR, in addition to some unusual conformations in the extended DNA binding domain that wraps around the minor groove of the DNA, but the space between the two dimers is an attractive site for some protein to bind. We tested and had previously explored OCT2 binding to that, and OCT2 binds quite well, but it does not bind well in the context of the EBNA1 DVD DNA binding domains. And GATA1 has also been implicated for binding here. It does not bind well. But the N-terminal domain of EBNA1 that contains this zinc hook that's been characterized by Ashok Iyer and Bill Sugden's group a number of years ago, depending on these two cysteine coordinating residues, does bind quite well. And I'll show you just a little bit of data on that. We compared purified N-terminal domain 1 to 90 with or without the cysteine residues. And what we're able to show is that the wild type protein from 1 to 90 can bind by itself to that AT-rich region. If you mutate it, it doesn't bind. I don't have that here, but in the context of the DNA binding domain, we can see the trimeric complex forming very nicely with the EBNA1 DBD and then addition of the AT hook domain. And that's absolutely dependent on the cysteine residues and also dependent on zinc. And I'm not going to show you that. So what this suggests to us is that if the N-terminal domain of EBNA1, and this is modeled into Joanna Wilson's full-length structure of EBNA1, is able to bind to that AT-rich element between this, it sort of suggests that EBNA1 at the family of repeats can form this really packed oligomeric filament. And that might be consistent with some other models we've proposed where one gets dimeric episomes being formed. So we're still in the process of trying to understand if this is in fact true with a larger array of repeats. So just to jump forward to our second vignette, we've also been interested in proteins that associate with EBNA1, especially in the context of ORI-P and the family of repeats. So we purified EBNA1 from ORI-P expressing cells, and we purified, of course, EBNA1 as well as other characterized EBNA1 interacting proteins like USP7. But we found a collection of these PLOD 1, 2, and 3, and I know that there's a recent publication from Laurie Frappe's group showing PLOD 1 interacts with EBNA1, and we can validate that. And these are really quite robust in the proteomics mass spec. And we can show by IP in Raji and Mutu Burkitt lymphoma cells that both the forward EBNA1 IP or the reverse PLOD 1 IP could bring down the partner, EBNA1. And interestingly, EBNA1 has an unusual mobility shift when it's pulled down with PLOD1, and we don't fully understand that. Other than that, PLODs are enzymes that are involved in lysine hydroxylation. And their function is to modify procollagen to mature collagen, and the way they do that is they hydroxylate the lysines, and then that leads to glycosylation and eventually protein-protein cross-linking through lysine cross-linking. So we don't know what the significance of that is yet, but functionally, if we knock down PLOD1, and we got a moderately good knockdown of PLOD1 with this shRNA and one other that we're not showing here. But what we found is that the knockdown of PLOD1 led to a significant loss of EBNA1 protein, suggesting it was controlling EBNA1 stabilization. A few other proteins like TRF1, which binds to REP, also was affected for protein stabilization. And we can show that knockdown of PLOD1 led to loss of EBV episomes in both Raji and lymphoblastoid cell lines. And a small molecule inhibitor of PLOD1 phenocopied the knockdown. This is dipyridol. And you can see it has an effect on PLOD1, but a really strong effect on EBNA1 protein stability, and some effect on EBNA2 as well. So both PLOD1 knockdown and dipyridol were selectively inhibitory towards EBV positive cells relative to EBV negative cells, particularly sensitive to LCLs. LCLs were particularly sensitive to this knockdown. And of course, we wanted to know whether or not PLOD1 might be involved in post-translational modification of EBNA1. So we did LC-MS. We were able to identify one peptide of EBNA1 that was subject to lysine hydroxylation. And this peptide, excuse me, was modified either at 460 or 461. These two residues are close by to each other, and it wasn't clear which of the two were the subject of modification. But interestingly, the cryo-EM structure shows that these have an unusual variable conformation, whether they're at the FR or the DS. They're sort of in either an open or a closed conformation. The closed where they're making direct contact with the minor groove. So I'll just summarize. I'd say that mutation of those residues does not affect DNA binding significantly, but mutation of K460 to either alanine or arginine completely abrogated DNA replication. And you see protein expression was pretty much unaffected, but the DNA replication with either of those mutations affected replication. So just to summarize this portion of the talk, again, looking at EBNA1 structure and post-translational modifications, and whether or not PLOD1 post-translational modification is important for FR. Protein-protein cross-linking is something we're very intrigued by. It may be related to what happens to collagen, but we really don't have evidence for that at the moment. We know that K460 is required for DNA replication function. So I want to spend the rest of the time talking about some of our recent efforts looking at EBV and EBNA1 in particular in multiple sclerosis. And I'm sure most of you are aware of the exciting new papers that came out from the Asherio group and Bill Robinson's group on looking at and really solidifying the role of EBV in multiple sclerosis, providing new mechanistic suggestions. So the approach we took to try and understand EBV in MS was to isolate B cells from MS patients and to use their endogenous EBV to get spontaneous B cells from them to be able to get the pairs of MS-associated EBV and MS-associated B cells relative to those that were immortalized ex vivo by B95-8. So we were able to get spontaneous B cells from either healthy controls or from multiple sclerosis patients that were either active or stable, and this is a collaboration with Steve Jacobson at the NIH. Unfortunately, this study is still a relatively small cohort of spontaneous B cells. Only 9 patients were used at the time. But what we did find is that the active MS patients were able to generate spontaneous B cells at a much higher rate. and that they had much higher copy numbers of EBV, either by digital PCR, digital droplet PCR or qPCR. We're able to look at RNA-seq and differential gene expression, and there are definitely changes between the active MS versus the spontaneous versus the healthy controls. And this just highlights some of those genes we found that were of particular interest to us, FOXP1 being highly expressed in the active MS, CD44 also, while the HAVCR2 involved in immune regulation of B cells was significantly lower in actives versus healthy controls. Inflammatory cytokines tended to be much higher in the active MS patients. Lymphotoxin A, TNF alpha, IL-12 were all upregulated by either RT-PCR or by intracellular cytokine staining for at least GM-CSF and IL-10. And interestingly, the active MS genomes tended to be more heterogeneous. So sequencing the DNA, the active MS had unusual structures, not so much specific mutation rates, but structural variations. And in fact, one of the AMS patients had more than one genome. If we looked at polymorphisms in EBNA1 and ORI-P in the active MS, we could find a number of unusual polymorphisms in EBNA1. They're listed here. They're not seen typically in the type 1 or type 2 EBVs. And surprisingly, in the ORI-P region, in the FR and in the AT-rich spacer, we found variants that were unique to the spontaneous LCLs from MS patients, but not in the healthy controls. And like what was described for Cornelia de Lange syndrome, we were able to show that the AMS, these active MS patient B cells, had much higher rates of ZTA and EAD expression. They were chronically lytic. They did not survive very long in culture. And so we're trying to understand why they have this chronic lytic activity. And that's shown here by RT-PCR as well. They generally use the CPWP promoter and not the QP, although there's a little bit of QP utilization in one of the active MSs. The QP is for more of a type 1 Burkitt lymphoma latency. But I think this is the interesting point to us, is that EBNA1 binding in chip assays was significantly compromised in the MS patients relative to the healthy controls. And so we're exploring the idea, and that was only on the viral genome, but not so much EBNA1 sites at the HLA locus, where just a cellular example of an EBNA1 binding site. So just to finalize, what's the functional significance of this? Is there any therapeutic approaches? And I just want to end with a quick study using an antiretroviral that was shown to have activity in multiple sclerosis and also to have activity against EBV DNA polymerase. And this was brought to our attention by Alberto Oscherio, who's been interested in this approach. So we explored whether this tenofovir might have any activity on the lytic active spontaneous B cells, and it does. It reduces their copy numbers. It reduces ZTA expression in the EAD expression, at least in one of our very active MS patients. And if you treat, and that's in contrast again, cyclovir, which did not work well. And I think this is the interesting point. If you treat these spontaneously active MS patient-derived B cells with tenofovir, you reduce their ability to activate T cells in a mixed T cell ELISpot reaction. So this is T cell expressing TNF alpha or IL-6 or LTA, and the B cells then that were treated with tenofovir have reduced ability to stimulate T cell cytokine production. Okay. So I'm going to stop there and just summarize that in these MS spontaneous cells, we're finding that, again, in a small cohort, that the EBV genomes are heterogeneous. They tend to be more lytic. We're finding that EBNA1 interaction with OREP appears to be impaired. And so we speculate that that has to do with, that will lead to a chronic lytic and inflammatory phenotype, and that might be an underlying source of MS. So again, just to summarize, MS patient-derived LCLs have deregulated lytic gene activity. The patient-derived B cells express inflammatory cytokines. EBNA1 function is compromised in MS patient B cells, and tenofovir inhibits lytic gene expression and replication and reduces inflammatory activity. So let me thank the people who've done the work. From my lab, Sam Soldan had done almost all of the work on the MS study. Yang Mei did all of the cryo-EM work in collaboration with Kenji Murakami. Raju Diklu did all the work on PLOD1. And again, our collaborator at NIH, Steve Jacobson, provided all of the spontaneous B cells. And this is Italo Tempura's group here in front, helping with us throughout all of the work, and our funders. So with that, I will stop and thank you for your attention and happy to take any questions.
Thank you, Paul. It was a fantastic talk, as always. There's a question over there.
Paul, does, right here.
Yeah.
Does the inhibitor you developed, does it inhibit the spontaneous growth and proliferation of those spontaneous
it's thank you for asking the question. It does stop all LCL growth, and it didn't have any enriched specificity for MS B cells. So EBV positive B cells will go static when we treat them with the drug, but there was no selective advantage to the MS B cells. Or disadvantage.
So it works just as well.
As best we can tell. Yeah, those are early studies still. Yeah.
But it's alive as a possible therapeutic.
We think so. We're exploring that. Yeah, thanks.
There's a question up there. A couple of questions there.
Where? Yep.
Thinking back to your first suggestion, treating this idea that the EBV is actually binding to the genome, can you speculate about what that means in terms of the ability of that to mediate chromosomal association?
Yeah, you know, that's interesting. The competition. So the AT hook is certainly implicated in tethering to host chromosome AT-rich DNA, right? I don't know. I don't think it's.
Could it be regulated in the cell cycle faster or something?
Yeah, of course. So, you know, a number of models could come to mind, and we don't have a good answer. But yeah, obviously, it has proximal affinity for the FR element, and how that competes with cellular binding sites. We really don't know yet, but yeah, thanks.
There is a question right there in front here.
Yes, had any of your MS patients had alemtuzumab before they were studied?
That's a great question, and I do not know the answer. I don't know the history of these patients. I could get that information, but thanks for asking. Don't know.
Go ahead. Yep.
Well, Paul, that kind of leads into my question as far as MS has a spectrum of disease severity, and I would be curious to look and see if that EBV reactivation chronic state is associated with those outcomes. So I don't know if you're planning to look at the spectrum of MS within your studies.
So those who had the active MS were the ones where B cells were easier to generate and also had this phenotype of being more lytic. But my understanding is that the patients cycle between being, very active to being more of a smoldering kind of, not as a relapsing type of situation, and that continues. So, but it's, yeah, we'll explore that as well. Thank you.
Jeb, one final question.
Paul, do you think this defect or alteration in EBNA1 function increasing lytic activity in your spontaneous LCLs from MS patients, would that lead us to understand the initiation, the pathogenesis of MS? Would this be a specific EBNA1 serotype or functional type that triggers B cell activity and T cell interaction differently than our common EBV prototype?
Yeah, well, MS is still very complex, but would a component of the way in which EBNA1 is functioning in that particular context be an underlying mechanism? I think that's likely to be the case. Obviously, how do we bring in the host component also? Obviously, there's
immune response.
Yeah, and we don't really yet have a full understanding of that.
We could discuss later.
Sure.
There are so many questions. Just one last question.
Oh, thank you. We have, there are so many questions, and so we'll take the last question here.
Okay. Yeah.
And then we have a specific MS session on Wednesday, so we can take all the other questions. I can understand it's pretty exciting talk and the field as well. Thank you.
Okay, so can I ask you about the first part? So when you are payload one is down regulated, knocked down, then you show the decrease of EBNA1, and you show the effect on EBV positive cells. But did you see the downstream effect of EBNA1 decrease, like if someone EBV genome is deleted, or any alteration in epigenome, like 3D chromatin interaction, as you showed in
with which knockdown?
Yeah, when payload is
Payload
knocked down,
one, yeah.
or maybe inhibition, then EBNA1 is protein level is deleted.
So
Then-
there is a trend to see some increase in EBV lytic activity when PLOD1 is knocked down and when EBNA1 is knocked down. And that depends a little bit on the cell type.
Okay.
So leave it at that.
And is there any difference from other model, like just simple EBNA1 knockdown or like EBNA1 decoy?
So in our experience, it's, again, knocking down EBNA1 has slightly different phenotypes in different cell backgrounds. And we're not fully sure why that is. In some cases, you do trigger lytic, but in other cases, you just see cells starting to go into arrest without any lytic reactivation.
Okay, thank you very much.
Thank you, Paul.
Okay, thank you.
Okay, so let's move on to the next talk. It's by Ed G. Akidil, and he's from Research Unit Gene Vectors, Germany. And he will, or she will speak about histone chaperone CAF1 is essential for the survival of EBV-infected primary B cells and the establishment of viral latency.
Yeah, thank you very much for the kind introduction. Welcome everyone, buongiorno. So I will talk about the histone chaperone CAF1 and its role in the survival of EBV-infected primary human B cells. So I will kind of change the direction of this, the talks that we have been seeing until now, and we will look at the latency part of EBV infection. So upon infection, delivers it is naive linear genomic DNA that is lack of methylation and lack of histone modifications into the cell. And around day 2, the nucleosomes, we know that nucleosomes are acquired and on the EBV DNA, and they this happens actually right before the DNA replication starts around day 3 post-infection. And beginning from day 4, the cells start replicating and proliferating, and they establish the LCLs. And in latent phase, we know that viral gene expression is controlled with the help of other histone modifications and CpG methylation. And within these all epigenetic changes that I have mentioned, we don't know that much about when this and how this viral DNA acquires nucleosomes. and which cellular factors drive this process. And to understand this aspect of the epigenetic regulation, we wanted to investigate the cellular chromatin factors and their presumed function in EBV-infected B cells. These cellular chromatin factors our cellular targets are some histone chaperones like HERA, DUX, ATRX, which are working in histone loading in DNA in replication-independent manner. And they are loading basically H3.3 histone. On the other hand, we have CAF1, which is working in DNA replication manner and loading H3.1 variant on the naked DNA. And in our protocol, we are isolating the B cells from a human source and nucleoaffecting them with 2 RMP complexes targeting the adjacent sequences of the target exons. And these knockout B cells were treated for their proliferation and growth with either with EBV infection or by keeping them with CD40 ligand supplemented with IL-4. And knockout efficiencies in those cells were quantified in genomic and protein level. And further on, we performed some phenotypical analysis where we performed some functional assays. So I have to mention that as a control, we targeted a cell surface protein, which is a CD46 gene, in parallel to the other cellular targets that we had. And this gene is high and homogeneously expressed on the cell surface on the B cells. Therefore, it allows us to detect the knockout efficiency via FACS analysis easily. And we always run this as a control in our experiments. Including CD46 with other targets, we reached to 90% of knockout efficiency in primary human B cells. And if you are curious more about how we established this CRISPR protocol in primary human B cells and how we use it to study some EBV-related functional assays, please check our recently published paper. So back to the biological question we had. After establishing the knockout cells, I checked their absolute cell number. And we see that HERA, DUX, ATRX knockout cells shows the same growth profile as the CD46 knockout ones. On the other hand, CAF1B knockout cells shows that they don't proliferate and they eventually die. And we wanted to know if this CAF1B, the role of CAF1B on B cell survival also will result in the same phenotype when they were stimulated with CD40 ligand feeder layer supplemented with IL-4. Therefore, I performed a parallel experiment, and as you can see, CAF1B is only critical for the survival of EBV-infected B cells. And HERA, DUX, ATRX chaperone seems to be that they don't have an apparent role in B cell survival. To understand this strong phenotype that we had, we wanted to perform further analysis by using this CAF1B knockout and CD46 knockout samples in 2 different culturing conditions. And we performed some functional assays on their early apoptotic marker, mitochondrial activity, and glucose uptake. And we can see here clearly that with this pink arrow, CAF1B knockout results in the apoptosis in EBV-infected B cells. And along with that, their mitochondrial activity also reduces. And in parallel, their glucose uptake also stays stable. On the other hand, the CD46 knockout EBV-infected cells shows higher level of glucose uptake by time. So we showed several functional analyses on this CAF1B knockout EBV-infected B cells. And we wanted to understand what is the biological mechanism behind this phenotype we are observing. So we went back and looked to the role of the CAF1B, and it is actually known as, well known as loading new nucleosomes downstream of the replication fork onto the newly replicated DNA. Therefore, we wanted to check if this CAF1B knockout EBV infected cells has a problem in their cell cycle. So I performed the cell cycle analysis by checking their EDU incorporation and DNA content over time. And we see that CD46 knockout cells, when they are infected or they were kept in the stimulation with CD40 ligand feeder cells, they show the almost same phenotype profile of cell cycle. On the other hand, CAF1B knockout EBV infected cells shows this sub-G1 population around day 4, which increases rapidly around day 7. And surprisingly as well, this CAF1B knockout cells do not show the same strong phenotype in terms of their sub-G1 fraction in their cell cycle. So what I show you until now, that EBV infection kills these CAF1B knockout cells. And from the other additional experiments that we did, we know that these cells also show a strong genotoxic stress. And we hypothesize that this CAF1B might be taking role in controlling the viral DNA replication. To answer this, I performed qPCR analysis quantifying the relative viral DNA versus the cellular DNA amounts. And what we see here is that viral DNA amount drops around day 3 at the time when the cells start cycling. But most excitingly is when CAF1B is knockout, we can see that EBV DNA over-replicates. Therefore, we can say that CAF1B is preventing viral DNA replication from unlicensed replication. And most likely that without CAF1B, therefore EBV induces this genotoxic stress that we have been observing. And my talk ends here, and I would like to summarize what we mentioned until now. So CAF1B is essential for EBV-infected B cells survival. And from our earlier RNA sequencing analysis that EBV infection upregulates the CAF1B dramatically in primary B cells in terms of their transcript level. And CAF1B decreases metabolic activity in EBV-infected B cells, and this leads to also later appearance of this sub-G1 population, which becomes dominant around day 7. And we also show that CAF1B prevents the over-replication of EBV DNA, which probably proceeds in an unlicensed manner. And we are at the moment hypothesizing that CAF1B is taking role in the chromatinization of the incoming EBV DNA, and therefore, in the absence of it, it fails to replicate properly. And we are now working on it to by performing MNAs and southern blotting experiments, and hope to share with you soon another meeting. And by this, I would like to thank you all for your attention and time. And big thanks goes to Wolfgang for his encouraging supervision along my projects, and to my group for daily discussions and for this journey that I had in the lab. Yeah, thank you. I'm happy to have your questions.
Thank you, SGC. Other questions? There's one question over here.
Lovely talk. The increase you see in the EBV genome content, do you think that's down to aberrant lytic activation or dysregulation of EBNA1 or EP?
It's a really good question, because we thought that there might be early lytic genes which can be expressed in the early phase of latency. Actually, we know that they are expressed, including BZLF1, but they cannot induce the expression of other genes because CpG methylation is not completed. But when we wanted to check if this phenotype we are observing is based on based on the expression of early lytic genes, we check this BMRF1 gene because we have this virus, reporter virus, which BMRF1, downstream of the BMRF1, there's red CD2 gene. And by this, on the cell surface of the B cells, we can say that, okay, BMRF1 is expressed or not. And when I did actually those experiments, because it was an interesting concept indeed, I showed that CAF1B knockout cells shows after day 3, they show also, actually I can show you if, oh, we have to go a bit, sorry. Here. So we can see that, so this green line is the CAF1B knockout reporter EBV, and then the blue one is the CD4 knockout reporter EBV. You can see that CAF1B knockout ones actually showing a bit higher level of CD2 positive, CD2 expression, meaning that BMRF1 expression. But then we wanted to check also BZLF1, and we had the in-hand BZLF1 knockout virus. And when we did these experiments, performed the same concept with BZLF1 knockout ones, we didn't see that the cells are actually surviving. So there's no direct correlation that we could observe, I would say.
Thank you, SG. There's Sumita back there.
Quick question. When you knock out CAF1B, back here.
Oh, okay.
So when you knock out CAF1B, do you see the entire genome replicate repeatedly, or are these partial genome replications? So does replication begin repeatedly from ORIP or ORILES, I don't know, and does it then abort, or does it go to completion?
You are trying to ask me that if, sorry if I understood right, if it is firing from ORIP or not, if I check that.
That's part of it, but the rest of it is, if you remove CAF1B, do you see the genome replicated completely over and over, or do you have partial genome replication?
Okay, I see. We didn't check that. It's a good question, actually. So the qPCR analysis is on a fraction of this where I am amplifying, but I didn't check for the whole. I checked for different regions of the viral genome, and it showed the same result. I checked 4 other regions, and it showed the same result. So I would say it shows the same, yeah.
There is one last question over here. Oh, there are a couple of questions. Bill also has a question. Go ahead.
CAF1B. In established LCL, I didn't try, I have to say, but I know from another paper, but they are working with Burkitt lymphoma cell line. So they show that recently CAF1B and HERA knockout, CRISPR screen when they did it actually, they show that it switches to lytic activation. But I don't have the experiment where I perform it in LCLs.
Bill?
Here.
Over there.
Can you get enough of your knockout cells infected with EBV so that you could, for example, label with BRDU and ask if you directly have unlicensed DNA synthesis?
Yeah, that's a good point. So yeah, this was one of the concepts we wanted to try actually. Yeah, then you would see localized what is happening on the EBV DNA, right? Yeah.
Okay, thank you very much, Edsi.
Thank you.
Let's move on. The next talk will be by Catherine A. Willard from Duke University in North Carolina. And she will be talking about novel Epstein-Barr virus strains isolated from endemic Burkitt lymphoma patients display unusual and reversible spontaneous lytic phenotypes. Thank you.
All right. Hi, everybody. I'm Katie Willard, and I'm a PhD candidate in Mika Luftig's lab at Duke. And I'm going to tell you about some of my thesis research that involves these diverse EBV strains that have spontaneous lytic phenotypes. So I want to start with some background about EBV genetics, which has already been covered today, so I won't belabor it. But the main driver of genetic differences between EBV strains is type 1 versus type 2. And the bulk of this difference comes in the EBNA2 and EBNA3 genes. However, type 2 EBV also has this ZPV3 polymorphism within the Z promoter. And the Kenny lab showed that this SNP in particular creates an NFAT consensus sequence. And since NFAT is a transcription factor, it can bind to the promoter and increase Z expression and increase lytic activity in these strains that contain ZPV3. And all type 2 strains sequenced to date have this ZPV3 polymorphism, and some rare type 1 strains also contain it, such as M81. But most type 1 strains are what we call ZPV. They don't contain these SNPs in the Z promoter. So in this talk, I'm going to focus on some new endemic Burkitt lymphoma-derived strains that we have thanks to our collaborators, the Mormon Lab, the Bailey Lab, and the Kenya Medical Research Institute. And the people at KEMRI see Kenyan endemic Burkitt lymphoma patients, and they have been able to take biopsies of the tumors and enrich for B cells and establish Burkitt lymphoma lines that we can propagate easily in culture. And I'm going to tell you about 5 of these new lines. They're all derived from different EBL patients and are all infected with different strains of EBV. They are both type 1 and type 2 strains. And as expected, the type 2 strains have the V3 polymorphism in the Z promoter. The type 1s do not. But interestingly, 3 out of these 5 new strains are lytic in the Burkitt lymphoma background. And when I make virus from these strains and infect PBMCs in vitro, the resulting LCLs are lytic in 2 out of the 5 strains. So this strain 720 is particularly interesting to me because it's a type 1 strain without the V3 polymorphism in the Z promoter, and it is lytic in both the Burkitt lymphoma and LCL backgrounds, which suggests that there could be an alternative mechanism beyond ZPV3 that can lead to this lytic activity. And this is similar to work recently described by the Delacluse lab, where they've identified some type 1 lytic strains as well. And our collaborator, Cliff, is going to talk more about these new Burkitt lymphoma lines tomorrow, so stay tuned for that. So now I want to show you some data about how I know that these new lines are lytic. The main way I assess this is through flow cytometry for the GP350 viral glycoprotein. And this is just a representative FACS plot. And we can see that these 3 lines, 719, 720, and 725, express varying degrees of GP350 just basally in culture. You can compare this to an induced lytic system where these P3HR1 HT cells are treated with hydroxytamoxifen for 24 hours, and this induces about 23% of the cells to express GP350. So these new Burkitt lymphoma lines just basally spontaneously in culture are producing ballpark similar levels to this induced lytic system. I've also assayed lytic in different ways, such as an immunoblot for the lytic transcription factors RTA and ZTA, and you can see that these lines that are GP350 positive are also expressing the lytic transcription factors, whereas the GP350 negative ones are not. So I refer to these 3 lines as spontaneously lytic. So interestingly, this lytic phenotype can also revert back to a basal state in culture. So in this experiment, I take my heterogeneous bulk culture of these lines that contain latent GP350 negative cells and lytic GP350 positive cells, sort based on GP350 expression, put the pure populations back into culture, and then daily over time reassay GP350 positive with a GP350 positivity with a different fluorophore, so I can identify newly GP350 positive cells. And these are the data resulting from this experiment. And you can see that in both BL cell line 719 and BL 720, the GP350 positive cells very quickly over time revert back towards a more latent state. And the GP350 negative cells over time, but more slowly, start increasing in GP350 positivity until by day 6, they reconverge at a basal level of GP350 expression in culture. And not only are these new lines expressing lytic proteins, they also produce virus. So in this time course experiment, I assay GP350 positivity of the cell lines and then use DNA qPCR of the supernatants to quantify the amount of viral particles released per cell. We can see that the 3 lines that we expect to be lytic are GP350 positive. However, only BL719 and BL720 are actually producing virus that is accumulating in the supernatant, which suggests that BL725 may be undergoing some sort of abortive lytic replication that doesn't actually produce particles. And this is corroborated through these electron microscopy experiments where I was able to detect viral particles in BL719 and BL720, but not in BL725. Importantly, and as I alluded to earlier, these lytic strains also produce lytic LCLs. In this experiment, I further induce the lytic cycle through treating with TPA and sodium butyrate. After 3 days, I harvest the virus and concentrate it and infect PBMCs. And in 5 weeks, I have LCLs derived from these strains. And we can see that strain 720 and 725 induce B-cell proliferation to an equivalent amount as the standard lab strain B958. However, strain 719 is non-transforming in vitro. And the LCLs derived from infections with strain 720 and 725 are also GB350 positive. So all of these data that I showed you suggest that the spontaneous lytic phenotype is encoded by these new EBV strains, and potentially in a EBV type specific manner, since I see this both in a type 1 ZPP strain and in a type 2 ZPV3 strain. So what I didn't tell you earlier is that there's actually heterogeneity in this lytic expression in limiting dilution LCLs derived from strain 720. So these are 3 donor-matched bulk LCLs, and you can see that the ones derived from infection with strain 720 are highly GB350 positive. However, the ones, the ones derived from strain 725. But the ones derived from strain 720 are GB350 positive, but have a much broader distribution of this positivity. When I do a limiting dilution infection with strain 720 and pick LCL clones that grow out, we get some that are very lytic and some that are tightly latent. And this was a pretty interesting result to me, and this probably explains the distribution that we see in this bulk transformation. So to try to determine the mechanism by which this result occurs, I sent some of these clones for RNA sequencing. So I sent 3 highly GB350 positive lytic clones and 3 latent ones that are all derived from the same blood donor, so it's the same human genetic background. And this is a volcano plot of the differentially expressed genes from this experiment. And over 1,300 genes were differentially expressed, again, in the same human genetic background between these lytic and latent LCL720s. And I aligned these data to a concatenated human and EBV genome so we could detect EBV reads as well. You can see that the EBV lytic genes are upregulated in the lytic LCLs. So we are looking at what we think we are looking at here. So obviously there is a lot going on in this experiment, but one thing that stood out to me is that B-cell receptor signaling was enriched in the lytic LCLs compared to the latent ones. And these lines are just sitting in culture. I'm not stimulating the B-cell receptor in any way, so this led me to ask if there is an EBV gene that could be signaling through this pathway. And indeed, LMP2A mimics BCR signaling. So LMP2A is a transmembrane EBV protein, and it has a cytoplasmic tail. And phosphorylated tyrosines and ITAMs within this cytoplasmic tail of LMP2A mimic the ITAMs in the B-cell receptor tail, so they can signal through similar pathways. And importantly, some of these pathways end with factors that can bind to the R promoter and Z promoter of EBV and induce the lytic cycle. Importantly, these lytic Burkitt lymphoma lines are actually in a latency 3-like state, and I've highlighted the lytic ones here. You can see that they express EBNA3, EBNA2, EBNA1, LMP1, and one of them, EBNA-LP. This is a little bit different than what we would normally think from a Burkitt lymphoma line, such as Akata, which is a prototypical Burkitt lymphoma where just EBNA1 is expressed. However, this isn't unprecedented. Data from the Bell Lab has showed that Burkitt lymphoma lines can have a variety of latency states from latency 1 to WP restricted latency and latency 3. So we do expect that LMP2A is expressed in the Burkitt lymphoma background of these lines as well. So this led me to ask if there are genetic differences in this type 1 lytic strain LMP2A. And when I aligned the genome of this new strain 720 to type 1 latent strains, I found 5 coding mutations within LMP2A. And this one in particular is interesting to me because a serine in the cytoplasmic tail of LMP2A is mutated to a tyrosine in this strain. And it is predicted to be phosphorylated. And this is the result from that phosphorylation prediction. And all of the ones, so here's tyrosine 37, and all of these ones with blue stars are ones from the literature that are known to also be phosphorylated. So this prediction algorithm works pretty well. Next, I wanted to know if this serine 37 mutated to a tyrosine is specific to this one strain or if it is more broadly distributed. So I aligned the LMP2A coding sequence of 315 different published EBV strains, and I found that 10 strains, all from Kenya, have this serine 37 mutated to a tyrosine. And then interestingly, I also found that 5 strains from East Asia also have the next serine 38 mutated to a tyrosine, which suggests to me that there could be separate evolutionary events for the virus leading to the establishment of tyrosines in this region of the LMP2A cytoplasmic tail. So I'll end now with some current and future directions for this work. Obviously, I want to know if this tyrosine in the LMP2A cytoplasmic tail can confer this type 1 lytic phenotype in this new strain. And then I didn't have time to get into it today, but obviously there's a lot of differentially expressed host genes in that RNA sequencing data set. So I would like to determine what host factors can enable this lytic phenotype to show through as well. And then also what factors, both host and viral, can enable the reversibility of this lytic phenotype. So I'd like to thank my lab and also my collaborators, particularly the Mormon lab from the University of Massachusetts and the Bailey lab from Brown and everybody at Emory that established these lines that I am basing my thesis off of. And I'll take any questions.
Thank you. There are a couple of questions over there on the left side of the hall. Yep, up there all the way top, up to the top, yes.
Hey, nice talk. You said this Burkitt lines that were lytic had type 3 latency. Did you look at the actual tumor, the Burkitt tumor? Did they have type 3 latency or were they type 1?
Yeah, actually, Cliff is going to talk about this some more tomorrow. There are a few differences between the tumor and the lines, but they mostly align, yeah. So we, again, Cliff will talk about this more tomorrow, but I believe they were xenographed into mice, and that's what we were looking at. But
yeah, really excellent talk. And over here.
Thank you.
You pulled out some unusual EBV strains from Burkitt lymphoma, and we just heard Paul talk about unusual EBV strains in MS. Could it be that there's just a lot more diversity, and now that we have the tools, we're uncovering that the old canonical rules just don't apply that much if you look at the right SNP variant?
yeah, I definitely think that's what's happening here. We're just able to learn a lot more about it now that we have this technology available to us.
There's one, Paul, there's a question. Yeah. There we go.
Thank you, very nice talk. I was just thinking that the phenotype is a little bit like the old P3HR1 with the het DNA in, and I just wondered if it might be smart to have a look to check you haven't got rearranged effective EBV present in the 720 line as well, which is co-packaging and might be driving lytic cycle. Or in case you could do a DNA plot or something like that to look at the viral genome.
Yeah, that would be a good experiment to do. Thank you.
There are a couple of more questions back there. Yep.
So if you have over 60% of your cells in the lytic cycle, how are they surviving in culture?
Yeah, they don't. So those LCLs, those lytic LCLs, they don't grow quite as robustly as the latent clones of them. So, but they do still grow. They're just slower.
And are you doing single cell cloning when you clone those cells out?
So it's
with
not
the
single.
LCLs?
It's not single cell cloning. It's just a limiting dilution of the virus in a plate of PBMCs.
All right, one last question. Did you immune phenotype the cell lines? So they look more like the classic type 1 to type 3. So have you looked at the phenotype of those lytic type 3 latency cells?
Sorry, like which phenotype?
The one you have the type 3 latency. Have you looked at the immune phenotype of those cells to see if they're Burkitt lymphoma-like or more LCL-like?
I have not specifically checked. Are you saying like surface markers that are
Yeah.
Yeah, I haven't specifically checked that.
Okay, thank you. If there are no more questions, let's move on to the next speaker, Bill Sugden from University of Wisconsin-Madison. And Bill will talk about Epstein-Barr virus manipulates cellular chromatin structure during its lytic phase. Bill?
Well, thank you for the opportunity. And let me say, I shouldn't be giving this talk. It should be given by Quincy Rosemarie, who couldn't come here because she couldn't get her visa. So she's from Indonesia and she couldn't have it, leave the country, come back to the States and get it in time. So I'm going to do my best. What Quincy works on is the Epstein-Barr virus ability to manipulate cellular chromatin during its lytic phase. And what she means by that is shown on the right part of this slide. If you look at the first row, where you're looking at cellular chromatin by virtue of its binding H2B fused to GFP, you can see that the chromatin is throughout the nucleus except for these holes, which really are nucleoli. But now look at the second two rows. That shows you what's happening in the lytic cycle. These are images from Yafang Chu. And in the middle row, you can see again that the chromatin is being gradually compacted now. You're looking at red dots. Those actually reflect an EBV replicon that's being detected by binding lac repressor fused to RFP. The other voids are P3HR1 DNA that you can't see. But by 36 hours, what you really can see is that the chromatin has been compacted even more. And it's moving to the periphery of the nucleus. So please just think about this for a moment. Viruses can compact their own DNA. They do so dramatically to get it into virus particles. But this is an instance in which somehow the virus is leading to the compaction and movement of cellular chromatin. It's fascinating, and it's not unique to EBV. So it turns out that 4 families of DNA viruses, the herpes viruses, the adenoviruses, the parvoviruses, and the baculoviruses, all display this reorganization of chromatin, which Quincy calls rock. They all rock. And in this same slide, you can see images from HSV1, human cytomegalovirus, and baculovirus of a kind of ROC. So it's general to DNA viruses that also do not have histones on their DNA when they package that DNA. That's a common feature of ROC. So what Quincy wanted to do is to understand which viral genes of EBV mediate ROC. And she did so with a genetic complementation assay. Just imagine taking 293 cells that have a mutant of EBV resident in them in such that a single gene has been knocked out. Then she can take those 293 cells, transfect them with RTA and ZTA to induce the lytic cycle, and either add the gene on another plasmid or not, and score for ROC. And on the right part of the slide, you're seeing what she would see for ROC when she has complemented a derivative of EBV that lacks the DNA polymerase gene, and she now has provided it. So this is true complementation. And each of the cells marked with an arrow, you can see features of ROC. The chromatin, again, you're always looking at H2B GFP. The chromatin has been both condensed, compacted, and it's been moved to the periphery of the nucleus. So which genes of EBV are required for ROC? This is a series of experiments done by Quincy to identify those genes. They're all the genes that are involved in DNA amplification in the lytic phase. And let me dwell just for a moment on the last of these, the origin of lytic replication, because this complementation gives us 2 kinds of information. First, it clearly says you need ORI-lit in a cell for ROC to take place. But it tells us one thing more, because Eric Johansson and his colleagues have shown that for late gene expression, ORI-lit must be in cis. That is, the DNA that's going to support late gene expression has to have ORI-lit integral to it. Quincy did these experiments by introducing ORI-lit in trans. That tells us you don't need late genes of EBV for ROC. And she has gone on to confirm that conclusion shown on the right part of the slide by again testing two derivatives of EBV that lack the gene, either of two genes for the VPIC complex. That's the complex that mediates transcription of late genes. When you don't complement them, you can see in this slide, you still, when you induce those cells, see ROC. So we know that EBV's ORI-LIT and DNA synthetic genes are required for ROC. EBV's late genes are not required for ROC. Knowing this, Quincy then asked a question: to what extent is viral DNA synthesis per se required for ROC? And I would have bet you really needed it, but I don't win bets often. And the way she went out and tested this question is by inhibiting viral DNA synthesis with two inhibitors, ganciclovir on the left, phosphoacenic acid on the right. Ganciclovir inhibits really EBV DNA synthesis by chain termination. Phosphonoacetic acid acts directly on the DNA polymerase product, or protein, excuse me. And shown in the bottom protocol, all these cells have GFP, H2B, so you can visualize cellular chromatin. She'd plate the cells. At 24 hours after plating, she would treat with either ganciclovir or phosphonoacetic acid. 24 hours later, after that, she would induce the lytic phase with transfection with, well, in this case, either induction or transfection. 24 hours after that, she would add EDU to detect viral DNA synthesis. Also, she would do that at 48 hours. And then fix the cells, use stain or not stain for BMRF1 to prove or not whether the cells were in the lytic cycle, and use click chemistry to detect EDU, and DAPI to detect total DNA. So let me give you her findings. And we've got to go through row by row for you to appreciate them. So if you look at the top row, that's typically latent or non-lytic. You can see in the nucleus with DAPI staining, it overlaps identically with the GFP H2B. That's the nucleus. That cell's not supporting the lytic cycle. You don't see any BRMF1 staining. It's not making DNA because she didn't happen to have these cells in S phase, so you see no EDU incorporation. That's the top row. The middle row is the one that I've talked about so far, where on the far left, you can see that cellular chromatin has been compacted and moved to the periphery, whether you're using DAPI or whether you're using GFP-H2B to visualize the cellular chromatin. In the middle, you see BMRF1 staining, telling us that these cells are in the lytic cycle. And then underneath the EDU, you can see it's well labeled. It's actually beautifully labeled. And she's going to refer to this as ROC type 2. And the reason she's calling it type 2 is in the bottom layer, or the bottom row, excuse me. And in this case, she's treated the cells with ganciclovir to inhibit DNA amplification. Look what she found. Either with DAPI or with GFP-H2B, you can see that the cellular chromatin is condensed. It's much smaller than up in the top row, but it hasn't moved to the periphery. You know these cells are in the lytic cycle because she's detecting BMRF1. And when she does the click chemistry for EDU, you can see barely any. They're not making viral DNA. She's confirmed that this lack of viral DNA detection is correct. She's used quantitative PCR to show that the amount of DNA when she treats with ganciclovir is identical to that which you find in the latently infected cells. And when she measures what's in the lytic cycle, it's increased by about 200-fold in her hands. So what does this mean? First, let me summarize. EBV reorganizes cellular chromatin during its lytic phase. EBV's ORI-LIT and DNA synthetic genes are required. They are required for ROC. EBV's late genes are not required for ROC. This is the kicker. Detectable EBV lytic DNA synthesis is not required for ROC type 1, where there's no margination, no movement to the periphery of the nucleus, but it is required for ROC type 2, where the condensed chromosomal DNA is somehow moves to the periphery of the nucleus. And below, she gives you her findings. And let me now give you her model for what's actually happening and why it even matters. So in the latently infected cell on the left, you see a normal H2B GFP stained nucleus. And then when the lytic phase is induced, gradually the cellular chromatin is condensed or compacted. When viral DNA is amplified, as shown, as you would imagine, in the far right rock type 2 cell, what's happening? Viral DNA does not have histones on it. It will not be as neutralized as it would be were it to have histones on it. So it is a large amount of negative repulsive force, which we assume, or we hypothesize, I should say, is actually doing the pushing. As you make more viral DNA in the absence of histones, that viral DNA really fills these voids, and the force of its not being coated with histones, the electrical repulsion, is driving the chromatin to the periphery of the nucleus. Why would this matter? Why would these 4 families of viruses do it at all? Well, remember, they're viruses that don't coat their genomes with histones when they encapsulate them. So what the viruses are accomplishing, we think, is by generating the movement of the cellular chromatin away from the space in which they amplify, or as they amplify their DNA, it's a space where they can now encapsulate the DNA. Thank you. I'll take any questions.
Thank you, Bill. Other questions? There are 3 questions over there on the right.
That was a great talk. Thank you. I was wondering if you could speculate.
Quincy would do it better. She would have.
I was wondering if you could maybe speculate about what the consequences of host chromatin reorganization look like in the context of aborted lytic. I'm wondering if it maybe is like the rock type 1 you're describing.
That is what I would assume. We've not done the experiment to answer, but that would make complete sense based on what we've seen.
So great talk. My question is about because if chromatin goes to periphery, so usually that is a region where happens a lot of repression of gene expression. And if you think or speculate that also can help to maybe some of the genes that are repressed in that way are genes that might induce an antiviral response. And the second one, based on your speculation, if this is just repulsive during the negative DNA, if you label your chromatin at the periphery to see if there are specific epigenetic marks that are associated with chromatin that has the nuclear lamina, like H3 canine dimethylation.
Okay, so first, I have to say, we've done nothing to ask what would be the consequences of ROC for transcription. I mean, people in this audience have really studied what are the differences in transcription as a function of the lytic cycle, and they're profound. How that might be affected by either the condensation or the movement of chromatin, I do not know. I apologize. Would you repeat the second part of the question?
So the second part, if you stain chromatin during lytic with markers that are associated with chromatin, that like H3 canine dimethylation, do you see that mark enrich or not?
We've not done it in these experiments. We've done things previously like that, and the answer has been there's no difference in the histone deposition level on cellular chromatin. I can't say anything about where in the cellular chromatin, but on average, there's no difference.
There's one over there and a couple of questions here. Yeah, go ahead.
What is it that makes ROC1 stable? It's like a transient state. So you've got inhibitors that look like it's a stable state.
So these experiments are done over the course of 48 hours following induction of the lytic cycle. I have no reason to think that it's stable ad infinitum, but I think that the reason it's stable is because of the lack of force to move that now condensed DNA. Let me point out a huge question about which we have no information is what leads to that condensation. I mean, we're measuring now to be able to answer what is the extent of that condensation, but I have no idea what mediates it. On the other hand, I think that the absence of viral DNA amplification is what allows it to be detected in these experiments.
So very nice talk, Bill. Have you tried to look at the difference between knocking out, for example, one of the viral replication proteins like BMRF1 or so, versus knocking out orylit itself, instead of using a ganciclovir or PA?
Oh, yeah, we have. There is no difference. When you knock them out, there's no ROC, and you can complement and then find the complete ROC by complementation.
Thank you.
One last question over there, top.
Do you know what's happening to the nucleoli when the chromatin condenses and is pushed to the periphery?
I think, okay, so just one statement. I mean, that's a fantastic question, but you know, the nucleoli are themselves fantastic in that regions of chromosomes that will encode ribosomal DNA all are clustered around where you find nucleoli. That's what the nucleoli are. As far as we can see, they in effect dissolve, but where they go within the condensed chromatin, we don't know, but by God, I'd love to know that, an answer to that question, but I don't know.
Okay, thank you very much, Bill.
The next speaker, that should be Gillian Bristol, that is not able to come today, so this will be moved to tomorrow and is replaced by Francesco Baccianti from Heidelberg, who will talk about the Epstein-Barr virus infection of resting B cells requires activation of multiple signaling pathways that converge onto the stress protein ZFP36L1. Please.
Good morning, everyone. I would like to thank first the organizer for giving me the opportunity to present the results of my talk. As we all know, the very first hours after infection are essential for the virus to overcome and control the innate immune response that would otherwise prevent the establishment of latency. And in order to study the interplay that is occurring at these very early time points between EBV and the host, we decided to perform high-throughput analysis using proteomics and phosphoproteomics. Here you see the workflow that we follow. We isolated CD19 positive B cells from healthy donors from our local blood bank. These cells were infected with wild-type virus, M81 wild-type, recombinant EBV that we have in the lab. And then at different time points, we perform proteomics and phosphoproteomics. In general, we perform two types of experiment. As I said, a time course experiment or a single time point experiment in which we use in parallel to the M81 wild-type virus genome depleted virus-like particles or the GP110 knockout, that, as you know, is a fusion deficient virus. And these results were further validated using either EBNA2 knockout or different tegument knockouts that we generated in our lab. So starting from the proteomic analysis, here I'm showing the volcano plot with upregulated and downregulated proteins at one single time point, 6 hours post-infection. We identified different targets, and one that caught our attention was these proteins ZFP36L1. The reason is that this protein is upregulated at earlier time points in proteomic analysis, and it is consistently upregulated also at later time points. So we decided to follow up with ZFP36L1. Regarding the phosphoproteomic analysis, in this case, we perform a single time point experiment. And in this case, we compare the M81 wild-type virus versus either VLPs or GP110 knockout virus. And we identified several phosphopeptides that were either up or downregulated upon infection. And in some cases, we identified common events that were shared between all these three conditions. And what was interesting is that we also identified events that were targeting the ZFP36 family members. But regarding the phosphoproteomic, we decided to follow up with the STAT3 activation. And in particular, we were able to identify this tyrosine 705. This is the unknown activatory phosphorylation event for STAT3. So our question was, what is inducing STAT3 activation, especially because we were able to detect these activation already when using GP110 knockout, meaning that this induction was already visible independently from the release of tegument proteins, but just by induction of the binding of the virus. So we validated the phosphoproteomic analysis by performing a Western blot. Here you see we look at the activation of phosphostat-3 by tyrosine 705 immunoblotting, and we included the EBNA2 knockout. On average, we could see an increase, so a very strong induction with M81 wild type and GP110 knockout virus, but on average, we could detect a low induction when either VLPs or EBNA2 knockout virus was used. So we concluded that, as I said, STAT3 activation was already induced upon binding, but there was a reduction in the STAT3 activation when there is a tegument release that is not followed up by complete establishment of latency. We then wanted to look at what was inducing STAT3 activation, and for this reason, we used the GP110 knockout in order to remove all the other possible effects that were happening because of the latest stages of infection, and we decided to use different inhibitors for the B-cell receptor. In particular, we use SYK, BTK, LIN, but we also use a p38 inhibitor that, since p38 has also been shown to be responsible partially for STAT3 activation. As you can see here, we were able to detect a very strong reduction in STAT3 activation upon using these inhibitors, especially with SYK and BTK, and a milder reduction with both p38 and LIN. We move on then to look at the ZFP36L1, and then what we did first was to confirm the results that we obtained in proteomics, and in particular, we perform a Western blot with time course, and we look at much longer time points. And as you can see here, we identified the induction already at 2 hours post-infection, so it has several bands, as you can see. And this induction was lasting between 1 and 4 days, after which the level of ZFP36L1 expression was strongly reduced. And we also look at the RNA level, and as you can see, there is a very strong induction, so roughly a twofold change induction at 6 hours post-infection, and which follows a strong reduction with ZFP36L1 RNA that goes back to baseline between 24 and 48 hours post-infection. We then moved on looking at what was the effect of the different knockout panel that we investigated, and we used M81 wild type, EBNA2 knockout, VLPs, or GP110 knockout. As you can see, we look at both RNA and protein level, and we detected the strongest induction with M81 wild type, a milder induction with VLPs, but EBNA2 knockout and GP110 were only mildly or to no extent inducing ZFP36L1, both at the RNA and the protein level. As you can see, they really nicely match. So what we concluded that we need the full establishment of infection in order to have the full induction of ZFP36L1 expression. ZFP36L1, together with the other components of the ZFP36 family, is strongly regulated by p38 and MK2 pathway activation. So we confirmed the activation of p38 and MK2 via phosphorylation. And we decided to look at what was happening when we were blocking p38 and MK2. And as you can see in this plot, when we blocked either p38 activation or MK2 activation, we got a very strong reduction in ZFP36L1 expression. And when we look at other pathways, there was no effect. And so ZFP36 family members are known to be very important regulators of mRNA stability, and they do so by downregulating especially pro-inflammatory cytokines by binding to the 3' UTR of these mRNAs and promoting either deannulation or decapping. So we looked first at what was the level of expression of IL-6 and TNF. There are two major cytokines that are targeted by this protein. And as you can see, we perform an experiment in which B cells were seeded for 6 hours at each of the following time points. And the strongest activation for IL-6 was at 6 hours and follow up by a almost complete reduction to nothing for IL-6 expression, while TNF-alpha was following a different kinetic, so induced, and then it was going up the later time points. We look at the mRNA level of expression, and we could see, so this is TNF and then IL-6. So we had induction by 2 hours post-infection of the mRNA of both cytokines, but then at later time points, they followed completely different kinetics, and they mimic what we have seen in the previous experiment, with IL-6 going back to even lower levels of expression, while TNF going back up. And what was interesting is that at 6 hours, there was a reduction for both of them, and this mimicked the level of expression of ZFP36L1. So we could conclude that there was definitely a correlation between ZFP36L1 upregulation and the expression of these two cytokines at these early time points. We then validated using a panel of tegument proteins what was the level of activation of P38, MK2, and ZFP36L1 axis. And as you can see, we detected that the BGLF2, BBLF1, and to a lower extent BCRF4 tegument knockouts were reducing the level of induction of ZFP36L1. So we concluded that these proteins were necessary for the activation of the ZFP36L1 expression. We then looked at what was the role of P38, and then going back to also to STAT3 in terms of latency establishment. So we blocked these pathways, and we performed analysis of expression of EBNA2, but also WP transcripts in order to see whether there was a difference in latency establishment, and we could see that in all, by using P38, MK2, but also STAT3 inhibitors, we could strongly downregulate the expression of EBNA2 and WP, and this was even stronger with STAT3. Since these could be also related to a defective nuclear transfer of EBV episomes, we used a fluorescent in situ hybridization to detect EBV episomes. So this is an example with the EBV episome here. And we could only detect an effect when we use the P38 inhibitor, but this was only like roughly a 30% reduction. So that doesn't completely explain what we see in terms of latent EBNA2 expression. There was no effect on STAT3. So in conclusion, our model is that EBV requires the activation of both STAT3 and MAP kinase P38 in order to induce establishment of latency. These, of course, due to the intrinsic pro-inflammatory activatory effect that these two pathways have, could reduce in a very strong TNF alpha or IL-6 activation by in parallel inducing ZFP36L1, that is a known repressor of these cytokines. These could be regulated, and this would allow to have all the benefits for the virus with a very little negative effect in terms of immune response. So in the end, I would like to thank the members of my lab that took part in the project, as well as the DHZ Proteomics core facility that performed the mass spec experiment. And of course, I would like to thank you for your attention, and I'm open for questions.
Thank you very much. And there are some questions. There are no question. Sorry,
one.
Okay, there is over 2 questions. Where? Okay.
I have a question. You said that that the ZLFP, the ZFP36 is induced in virus infection as well as the tumor necrosis factor alpha. But does this mean that also, because the tumor, as we know that tumor necrosis factor alpha is also involved in it's a way that the immune system also used to destroy cells. What effect does this has on virus infected cells?
So this is exactly the model we propose, so that there is an immediate response from the cells that is inducing these cytokines, these pro-inflammatory cytokines, but the virus, by inducing at the same time P38, and that therefore is inducing ZFP, is able to control, to some extent, not completely, the activation of and the expression of these cytokines. We didn't look at the later stages when TNF goes back. So I cannot tell you like what's the effect of this second round of expression of TNF has on established latency. So this is the model we propose for the beginning. Because then when you have latency that has been established, there is a full set of other proteins that are involved that can control the process.
There is another question, or if there is no question, we can go, since we are a little bit late, to the next speaker, that is Italo Tempera from the Wistar Institute in Philadelphia. And we'll talk about PARP1 and CTCF interaction guides EBV chromatin refolding to promote viral gene expression and sustained cell proliferation during latency.
Okay, so in the meantime, I want to thank the organizer for the opportunity to present my work about how the three-dimensional structure of EBV is important for latency. And so, the success of EBV in establishing and maintaining a latent infection in the host relies on EBV ability to dynamically change gene expression during infection. And from work in the field now, we know that these gene expression programs, these latency types, are regulated by epigenetic modifications. And if you look at EBV epigenome between latency types, you observe different patterns of epigenetic markers. And we and other groups clearly demonstrate that the cellular factor CTCF is really important for the maintenance and organization of the epigenetic markers across the EBV genome. But another function of CTCF is that to partition the genome into topological domains. So the question that we have is, since CTCF can bind EBV and CTCF can organize the three-dimensional structure of the genome, that's the 3D structure of EBV really relevant for latency. And we use IC method to study EBV. However, we introduce a step here where we enrich for EBV sequencing. This has allowed us to study EBV genome with a 5K resolution. Otherwise, it was impossible to do this experiment. And what we did, we decided to look at the 3D structure in 2 B cell lines and 2 gastric cancer EBV positive epithelial cells. So we look at B and epithelial cells, and also we look at different latency type. And this is our results of the look. So this is a circle plot where EBV genome is represented as a circle. And here there is an annotation. So each of these blue lines represent chromatin interaction that connect regions of the EBV. And this in yellow represent where CTCF binding sites are along the EBV genomes. And what's really interesting for us that, if you look at this data, it's clear that the 3D genomic structure of EBV, it's quite dynamic and change between latency and change also between B cells and cell types. So in terms of latency, what we observe is that there is a correlation between the amount of the loops and the chromatin interaction that can be established across the EBV genome with the level of expression of latent genes that we observe. In fact, you see type 3 where all the latent genes are expressed as the most loop, while type 1 where only EBNA as the fewer. So, but there was another thing that was interesting to us, because when we started to compare B cells with epithelial cells, we realized there were some kind of loops that were specific of one cell types compared to the other one. So we started to think if that could be, in reality, if there are some cell type specific 3D genome conformations. So, and looking at simply at the intensity of the loops, so these principal component analysis show that it's possible to group EBV cell based on their cell origin. So basically, the structure of the 3D separate B cells from epithelial cells, suggesting that there are some specific loops that not only depend on latency, but also on their cell types. And this is an example, so how we analyze our data. So in green, you see all the loops that are in B cells and epithelial, and in red, those that are in epithelial cells. And I give you 2 examples of regions where we found profound difference. One is on REP, And what we observe in REP is that this region is engaged in chromatin loops. So it's forming some of these interaction only on B cells in green. And while in epithelial cells, there are not interaction established from that one. Another region that is interesting, it's this region around 135 kb of EBV genomes that is adjacent to one of the original lytic replications. And this region is able to form form loops both in B cells and epithelial cells. However, what we found is some specificity on epithelial cells with the TR region, suggesting that basically this region is engaged with TR more in epithelial cells than B cells. And for the introduction, of course, loops are linked to CTCF. So we wonder if these differences in 3D organization of EBV genome somehow reflect difference in CTCF occupancy between cell types. And this is ChIP-seq for CTCF across the EBV genome. We did in B cells and epithelial cells. And what we found is that when you compare CTCF profiling among B cells or epithelial cells, you see that the B cells, they express the same profile. This is, oh, sorry, there is, you cannot see. So these on, oh, no, it's on the top, sorry. So you can see these are LCL and MUTO, and CTCF binding, it's similar. And same when you compare the two different gastric cancers, NU and YCCL, you see that CTCF profiling is very similar, suggesting that CTCF binding is maintained in cells from the same origin. But when you compare between B cells and epithelial cells, now you start to see differences, and you're not surprised that you see some difference are near OREP region, and also there is a large region encompassing one of the origin of replication where seems not that there is not so much CTCF binding in epithelial cells. So that started to be interesting to us, and we wanted to study how this is regulated. So in a work that we published recently this year, we found that in LCL, CTCF is regulated by PARP1. This is an enzyme that uses NAD to transfer a unit of ADP ribose on protein acceptor. In the case of CTCF, this interferes with the ability of CTCF to form chromatin loops. And when we analyze the 3D structure of EBV in LCL before and after PARP inhibition, we found there is a dramatic change in the 3D structure. We observe there is a loss of loop formation, there are new loops. So suggesting that in reality, there is at least we can regulate some of this loop with PARP activity. So we decided to see if something similar also happens in epithelial cells. So the first things we wanted to assess if there is any difference in PARP activity between EBV positive and EBV negative epithelial cell lines. So this is a Western blot, a dot plot, sorry, assessing PAR level. And you can see that EBV positive cells, they tend to have more PARylation compared to EBV negative. And we were able to validate that PARP1 can bind across the EBV genome in both epithelial cells and the regions that are interacting with CTCF. So the question is, can we also affect 3D structure of EBV in epithelial cells by PARP1? And the answer is yes. So you can see here, this is the 3D structure of EBV genome before and after PARP inhibition. And you can see there is a dramatic change. But what is surprised us is that when we look at this, we found that yes, it's interfere with 3D structure as we observe in B cells. However, in epithelial cells, it seems that the PARP activity is important to maintain long-distance interaction. If you break or interfere with that, now the loop tend to be smaller and involving adjacent regions. So this is kind of the model that we have in mind, is that, of course, the 3D structure is relevant for EBV latency. You have differences that correlate with latency types, but also there are some differences that are specific of B cells. Some loops, they need to happen in B cells, some other loops, they need to happen in epithelial cells. And now we start to understand why, and because. So we also we found that CTCF seems to be involved in this process, and at least some of this loop can be explained by PARP activity. So we decide to get more into CTCF stories, so to see, is CTCF really required for all these loops? So we did iChip. So basically, it's the same technique of IC, except now that you pull down with the protein of interest. In our case, it's CTCF. So we study all the loops that are CTCF mediated. And this is the results. So in the top is the ChIP-seq for CTCF. And this is the signal that we have with the iChip. So interesting, CTCF established loop only between regions that are both interacting with CTCF. So it's very surprising because if you do this in the human genome, you find that that some loops, they have only one CTCF binding site. Here across the EBV, the loops require both CTCF. So as we met, and there were some regions, if you look at the intensity, they seems to establish more loop with high frequency compared to other ones. So since CTCF loop involve promoter and enhancer, we try to see if, looking at this map, we might identify some other regions that might have, and can be regulatory elements. And to do that, we overlap our IC map with histone marks. They are specific of active enhancer, like H3K27 acetylation and H3K4 monomethylation. And what we found, there are 4 regions of EBV where you have chromatin loop by CTCF, and they have the histone mark that's similar to those to active enhancer. And of course, OREP, everyone knows that OREP is an enhancer for regulation of CP and EBNA2, but we identify potentially 3 regions they might have a function as an enhancer. And this is also matched with data that has been published before, and interesting, with a very exciting observation from Bozau group at Harvard that also has made similar observations. So what we found that interesting, especially the region adjacent to orelite and the TR, they have characteristic, a feature of enhancer, and they might play a role in regulating latency, or as we heard, for previous talk, maybe orelite has doing something more than just an original replication, maybe something more regulatory that we anticipated. And with that, I want to thank all the people that make possible this work. If you're in the lab, the bioinformatics from Andrew Kosenkov, our collaborators, Ben and Rui, that helped for the project, and I will happy to take any questions. Thank you.
Okay. Thank you very much for keeping the time at the hand.
I know, I was the only person between the audience and the lunch, so I realized that.
So there are some questions for okay, there is one question over there.
Hi, thank you for the talk. I was wondering if you've been able to explore high C with host and virus, or if there's a detection sensitivity limitation without the enrichment of the EBV genome.
No, actually, with that capture, we are able to get a better sensitivity between EBV human, because also you catch that one. It's more variable that compared to EBV, because the majority, of course, is EBV, but we use that one, and we're able to identify regions that interact. And actually, we found that most of the reports showing OREP. However, we identified that also the orelite and TR might be involved in tethering between EBV and human.
Great, thank you.
Great talk.
Thank
Just
you.
a question, like the difference between the B cells and the gastric cancer cells, how much play a role that there's copy number differences and then heterogeneity between genomes within singular cells or individual cells?
Okay, so I start for the first one because it's simple. So since we normalize some of the data, I think the copy number might be involved, but we have to find a better way to spike in something. So for right now, it seems not, and we think, but we need to find a better way. For the second one, there's a lot of limitation when we do this, because when you do this analysis, ChIP-seq, IC, we study basically a cell population. Now, even if you go to single cells, in every single cell, you have multiple episomes. In other words, so what I show you, these are the loops. But what we think is that not all the episomes inside the cells, they have the same loops. And you don't need a genome having exactly all the same loops to be expressed, for the same reason that you might simply need one episome and 49 repressed to still have maintenance. So we cannot address the question with IC, but we had tried to address that one using DNA methylation study and see if we can see epialleles. And for B cells, it seems that it's not so many. So all the episomes seems to be similar. Let's put it this way. On gastric cancer cells, instead, you start to observe population of episome based on methylation. We cannot, but only with methylation, I cannot make any comments because there are some limitation in the techniques.
The last two questions, and then we go for lunch.
So trans-episomal means between two episomes. The quick answer is no. And we try to figure out, and we cannot rule out that, actually we believe that it might be possible that there is multiple concatenation, but in that case we cannot distinguish. And with multiple episomes, we try to figure out if we can label episomes and get that once. But yeah, that we cannot rule out, but I think it's possible.
Hi, thank you. Nice talk. Can you distinguish different chromosomal arrangements based on different states of DNA repair?
Answer is not yet, but we think, especially if you consider PARP1 is involved in DNA repair, it might be possible. And also it might be possible that during replications, not leading, just latent replication of the genome, there might be something else occurring. And I think also, if I add a layer of complexity, there is also splicing and other elements that I think influence the 3D structure. That's not surprising us that you see more loops when there is more transcription.
Okay, the last question, and then we go for lunch.
Hi, fantastic talk. Actually, CDC binding site is rather rare in gastric epithelial cell compared with the B cell. But is that because of DNA methylation status? Actually, episomal DNA is highly methylated in gastric cancer, but CDC may not bind. Is there any effect on DNA methylation on the difference of high C status between those two types of cells?
So your question is how DNA methylation level influence the structure?
Right. Actually, yeah, today's talk is mainly on CDCF or
Yeah.
PARP, but how about DNA methylation?
Yeah, that's a great question. So I didn't have time to show the data, but what we found on epithelial cells that it's the highest level of methylation that you can observe in EBV, similar to Mutu sometimes, but gastric is much more. Short answer, where you have a CTCF binding, you don't see methylation. The methylation happens either upstream or downstream of CTCF binding, except for CP, where we think there are two different populations. The second thing is that when you have methylation and heterochromatin, the genome, it's physically less flexible. So, and then that's probably reflect some of these loops, because you have to think if you bend something that is already unflexible, require a lot of energy and cannot be maintained. But definitely, I think has an effect on that.
Okay,
Thank you very
much again. And I think we can go for lunch. You are welcome for lunch this side. And we start again at 2:30. Thank you.
Thank you.
So while you are having your lunch, you are also welcome to check out some very interesting posters. Thank you.
Quello che deve essere il progetto temporaneo. A meno
Just a moment. There is a problem. Ha tirato via il filo e ha spaccato tutto. Lei?
Io? Ma guarda, il filo era già prima.
No, ma ora proprio la... Miseria.
Are you using
that one to forward the slides?
I think it should work.
That's because I'm used to a Mac, and so I just wanted to.
Yeah, yeah. Maybe it's this one here. This one. We can check. Che è successo?
Ha tirato via un cavo e...
Io l'ho rimessa a posto, però mi sembra un po' danneggiato il cavo.
E ora ci ha riappuntato un'altra volta.
Eh?
Ora ci ha riappuntato un'altra volta e quindi si è...
Accendiamo un tablet. Perché?
Non ho capito, ora bisogna capire. No, ora l'ha ritirato quando si è rialzato.
La seconda volta. La seconda volta.
Perché prima gli ha dato vabbè, già fatto.
Quindi non parte più.
No, ma penso la giusta. Penso ora la giusti, non credo sia l'attacco del... sarà il cavo.
Shannon, we have some issues with the cable. Yeah. He's going to fix it. I think he's getting over the cable, so it should be okay.
Credo che il cavo sia un po' andato. Non prende proprio il segnale. Andato.
Come la chiamo?
It's working.
Ok.
Vieni a mangiare poi.
No, sì.
È meglio. Allora vieni a bere.
Allora, chi c'era?
Eccola, Shannon.
Ho aggiustato perché non aveva fatto duplica schermo così. Aspetta.
No, fermo. A posto. No,
adesso
perché non era selezionato lassù. Lo perde da lassù poi.
No, ho fatto duplica schermo, ho fatto così.
Sì, ma per...
Era
No,
messo solo PC.
no, non c'è bisogno di fare quello. Perché la matrice video lassù non era selezionato. Quando l'ha staccato...
Allora è stata una coincidenza temporale il fatto che l'abbia fatto.
L'ho pigiato da lassù. Perché quando lo perde va riselezionato. Pensavo che invece lo riprendesse da solo. Vedi, c'è un selettore ingressi qua sotto.
Sì, no,
No, basterebbe che non ci si...
outlet and then press it again. Okay, see that.
Oh, I see.
want to do it? Then we'll come back. Okay, well, welcome back to the next session. So we're starting off, where is Bill? So we're starting off with a topic lecture from Bill Sugden, who's going to be talking to us about what Epstein-Barr virus contributes to B-cell lymphoma. Bill.
There we go. I'll repeat myself. Thank you. Thank you for the invitation to give this talk. And let me describe that this is really going to be work done by Aurelia Four, Mitch Hayes, Rebecca Hutchinson, Dave Verady, and a tiny bit by me too. And what I'd like to do is address this topic, what Epstein-Barr virus contributes to B-cell lymphomas, from two sides. One side is to just consider what EBV confers upon primary B cells when it infects them. What are those attributes that might contribute to B-cell lymphomas? And then the other side is to take bona fide B-cell lymphoma cell lines and force the loss of EBV from them and ask what happens. And by understanding what happens, we can then infer what EBV was providing those tumor cells. So there'll be two parts, or one talk from two sides. Epstein-Barr virus, we know, confers multiple properties to B cells infected in vitro that are shared with EBV-positive lymphomas. And two essential ones are proliferation and survival. And I'm going to focus on them. But let me first admit that we also know that EBV plays a really profound role in regulating an immune response to EBV-infected cells. For example, Wolfgang Hammerschmidt and his colleagues, EBV encodes microRNAs that really limit CD4 and CD8 responses to EBV-infected cells. And actually, we've shown that EBNA1 limits recognition by natural killer cells. So EBV has multiple functions to inhibit immune responses to infection, but I'm going to focus on cell intrinsic properties, proliferation, and survival. And let me just say, we know that EBV has to be doing something proliferative by looking at what happens to B cells put into culture in the absence of EBV. And this work is really noted in the references below. We know that if you take primary B cells from the peripheral blood, if you take them from GC cells, that they can proliferate in the presence of cytokines. But you all know you can transform primary B cells with EBV and they'll grow in the absence of cytokines. So I'm going to elaborate on that kind of proof that EBV provides proliferative signals to infected B cells from a funny angle. I'm going to show you work we've done, particularly Aurelia has done, on infecting primary B cells with two viruses, EBV and KSHV, to model primary effusion lymphomas. So she set out to ask the question, could she generate a cell culture model for primary effusion lymphomas by infecting B cells with both EBV and KSHV, such that cells could be transformed, grow out, eventually become immortalized, and maintain both viruses. Now, at the time she began this work, we knew that tonsillar B cells can be propagated in cell culture with CD40 ligand and IL-4. When they're infected with KSHV, they'll grow for a while. You have to have these cytokines, but then those cells die. So we know that KSHV infection of B cells cannot maintain them for more than a few weeks, perhaps a month, or in a proliferative state. So Aurelia set out to ask, could she infect these cells? Could she get both viruses into one cell? And would that one cell continue to proliferate? And the answer is yes. It's not terribly efficient, by the way. You can get about 2% of those cells infected, and they don't all clone, but you can do this, or she can do this. So EBV supports proliferation of co-infected cells. Both it and KSHV are maintained as plasmids in duly infected B cells in vitro without added cytokines. And proof of their maintenance in the same cells is shown in these two images. This is fluorescence in situ hybridization to detect KSHV in red and EBV in green. Both plasmids are in all of the cells. Well, in this case, most of the cells. This is an early on experiment. So we know that really EBV is supporting proliferation because KSHV could not in these cells. Okay. Mitch Hayes, working with Wei Wong and Paul Lambert, have carried this further. And let me show you what they've done. So when you infect primary B cells with EBV and KSHV, most of the time, most of the cells are being infected with EBV. And again, it's a minority that get co-infected. They took populations of cells that were co-infected and grown only in cell culture. And what would happen over time, almost always, is that the EBV-infected cells outgrew those cells that were co-infected. And I think, by the way, that's really why these are not trivial experiments to make work, because usually the cells infected with EBV alone, and there are many, many more of them initially, outgrow those cells that are infected with EBV plus KSHV. Well, they took mixed populations of these cells where there were about 10-fold more infected with EBV than with KSHV and EBV, and put them into the peritoneal cavity of non-skid mice. And 28 days later, it was quite impressive. The co-infected cells grew preferentially. So by the time they tapped those mice, there were about an equal number of cells co-infected with KSHV and EBV as with the cells infected by EBV alone. So here's a point. The two viruses in one cell confer upon that cell a massive growth advantage in the peritoneal cavity of non-skid mice, making it, I think, a pretty good model to consider studying for to use for primary effusion lymphomas. Mitch took cells in the two following categories: cells that were purely duly infected but had only been grown in cell culture, and we can purify them because the KSHV encodes GFP. So you can use FACS to pull out co-infected cells, and this he did. And then he took the cells immediately out of the peritoneal cavity of the non-skid mice, isolated RNA, and did RNA-seq, and the results are shown in that volcano plot on the right of this figure. And I just want to illustrate that what this is showing is that some genes that KSHV and EBV regulate go up, and some go down. But then if you look at what those genes are, they're really quite informative. So I'm just going to use two as an example. IL-10 goes way up, and that's up in the top right of the figure. IL-10 is going to promote the growth of PELs and co-infected cells dramatically. DUSP4 goes down. DUSP4 is pro-apoptotic. So this pro-apoptotic signal is being inhibited as you passage the co-infected cells as tumors in non-skid mice. So EBV and KSHV in co-infected B cells grown as tumors in non-skid mice regulate cellular genes to inhibit apoptosis and promote cell proliferation. And examples include DUSP4 is downregulated to inhibit apoptosis, IL-10 is upregulated to promote cell proliferation. So what we can say is that co-infected B cells, when grown in vitro for EBV, or as tumors for both viruses in mice, demonstrate that EBV and KSHV together promote survival and proliferation. Okay, I came from one end. We were infecting cells in this case with both EBV and KSHV. Let me come from the other end. And let's consider, or ask you to consider, what happens when you have bona fide EBV tumors in cell culture, but haven't been propagated for long in cell culture. We made sure of that. And now force the loss of EBV from these tumor cells. What happens to them? Because by understanding what happens to them, we can infer what was EBV doing to those B cell tumors when it was in them. So that's the idea of the experiment. And this part of the experiments were done by Dave Arady. So he wanted to force the loss of EBV from B cell lymphomas to uncover what contributions EBV makes to these lymphomas. So imagine you have a B cell tumor, OKU1, I never can say this, PTLD1, primary PTLD1, I'm going to call it that. You have these two tumor lines, and now you engineer them so that they will express a dominant negative derivative of EBNA1. This is a derivative of EBNA1 that still binds all the sites that the endogenous EBNA1 will bind, but it lacks the AT hooks of the aminoterminus of the protein, so it can't tether EBV DNA to chromosomes. So over time, EBV is going to be lost when you have an excess of this dominant negative derivative. EBV is going to be lost from these cells. And now you can ask, what happens? So he did this with two cell lines, multiple clones of these two cell lines. Post-transplant lymphoproliferative disease, I said it, PTLD, and WP-restricted Burkitt lymphomas. WP-restricted means that these tumor cells have, or the EBV in these tumor cells have a deletion in EBNA2. And one thing I think that can immediately lead us to expect is that these tumor cells have evolved in vivo to become less dependent on EBV because the EBV is not expressing EBNA2, and we kind of know you have to have EBNA2 initially to transform cells. So when you think about comparing and contrasting PTLDs and WP-restricted Burkitt lymphomas, the latter have probably evolved in vivo to be less dependent upon EBV. So what did Dave find out? Let me show you his experiments. So I'm going to show you two slides. They're the same format. In these experiments, in the y-axis, you see the number of doublings. What's the increase of the cell number? And on the x-axis is time. And these cells have been engineered to have a conditional derivative of the conditional dominant negative derivative, meaning he can turn on a switch and turn it on. It's not on all the time. So he could turn it on and then force the loss of EBV and ask over time what's happening. He also had learned by the time he made these clones that if you force the loss of EBV from either of these cell lines, they die by apoptosis. So he also engineered them to have either a gene expressing BCLXL, which will inhibit apoptosis, or an empty vector. So there are four flavors of constructions you're seeing in this particular slide. When he did not force the loss of dominant negative, did not force the expression of the dominant negative derivative to force the loss of EBNA1, whether or not you had BCLXL in that cell, you see the straight line, the orange line and the black line. This is a semi-log plot, and what that's telling you is these cells are growing logarithmically. The expression of BCLXL in them doesn't matter at all. They're just growing logarithmically. They're happy in cell culture. But now look what happens when he flips the switch and begins to express the dominant negative derivative of EBNA1 at time 0. Please first look at the black dotted line. There's no BCLXL added to that cell line. And what you can see is that it's stopped proliferating. It's gradually diminishing in the number of cells. And in fact, if you go out for another 4 days, they're all dead. So in the absence of EBV, he's forced the loss of EBV with the expression of the dominant negative derivative of EBNA1. In the absence of EBV, these cells are dying by apoptosis. And he showed that with single-cell apoptotic assays, from which we can infer that EBV is blocking apoptosis in these cells, okay? EBV is essential to block apoptosis in PTLDs. Now please look at the dotted orange line. Those are the cells in which he is evicting EBV genomes, but he's also expressing BCLXL. So he's inhibiting apoptosis with BCLXL. Look what happens. Those cells are plateauing. They're not proliferating. They're not dying. And in fact, he's carried this for 30 days, and the cell number remains constant. They're not proliferating. And what that tells you is that EBV must be providing pronounced proliferative signals to PTLD1. And we know from the black dotted line, it's blocking apoptosis. So that's two profound contributions to PTLD1 or PTLD cells made by EBV. Now let's look at what happens for WP-restricted Burkitt lymphomas, the cell line OKU1. Same format of the experiment, same constructions. And again, if you look at the solid lines, orange and black, where there is no dominant negative turned on, those cells are growing logarithmically. It's a straight line on a semi-log plot. Please focus on the black dotted line. There's no extra BCLXL in that cell, but the dominant negative has been turned on at time 0. you can see that the cell number plateaus, declines, and if you go out another 4 days, you ain't got no live cells. So again, we can conclude that EBV in WP-restricted Burkitt lymphomas is essential to block apoptosis. Okay. Look what happens to the orange dotted line. There, BCLXL has been provided. EBV is being evicted. 90% is gone by the day 18, I think. So and that means those are EBV negative cells, not one copy, they're negative. But look what happens to them. They're not dying. BCLXL has been added to them. And they're sort of proliferating weakly. They're limping along. So that by day 24, they're 2% as many cells, but they're not dead. And I think what that tells us is that EBV in this Burkitt lymphoma cell line is providing some proliferative signal, but nothing as profound as, for example, it was to PTLDs. So, and obviously, no, I didn't say that correctly. EBV is providing a pronounced proliferative signal, and in its absence, those cells still can proliferate, but weakly. So what we're observing is that EBV provides proliferative signals to the cells, but not as profound as to PTLD1. Okay. Gotta go back for a moment. So let's consider for a moment, what are the cellular genes that are being regulated by EBV to inhibit apoptosis in PTLD1 and in OKU1 cells, the WP restricted? We kind of know. So Dave measured, for example, BIM. BIM is incredibly pro-apoptotic. And when he forced the loss of EBV from PTLD cells, BIM levels went up between 5 and 50-fold. When he forced the loss of EBV from OKU1 cells, BIM levels went up 2 to 4-fold. That's perfectly consistent with when you lose EBV, you're losing EBNA3A, you're losing EBNA3C, and as many people have shown, EBNA3A and 3C block the expression of BIM. For example, Martin Alday and his colleagues have shown that. So by losing EBV, you're losing EBNA3A, you're losing EBNA3C, you're losing their downstream inhibition of BIM expression, and BIM is pro-apoptotic. And then work that we've done with Wolfgang Hammerschmidt has shown that the microRNAs of EBV actually lead to the destruction or the inhibition of caspase-3. So when you lose EBV and you lose their microRNAs, now the levels of caspase-3 go up, it's pro-apoptotic. Again, apoptosis ensues. So let me pose a question. EBV is really using multiple mechanisms to block apoptosis. Could there be more? And we think the answer is yes. Let me just briefly outline work that's being done by Rebecca Hutchinson. She's going to talk about it tomorrow. So which cellular genes mediate the apoptosis blocked by EBV in WP-restricted Burkitt lymphomas? And she's has addressed this question by performing a genome-wide CRISPR screen in OKU1 cells. And then she gets a huge number of cells that survive this screen, and she evicts EBV from them, again, using CRISPR-Cas9 with guide RNAs targeting EBV. So you now could ask, which genes in those that survive, that have been activated by the initial screen, foster growth in the absence of EBV? And that's a remarkable finding. Rebecca, and she's going to describe it in detail tomorrow at 9:55. But in brief, 70% of the identified cellular genes, so these are the genes which are inactivated and allow those cells to proliferate in the absence of EBV, 70% of the identified cellular genes encode the Hush complex or genes that interact with the Hush complex. What is the Hush complex? Because I didn't know it until she discovered or found it. Other people have found it too, obviously. Hush regulates the deposition of epigenetic marks, H3, K9, methyl 3. So that's a fascinating finding. So, first summary. Epstein-Barr virus contributes anti-apoptotic and often pro-proliferative functions to B-cell lymphomas. EBV blocks apoptosis by multiple means, including inhibiting expression of BIM, degrading caspase-3 messenger RNA, and affecting the function of Hush. It drives proliferation in PELs and PDLDs, but contributes only modest proliferative signals to WP-restricted Burkitt lymphomas. Okay, now I'd like to go in a totally different direction for a moment and ask you to consider something about all of the information that EBV is providing to promote this B cell tumors. So let's just consider for a moment. Today we have something like 7.8 billion people on the planet. And we say 90% of us are infected with Epstein-Barr virus. Well, that turns out to be there are about 7 billion of us that are infected with this virus. And you know, if you look at sort of the papers that compile what tumors are caused by EBV, they're on the order of 200,000 new tumors, new cases of cancer associated with EBV-infected people each year. So you can divide one into the other, and you're going to come up with a number, if I did my math correctly, that about 1 in 35,000 people infected with EBV each year develops a tumor associated with EBV. Of course, for the person who develops that tumor, that's hell. But think for a moment about the other 34,999 of us who are infected and are not developing EBV-associated cancers. All of this information that is so imperative for EBV to contribute to tumor formation must be contributing something to the normal life cycle of EBV in all of us. That's an amazing thought. Thank you. Can I take questions?
Thank you. So we have a question down here. If you mic, Samita.
Very interesting talk, Bill. Can you hear me? Okay. So the Hush complex is essentially the constitutive heterochromatin machinery.
Yeah.
Sorry. The Hush complex is essentially the constitutive heterochromatin machinery that brings about H3K9 trimethyl marks by ZTB1,
Right.
the histone methyltransferase. And in the context of both EBV and KSHV maintenance of latency, H3K9 trimethyl marks are very important, and this complex is known to be very important in our process. So what is unique about this co-infection scenario that makes you think that the Hush complex is exclusively or primarily involved in the silencing mechanism that contributes to the maintenance of co-infection?
So let me repeat the question and make sure I'm getting it. What is peculiar about the Hush complex such that it could contribute? So these are just EBV positive tumors.
Right.
Somehow to their, and let me be explicit. It's only when the Hush complex is inactivated that you can afford to lose EBV. So EBV must be doing something to the Hush complex and those other molecules that associate with members of the Hush complex to limit its function so cells survive. If you ask me what it's doing, I would desperately love to know, but I'm ignorant. I don't know.
Let's talk about this later.
Sorry, we have
yeah.
a few more people waiting. Maybe you could pick it up in the break. So we have a question down here.
Thank you. Bill, the double positive cells that you isolated from lymphomas, that you isolated from non-SCID mice,
Yes.
Do they recapitulate the EBV latency that we see in PEL? What
Yes.
kind of latency do they have?
Yes, they do. So the question, oh yes, everybody heard you. Yes, they do.
Did we have another question? Yeah, there's one more there.
Yeah, it was very interesting. I was also wondering about these double infected cells that you transfer in the not-SCID mice. So not-SCID mice still have some NK cell reactivity, mouse NK cell reactivity. So do you think this residual NK cell reactivity could enrich for the double infected cells? Or do you rather think that the double infected cells have a benefit in growth in vivo in the not-SCID mice?
I'm sure they could. All we can say is that whatever is allowing them to win the race with the cells infected with EBV only, that is giving rise to the differential gene expression that also clearly is driving proliferation and survival. But yes, I think to your question.
Okay, thank you very much. So we'll move on to the next speaker. Well, if I can find my glasses. who is Alejandro Casco, and he's going to be telling us about what can a fluorescent Epstein-Barr virus tell us about host shutoff.
Can't read that. Okay. Good afternoon, everyone. So my name is Alejandro Casco, and I am a PhD candidate in the lab of Eric Johansson's. And today I'm going to be presenting my work titled, What Can a Fluorescent Epstein-Barr Virus Tell Us About Host Shutoff? So host shutoff is an important feature of many herpes viruses that allow this virus to effectively suppress antiviral responses while simultaneously reallocating cellular resources for its own viral replication. And in EBV, by the BGLF5 protein, which targets and degrades cellular mRNAs in the cytoplasm. However, the extent of this degradation, especially in EBV, is poorly characterized. In other words, we don't really have a great understanding of which cellular genes are escaping this host shutoff and how they may contribute to the progression of viral replication or perhaps limit viral replication. And as many of us know, the primary barrier to studying lytic replication is that it is inefficient in many of the methods to induce lytic reactivation are highly inefficient, and this is especially true in physiologic cell lines. And this results in a minority of cells that are able to support lytic replication. And furthermore, cell culture is by nature asynchronous, making it difficult to separate the effects of early versus late replication. So we wanted to address these limitations and develop a method that would allow us to enrich cells in the early and late stages of replication to better understand the effects of host shutoff. So to do this, we generated a fluorescent reporter EBV that expresses a green fluorescent protein driven by an early BXLF1 promoter and a red fluorescent protein driven by the BILF2, a late promoter. And these are two non-essential genes. And then we can couple cells harboring this reporter EBV with FACS and sort cells that are in the early and late stages of EBV replication. To show the sensitivity of this approach, we infected 293 cells with this reporter EBV and performed flow cytometry analysis. And what we see is that when the cells are not induced into the lytic cycle, there are no cells that express GFP and RFP, and then therefore not in the lytic cycle. When we transfect the cells with RNZ to induce the lytic cascade, we can identify with great sensitivity and distinguish cells that are supporting the early stages of replication due to expression of GFP and cells that are in the late phases of replication due to expression of GFP and RFP. So we can also quantify the purity of this approach by rerunning cells that we sorted through FACS. And what we find is that cells that are not in the lytic cycle, which we term colorless, we can achieve nearly 100% purity. Cells that are in the early phase of replication, we're able to achieve about 99% purity, and cells in the later phase, we're able to achieve over 96% purity. And so now we can start asking more interesting questions. But to further validate this approach, we wanted to perform a pilot RNA-seq experiment, again, using 293 cells infected with this virus. And these cells are inducible with treatment of 4-HT due to the stable integration of ZHT. And we sorted 5 populations, an uninduced population, an induced but unsorted population, and then the 3 subpopulations that were induced, the colorless and the lytic early and late populations. And we sorted these in equal number and doped each condition with an equal amount of ERCC spike in RNAs. And this is a very important consideration for downstream RNA-seq analyses that allow you to normalize accurately. And furthermore, because we are adding known molar amounts of these spike in RNAs, we can estimate the total RNA molecules in a cell, as shown on the y-axis. And in this figure, I'm showing total viral RNA molecules in each condition, and color-coded are the gene expression kinetics. And we can see in the colorless fraction, we have a substantial drop of viral gene expression relative to the unsorted. And in the early fraction, we see viral gene expression primarily, that is, of early gene expression kinetics and leaky gene expression kinetics. And finally, in the late fraction, we see a substantial increase in viral gene expression, importantly of late gene kinetics. When we look at total RNA molecules in the cells, we can see that as the lytic cycle progresses, there's a substantial drop in total RNA molecules, shown in light blue, relative to cells that are not in the lytic cycle or are unsorted, or uninduced, I'm sorry. And there's also this increasing viral gene expression, similar to what I showed in the previous slide. And what this indicates is a host shutoff event that is not only promoting viral replication, but also viral gene expression. So to better observe the utility of this approach, we can look at different patterns of gene expression during the early and late stages of replication relative to the uninduced conditions. And what we see is that during the early phase of replication, there is a massive host shutoff event of over 10,000 cellular genes that are significantly downregulated. Interestingly, some genes are able to escape this host shutoff and are actively expressed during the early phase of replication. And there's a subset of these transcripts that become downregulated in the late phase, and then another subset that are commonly expressed throughout the lytic cycle. So in the next few slides, I'm going to show some examples of these genes. And I'm going to start with those genes that are commonly expressed throughout the lytic cycle. So we performed a gene set enrichment analysis that groups these genes based on their biological functions. And what we find is that these pathways are primarily enriched in transcription and DNA damaged stimulus. To show some examples, we can look at the transcript that encodes the RNA polymerase II, and we see that it is similarly expressed in the uninduced and the cells that are supporting lytic replication, with a slight but insignificant increase in expression during the early phase. We can also look at a CDK12 transcript, and this transcript encodes a kinase that promotes the elongation phase of RNA polymerase II. And again, we see similar levels of expression. What's perhaps more interesting is those subset of genes that are actively expressed in the early phase, but become downregulated in the late phase, as some of these have antiviral functions. For example, RIPK3 is a gene that is involved in suppressing the replication of viral genomes by enhancing ROS production, which can lead to cell necrotic death. And what I think is particularly interesting is that this gene is expressed at relatively low levels in the early fraction. And what that may suggest is, because we're using bulk RNA-seq analysis, within this early fraction, there may be a subset of cells that are undergoing successful antiviral responses that limit the progression into the late phase, hence the lower expression in that late phase. And these type of genes would be fascinating to pursue in the future. We also performed gene set enrichment analysis, all the genes, and we find that the most enriched pathway is that of hallmark MYC target genes that are significantly downregulated. Indeed, the MYC transcript itself is significantly suppressed. And this is consistent with the recent publication by the Benjamin Gord's lab that found MYC to be a crucial antilytic factor. Although we think about host shutoff as being this RNA degradation-mediated event by BGLF5, there are likely alternative mechanisms of host shutoff we don't necessarily think about. For example, this XOC4 transcript that is slightly downregulated, is involved in exocytosis, but is slightly downregulated in the early fraction, still shows considerable levels of expression. However, during the early phase, EBV seems to induce an isoform switch, resulting in a non-coding transcript. And we can see this in the bar graph down here, where in light gray, we have the early fraction, and in dark gray, the uninduced fraction. In isoform 2, we can see there's preferential expression of it in the early fraction, and it is non-coding. When we look at isoform switching events at a global scale, specifically with genes that are actively expressed in the early phase, we can look at the consequence of these isoform switches. And we identify about 70 to 80 transcripts that undergo an isoform switching event that results in a complete open reading frame loss, such as XOC4 that I just showed. There are also other consequences, such as perhaps loss of important domains, or making the transcript sensitive to nonsense-mediated decay. Again, just showing different methods, potentially, of host shutoff that are not related to RNA degradation. Of course, that was only to show the utility of this virus by using 293 cells, but our main objective is to do this in physiologic cell lines where the immune system is likely more competent. Specifically, we want to do this in lymphoblastic cell lines to model B cells and epithelial cell lines to model, NOx to model epithelial cells. And it would be fascinating to identify different antiviral responses between LCLs and NOx, as well as those common antiviral responses. We certainly would want to further interrogate those genes by knocking them out and overexpressing them. But again, what would be especially fascinating would be flow cytometry analyses looking at these antiviral responses at a single scale level. and potentially determining heterogeneity in these antiviral responses, which may give some insight into this complex virus versus host arms race, where sometimes most of the time I suspect the EBV wins, but maybe there's a small subset of cells where the cell incites a successful antiviral response. In conclusion, I have shown that the fluorescent reporter EBV allows efficient enrichment of cells undergoing lytic replication, and this enables us to perform high-resolution transcriptome analyses of host gene expression. And we did this using a pilot RNA-seq experiment in 293 cells. But it really paves the way for future exciting analyses and more relevant models. And with that, I would like to thank the Johansson Lab, my committee members, and of course our collaborators at Sugden, Kenny, and Mertz Lab. And I will take any questions.
Thank you. Do we have any questions? Okay, we have a question up. Oh, we have one down here, and then we have one up there at the back.
Thanks for the good presentation. Just one concern. In your findings, you saw that the MYC gene is suppressed, but we know that the MYC gene is usually overexpressed in bucket lymphoma. How do you explain that? And also, you know also that EBV contributes to bucket lymphoma.
That it is overexpressed in Burkitt lymphomas. In lytic state?
Generally, the MYC gene is overexpressed in bucket lymphoma.
Yeah, that was only really meant to model. I mean, this is 293 cells. So I imagine there's a lot of immunity that is not competent in 293 cells, but it's really meant to model this idea that there is likely some antiviral responses that we might be able to observe at a single cell level that may limit progression into the late phase. Yeah, it's only meant as an example of this idea of antiviral responses and the heterogeneity. Yes.
Thank you. So we had another question here. Thanks.
Thank you again. Very interesting model you've established. So my question is regarding the isoform switch that you talked about. So how common of a mechanism is that? Is that limited to only a few transcripts? Is that very common? And if yes, is there also an overarching mechanism that leads to these isoform switches? Can you comment on that?
Yeah, I definitely can't comment on a mechanism. Perhaps, there are some EBV encoded genes that are involved in RNA export and RNA binding, such as SM. Perhaps those are eliciting functions that result in these isoform switches. But what we found, it wasn't a significant amount of transcripts. It was about, at least for the complete open reading frame loss, we could only observe about 80 of these transcripts. Was there anything else to your question?
Thank you.
Okay. Thank you very much. So we'll move on to the next talk. So our next speaker is Elliot Sorrell, who's going to be telling us about time-resolved multiomics, reveals distinct B-cell fates and gene regulatory signatures in the early stages of EBV infection.
Good afternoon, everybody. Thank you to the organizing committee for giving me the opportunity to speak today. My name is Elliot Sorrell, and I'm a postdoc in Mika Luftig's lab at Duke University. And today I'd like to tell you about some of our recent time-resolved single-cell sequencing experiments. So I'd like to introduce our work in the context of the germinal center model that was originally developed by David Thorley Lawson and has since been experimentally refined by many groups, including our own. This GC model essentially captures the progression through viral latency states that affords access to a latent memory B-cell pool, essentially by mimicry of B-cell responses to cognate antigen and lymphoid tissues. However, we know that successful establishment of latency in memory B-cells is not the only possible outcome to infection. So prior work in the lab has demonstrated that the hyperproliferative dark zone-like expansion that the virus induces can lead to significant replication stress that leads to DNA damage and subsequent induction of p53-mediated senescence. More recently, we've used single-cell methods to examine heterogeneity within lymphoblastoid cells that are successfully immortalized. And even within this cell culture model, we see significant heterogeneity that captures aspects of the germinal center reaction dynamics, particularly a light zone-like phenotype that would be considered a classic latency 3, but also post-germinal center B-cell effector states like differentiated memory B cells. A number of groups, including the Hammerschmidt, Lieberman, and Zhao groups, have used time-resolved methods to establish really important insights into early infection. But it's clear that these distinct cellular fates manifest simultaneously and also, in fact, asynchronously. And so because of that, we wanted to use single-cell resolution to try to be able to dissect them and better explore them. And so to do that, we started by collecting enriched B cells from the blood, peripheral blood of 2 donors. We collected samples of those cells before and at 3 time points after infection with the B958 strain of EBV at days 2, 5, and 8. And we cryopreserved those samples. And simultaneously, we validated both B cell enrichment by CD19 expression and a successful viral infection by the induction of CD23 expression over that time course. Once we knew we were working with quality samples, we worked with the Duke Molecular Genomics Core. We thawed the cells simultaneously, enriched them all to greater than 90% viability, and then we prepared them using the 10x Genomics Multiome Kit into cell-matched single-cell RNA and single-cell ATAC-seq libraries to get gene expression and chromatin accessibility information. We then sequenced those libraries on an Illumina NovaSeq. Those base calls were assembled into reads. The reads were mapped against a concatenated reference genome containing HG38 and the type 1 EBV consensus sequence. And those mapped reads produce count matrices, which we analyzed using Seurat, Monocle 3, and a number of custom informatic methods. Here is a global representation, a dimensionally reduced representation of the changes in transcriptome over the course of our early infection experiment that have been merged together by time point. And I want to emphasize that within 2 days, we see virtually a complete loss of the resting cell transcriptome that we observe at day 0, which is shown from the blue to green transition here. And then by days 5 and 8, we see the additional formation of a number of additional distinct phenotypes. We performed unsupervised clustering on this merged time course single cell data, which allowed us to preliminarily identify a number of distinct phenotypes that arise in the early infection time course. And we started out by looking at percentage of viral reads, mitochondrial reads, and total feature mRNAs per cluster that was identified, which is depicted here. And the gestalt of that is that clusters 6 through 5, which are depicted in the kind of yellowish to red hues, look like successful infection trajectories from a viral perspective, whereas clusters 4 and 7 in green look like unsuccessful trajectories. They have much lower feature content. They have virtually no to low viral reads, although viral reads are present. And of course, we have our uninfected cells, which are shown in purple and blue, which are our resting memory and naive cells. And as a sanity check, when we look at the induction of interferon response genes, we can see that every cluster except for our resting cells have some level of an antiviral response that's mounted. So when we dive into specific clusters more thoroughly, we can get a better appreciation for some states we recognize, but also some novel phenotypes. So the first one I want to talk about is one that is defined here as a pre-activation or antiviral response. That's sort of the earliest stages of B-cell response to infection. And you see the holdover of some genes that were present in the resting B cells, and you see the beginnings of the mounting of an antiviral response. So cluster 4 here was most present within day 2. It peaked at day 2. However, there were still cells at days 5 and 8 in our time course that had this phenotype, which is indicative of the number of cells that fail to ever progress past this initial innate antiviral sensing phenotype. By contrast, we also found what has been described in the B-cell literature as an activated precursor or early memory B-cell state, which is a pre-germinal center multipotent state that can give rise to low affinity early memory B cells if they have limited access to antigen, or can contribute to cells that actually engage in germinal center dynamics if they have increased accessibility to antigen. Those cells then undergo the GC reaction affinity maturation. One of our most favorite clusters from our time course was a cluster that discretely separated out based on its expression of cell cycle markers. This is akin to the dark zone-like hyperproliferative state. Again, peaked at day 2. It was the other major state besides the early arrest. And some cells still persisted in this state at day 5 and less so at day 8. There was a cluster that was similar to the hyperproliferative phase that also had elevation of some cell cycle markers and clearly had undergone some extent of proliferation, and yet it was typified by its upregulation of oxidative stress genes, DNA damage response genes, and particularly a broad host of ribosomal gene transcripts that were indicative of ribosome biogenesis-mediated senescence. So these are stress-induced arresting cells in early infection. By days 5 and 8, the most prevalent states we saw were a cluster that looks like our classic light zone or latency 3 NF-kappa B activated state that has apoptosis resistance and host biomarkers of latency 3 that have been previously described. And the other that we saw was clearly a post-germinal center differentiated B cell state that looks like memory B cells or pre-plasmablasts based on BLIMP1 and XPP1 and many other genes. So time-resolved sampling alone cannot tell us the distinct outcomes of infection. But with these annotations in hand, we applied other informatic methods to help try to parse the trajectories from one fate to another. And the one I have shown here is called pseudotime analysis. And essentially, you identify how related cells are in a graph-based representation based on their transcriptomic similarity. And we can look at the evolution of gene expression along pseudotime across the genome. And so the combination with time-resolved sampling and unbiased phenotype identification, an extensive literature-based annotation, and pseudotime calculation allowed us to construct an updated multi-state model for the diverse fates of infected cells in the earliest stages. And what's satisfying is that if you look at clusters 2 and 5, those are the 2 most dominant states that we see present within LCLs. So that's kind of the trajectory towards successful immortalization. As I mentioned, we collected multi-omic data. So now I'd like to tell you a little bit about the joint analysis of that gene expression data, which I just showed you with single-cell chromatin accessibility match for the same cells. So of course, we can look at chromatin accessibility now aided by our phenotype model in the same colors. And when we look by cluster, we see reduced chromatin accessibility in our unsuccessful infection trajectories. We see elevated chromatin accessibility to an extent in the successful infection trajectories. I would point you to cluster 6, which is the hyperproliferative phase, which has extensive increases in accessibility that match the extensive upregulation in total unique mRNA transcripts in this phenotype. So with RNA and attack, we can now start to look at what I like to call DAP-linked DEGs, which are differentially accessible peaks between states that are linked to differential gene expression between those states. And the first thing that struck us was that about 20% of all of the differentially expressed genes that we identified in our early infection time course had at least one DAP associated with them. That is one differentially accessible regulatory loci associated with that gene. I'm not going to go into any of these fate trajectories in particularly extensive detail, but I would just point out that this multimodal resource with cluster level resolution allows us to explore and pose very specific questions as to what the regulatory differences are as a function of virus-induced fate. So this one here is virus-induced cellular arrest with an example gene shown, and you can see at particular loci the correlation of RNA expression by phenotype with changes in accessibility at that locus. Likewise, we can also look at the difference between a successfully transformed latency 3-like phenotype relative to resting cells. In this case, we're highlighting a negative regulatory locus where increase of accessibility is significantly correlated with loss of gene expression. And we can even examine the regulatory differences that exist within the states that are found simultaneously within successfully immortalized cells. In this case, the light zone-like a latency 3 state and the post-germinal center differentiated cell state. Of course, this is an EBV meeting, and we aligned our data against a concatenated reference, and so we were actually able to collect time-resolved chromatin accessibility differences within the EBV episome. I won't go into this data too much, but I will just point out that we're not just collecting quote-unquote garbage reads here. We're actually seeing real biologically significant sites here, including the C promoter, some reads in the Q promoter, the LMP1 transcription start site, and the BZLF1 TSS. Of course, we can look at episome accessibility by cluster, not just by time, and see which phenotypes have greater accessibility at different loci across the viral genome. The spoiler is that within 8 days, the 2 states that have the greatest accessibility in terms of number of unique sites are the hyperproliferative dark zone-like state and the NF-kappa B activated light zone-like state. The next thing we wanted to do was to try to integrate a wealth of data that the research community has established into this analysis. And one of the richest resources that we wanted to integrate was an extensive array of chip data sets from labs like Michelle West and Bo Zhao and Ben Gevirtz that have profiled EBNA binding genome-wide. Now, those have been bulk experiments, and they've been in the lymphoblastoid cell line GM12878, but we essentially reasoned that LCLs mark, they are a proxy for the quote-unquote successful infection trajectory. And so it might be possible to reference our single cell data against data that was collected for EBNA transcription factors in GM12878 to see what state-resolved differences in accessible EBNA binding loci might look like. That distills into one technique that we call CRISP attack, which is chip-referenced inference from single-cell phenotype attack data. And essentially, the workflow looks like this. So you as a user can take the RNA and attack data and specifically look at the attack cluster data that we've identified, and also collect any transcription factor data sets of interest from any number of ChIP-seq experiments, whether they be viral transcription factors or co-activators or host. You can then prepare all of those data as called peaks, so as genomic range objects, and then perform one large multimodal intersection across single cell cluster attack and bulk ChIP-seq data to create one intersection matrix. And then the user can specify particular recipes or combinations of logical gating criteria to identify peaks of interest. So for example, if you wanted to find all genome-wide peaks that were present in one cluster but not another, that overlapped a binding site for one EBNA but not another. And then you can take those loci that match all of those gating criteria and do cis-regulatory prediction to identify genes that are predicted to be regulated by those differentially accessible transcription factor or co-activator associated loci. One example of this in action. So we looked at the light zone NF-kappa B activated state and asked what was different in that state than the innate sensing mediated arrest. And so we identified about 1,900 differentially accessible peaks between these two states. Those 1,900 DAPs had about 1,500 unique linked genes based on cis-regulatory prediction. Among those 1,500 genes were 71 of 167 genes that have been characterized to be linked to known EBV superenhancer loci. An example of one of those is shown here. This is TRAF1. And you can see that there are 3 loci highlighted that are EBNA3 or EBNA2 binding sites based on ChIP data that have significant associations to the level of gene expression that's resolved by single cell clustering. And what's nice is that if we look at the representation of EBV superenhancer linked genes from this predictive recipe, it's significantly enriched relative to a size match random sample of the genome. In a second example, I'll just highlight this kind of trimodal integration idea. So here we're looking at the difference between a light zone latency 3 NF-kappa B activated state and a post-germinal center differentiated memory B cell state. And we're specifically looking for peaks that are present in the light zone, not in the post-germinal center, that overlap with at least one of the 3 EBNAs shown here, LP, 2, or 3C. We identify the linked genes based on that analysis, and then we can cross-reference them with our empirical measurements from the single-cell RNA assay that is cell-matched. So that's sort of a depiction of what the process is that we're doing here. We've got direct measurement linking RNA and chromatin accessibility. We are making an inference based on ensemble ChIP-seq data set, and then we are validating the predictions from those inferences that are TF and cluster resolved back to our empirical measurement. And among the genes we see some that are upregulated in black, some that are downregulated in red, and EBV superenhancer linked genes are shown in bold. And we can resolve by transcription factor for sites that are accessible in cluster 2, inaccessible in cluster 5, what those gene expression profiles look like. In the last bit of time I have, I'm going to
wrap up now.
I will, yeah, thank you. In the last bit of time, I'm going to share just one of the stranger things we've seen in our experiment, and that's that we identified this non-canonical or atypical memory B cell subset. These cells are described in the literature as being expressing the TBET and CD11C, which are usually expressed in T cells and dendritic cells. They're pathogenically expanded in a wide range of chronic infection contexts and in autoimmune diseases. What's interesting is we found them at day 0, and then after 8 days of infection, we still found a population matching this phenotype. The strangest thing that happens with them is they show upregulation of pro-inflammatory factors, but they also show upregulation of significantly enriched gene ontologies involved in neural cell adhesion, neuronal signaling, and nervous system development. And so we lack a current mechanistic understanding for this, but it appears that EBV de novo infection within this particular B-cell niche can impart some sort of neural tropism. As a sanity check, they express viral reads. They cluster distinctly from the light zone expression pattern, though, as you see here, based on their distance from LMP1 and the host proxy ICAM1. So with that, I'd like to thank you. I've summarized a time-resolved single-cell multiomics resource that provides a genome-wide roadmap for studying fate-specific viral trajectories. We've developed an informatic method to integrate that with existing ChIP-seq resources. We've leveraged single-cell resolution to identify infection dynamics within a novel subset. And in the future, we hope to do mechanistic studies to investigate the role of EBV-specific regulatory loci, to extend our studies to clinical lymphoma samples, and to further explore the properties and responses of infected atypical memory B cells. Thanks for letting me run over. Thanks to everyone in the lab. Thanks to all the cores. Thanks to the funding. Thank you again.
I think we only have time for one question. Do we have one quick question? Hand up here.
Just a quick question about whether or not you've looked at any pathway analysis for the gene expressions that you have, and if they overlap between cells.
Correct. Great question. Yes. We have this in a bioarchive preprint and in a forthcoming paper. There is significant gene ontology analysis there, geoprocess specifically, and you do see distinctive, I mean, of course, most of them are all within the umbrella of immune responses and antiviral and specific lymphocyte signaling pathways, but you do see distinctions between cluster versus cluster analyses. Yeah.
One more question, maybe?
I think we better move on, because if you could catch him in the break, we've run significantly over and we have a short coffee break at the end of this session. So thank you very much, Elliot. I think we'll move on.
So our next speaker is Naomi Naji, who will be speaking about from Maria Masucci's lab, who will be talking about her work on BPLF1.
Hello, everybody, and I would also like to thank the organizers for allowing me to present the results of the Masucci group, which, of course, deals with the BPLF1, the major tegument protein of EBV, sorry. So BPLF1 is expressed in the early phases of virus replication. It helps to assemble the virus, is delivered to the newly infected cell, and there it helps for the virus to enter the nucleus. It shares a common conserved domain with the other members of the major tegument proteins of the herpes viruses. It is produced as a large protein with a very high molecular weight marker over 300, and this is processed through caspase-1 dependent cut, and an N-terminal shorter fragment is released, which has the catalytic activity. And this catalytic activity is D-ubiquitinase and D-nedylase, as it has been shown previously in the lab. Maria's lab has been working continuously on this protein, and it has shown involvement in different functions in the cell, like autophagy or innate immune responses were influenced by BPLF1, and all these functions were, of course, mediated by the D-ubiquitinase or D-nedylase function. So in this present work, we turn our attention to the DNA topoisomerases. First of all, because they have been already shown to be involved in the DNA replication of the different herpes viruses, as shown here. For examples are EBV, herpes simplex virus 1, or Kaposi sarcoma associated virus. And based on these results, inhibitors of topoisomerases have been suggested to be used as antivirus. In all these experiments, the inhibitors of topoisomerase have been added to the cells before or at the time of inducing the lytic cycle. And then it was when it had an inhibition of the lytic infection. But we wanted to see what happens if we add these inhibitors when the cells already express BPLF1, when they already progress through the lytic cycle to a certain extent. So for this, we used a cell line, HEC293 cells, which express the wild-type BPLF1 or the catalytic mutant BPLF1 under the control of the TET-regulated promoter. So when we add doxycycline, BPLF1 will be expressed, and this can be detected. So these cells were treated with etoposide, which is a TOP2 inhibitor poison. And then we detected if we see any response as DNA damage responses. So what we could see that when the cells expressed BPLF1, seen in red, then, and they were treated with etoposide, gamma H2X as this first sign of DNA damage was downregulated in those cells where BPLF1 is expressed. But if the BPLF1 is catalytically mutant, then expression was not influenced. Also, the other protein of the pathway, what we tested was 53BP1, where we also see a BPLF1 catalytic activity dependent response after etoposide treatment. Maybe in this figure it's quite difficult to see, but the dots of the P53 of the 53BP1 are not formed as obviously in the cells that express the wild-type BPLF1. Here we see the normalization of the data, which shows that BPLF1 indeed has interaction and an effect on the etoposide treatment. Here down we have a control where we use neocarcinostatin, which is a radiomimetic, which induces double-stranded breaks, just to see that it is not the BPLF1 expression which is doing the inhibition, because in these cells treated with this drug, the gamma H2X was equally expressed whether or not the BPLF1 was catalytically active. Topoisomerase 2 toxin sustains DNA replication and transcription by binding to the, and this is the reason, the goal is to resolve topological problems when the DNA is translated or transcribed, replicating. So the torsion of the DNA should be eliminated. TOP2 binds to the DNA, cuts, induces a double strand breaks. When the DNA unwinds, then it can be reassembled, religated the DNA. When etoposide or other endogenous damage comes to these complexes where the DNA is cut, TOP2 is bound, then it will be trapped there, and it cannot be resolved. So the cells try to solve this to be able to survive by adding ubiquitin or moieties to it, which leads to proteasomal degradation, leaving the damage with a short peptide on the end, which is cut then by the TPD2 enzyme. In this moment, now the DNA damage can be sensed and the cell try to repair. But if it's too extended, then genomic instability and apoptosis will occur. So we wanted to see whether BPLF1 has any effect on etoposide-induced degradation. We treated the same inducible cells with etoposide and, and checked TOP2 beta and TOP2 alpha expression. If the BPLF1 is wild type, we can see that without BPLF1 etoposide treatment, we see downregulation of TOP2 alpha because the cells try to get rid of TOP2. proteasomal degradation occurs. This can be inhibited by the proteasomal inhibitor, but then if BPLF1 is expressed and it is wild type, then the degradation does not occur. Sorry again. And with the wild type, with the catalytic mutant of BPLF1, degradation occurs when it's not expressed, and a similar level of degradation occurs when BPLF1 is expressed. So BPLF1 prevents the degradation of TOP2 beta. This TOP2 beta is ubiquitinated, as we can see here, after an IP with TOP2 alpha or TOP2 beta, and blotting with ubiquitin, we see a high molecular weight TOP2 beta and some modifications on it, which is decreased when the wild type BPLF1 is expressed in both TOP2 alpha and TOP2 beta. TOP2 BPLF was also, we wanted to see whether this TOP2 which is trapped on the DNA can be stabilized by BPLF1. For this reason, we isolated the cleavage complexes which contains of the TOP2 and the DNA by extracting DNA in the conditions where the covalently bound proteins are isolated, and then loading 10 microgram of DNA, we detect the TOP2 expression in these products. We can see that if we treat the cells with etoposide for half an hour, these complexes are formed. If we leave them longer, for 4 hours, the complexes start to disappear due to the proteasomal degradation. If BPLF1 is expressed, the complex is formed, but after 4 hours, they are still there. They are not degraded. Here we have quantification of 3 experiments. So the BPLF1 is in stabilizing the cleavage complexes. We wanted to see what kind of modifications these cleavage complexes have. So we detected ubiquitin and SUMO in the etoposide-treated cells with or without BPLF1. When BPLF1 is present, ubiquitin levels obviously are decreased. But to our surprise, SUMO levels were remaining the same level, or actually it's not a surprise, it doesn't have the anti-SUMO activity, but it helped to understand what is going on with these cells. So the ratio of ubiquitin to SUMO in these etoposide-treated cells is shifting towards the SUMO levels. We repeated the effect on the cell viability by treating the cells which were exposed to doxycycline to express BPLF1, and then to increasing amount of etoposide was added to them, and they were left for overnight culture. And after that, we recorded by MTTSA the viability of the cells. And as we can see here, the viability was higher when the wild-type BPLF1 was present in the cells, and the catalytic mutant could not rescue the cells. So the next question was, what happens if physiological levels of BPLF1 are expressed in the cells? So for this, we used LCLs, which were established with the wild-type EBV, or EBV derived from the 293 cells expressing the catalytic mutant virus. LCS were also further manipulated to help us induce them to lytic cycle, so we introduced a TET-regulated BCLF1 to them. So when we had doxycycline, cells entered the lytic cycle. We could see that when lytic cycle is induced, TOP1 alpha is downregulated in the wild-type BPLF1 expressing cells, which is in line with the fact that in the lytic cycle, there is a pseudo S phase appears. TOP2 beta levels were maintained the same. The ubiquitination of the total cell lysate was not visible influenced in the total lysate. We don't see influence of the ubiquitination. Maybe this is or most likely it is because of the lower levels of BPLF1 expressed in these cells. But if we immunoprecipitate TOP2 beta and then look for ubiquitin in those complexes, then we see that the ubiquitination is decreased if the BPLF1 is wild type. So indeed, BPLF1 regulates the TOP2 activity in LCS, induced lytic cycle. The cleavage complexes are stabilized also in these cells when the BPLF1 is wild type, as you can see in this figure. Sorry. Why? The viability of this cells-induced lytic cycle is better if BPLF1 is wild type, as proven here by the gamma H2AX levels, which are always lower than when the cells carry the catalytic mutant BPLF1. This was also reflected or paralleled by the virus production, both in cells or released in the supernatant. So we wanted to see whether cells in this stage, they are in lytic cycle, they have BPLF1. BPLF1 is stabilizing the TOP2 on the cleavage complexes. What happens if we add etoposide to these cells? So we added increasing amount of etoposide. We left them overnight and detected viability after 16 hours. And again, we see that viability was increased, or it was better. So cells were rescued if BPLF1 is expressed in the cells. There is another way of rescuing. There is another enzyme, as I show you, that is involved in the rescuing from the debulking these complexes from the DNA, is TDP2. In order to check whether this is also involved in the BPLF1 effect, we used an LCL already established, which has the wild type and the knockout TDP2 enzyme. Again, we induced them with the tetracycline-inducible BZLF1 and tested their survival upon etoposide treatment. And again, we see that if TDP2 is there, the survival is better and virus production is better. So TDP2 is also involved in the BPLF1 mediated rescue. So our model, yes, I'm at the last slide. So our model is that when BPLF1 is there, cells are in productive cycle. BPLF1 stabilizes the TOP2 on the DNA and the broken DNA, but ubiquitination is decreased and the sumoylation probably makes a sterical change in the conformation, which allows TDP2 to directly cut the protein and DNA binding. And then since TDP2 is not degraded, it's still there, TDP2 itself can repair the DNA with an error-free repair. So in this way, survival and virus production can be maintained, even if etoposide is added. So as a consequence of our results, we could think about when tumors expressed BPLF1, if that would give them an advantage if they proliferate faster, because they can handle better this breaking and reassignment of the DNA. And this should be also possibly considered if etoposide treatment would be used in tumor treatment, and to select which tumors could have a benefit of this treatment. And sorry. And thank you for the Maria Masucci group colleagues and long-term collaborators and founding agencies. Over.
Okay. In the interest of having a coffee break, can we just take one quick question and hold the rest for the break? Okay, over 0. All right, thank you. Our next speaker is Sheldon Stein of the De La Cluse lab, who will be talking about oncogenic properties of the Epstein-Barr large integument protein, BPLF1.
Good afternoon, everyone. It's a great privilege for me to be presenting our group's work. I'd like to thank the organizing committee for inviting me here. And this is another paper, presentation on BPLF1. So I'd like to start with this. So there have been several identified to lead to the induction of the lytic cycle of EBV. And this is commonly found in foods that we eat and in tobacco products. And this upregulation of lytic activation has been positively correlated with the development of NPCs and gastrocostinomas. And in our lab, we've had a lot of interest in the studying of early events of EBV infection. And in the study, we found that exposure of B cells to M81 virus-like particles that are devoid of the EBV genome, we found that there is an increase in aneuploid mitoses when compared to our controls that we use, which is the CD40 ligand IL-4 treated B cells. And one of the main contributing factors to this was the tegument protein BNRF1. And they found that the centrioles were found to be over-amplified when BNRF1 was overexpressed in like 293 cells, and they were able to demonstrate this would lead to mitotic abnormalities. And recently, BNRF1 has been found to lead to the SMC56 to be degraded, which leads to impaired chromosomal segregation. Another example, which is the one I'm studying, is the BPLF1, which has been linked to the degradation of colons, which leads to the accumulation of DNA lysosome factors such as CDT1. And this leads to the increase in DNA re-replication. This is allowed to permit an S-phase environment, which is favorable for EBV genome replication. So in my study, I looked at a panel of tegument proteins and found, including a full-length BPLF1 expression vector, which was flag tagged. And when compared to the panel of tegument proteins that we looked at, the BPLF1 protein showed the highest amount of irregularities, nuclear atypia, within these cells that were transiently expressing the proteins. And this example that I show here is the multinuclearity, the tendency for the cells to develop multinuclearity. And so what we did next was perform some live cell imaging. So we developed a reporter plasmid that expressed the full-length BPLF1 alongside a fluorescent protein, and we fact sorted the cells and visualized them under a microscope, which showed that which showed different mitotic phenotypes and mitotic outcomes. And we found that this video that shows here, in particular, one of the results that we found that was very interesting was the development of micronuclei was a lot higher in the BPLF1 expressing cells. You can see, yeah. And I'll go into that a bit later. It'll be relevant to the. Okay, so we wanted to see what exactly was happening, what interactors of BPLF1 were leading to this this formation of these micronuclei and these nuclear irregularities. So we did a co-IP coupled to mass spec analysis, and we found through 3 independent studies that the top interactor was SEMP6, which is a SUMO23 deconjugase. And we verified it through reciprocal co-IPs, where we pulled down the flag tag BPLF1, and then endogenous SEM6 co-precipitated with it. And through the reverse pull down, we reverse pull down the endogenous SEM6, and we found that the flag tag BPLF1 co-precipitated. And we also looked at the catalytic mutant to see if that was able to pull down the SEM6. And yes, indeed, it did do that. And so we wanted to see if, how did BPLF1 affect SEM6 activity in the cells? And we looked at, since it's a sumo 2,3 deconjugase, and previous experiments have shown that there is an uncontrolled polysumulation in cells that have depleted SEM6. We found that in cells that are expressing BPLF1, and as well as the BPLF1 catalytic mutant, we found an accumulation of sumo foci within the nucleus of these cells. And when you look at the Western blots, we also found that there is an increase in these polysumulated species in these gels. And when we look at the B cells that are infected with M81 wild type, and we compare it to M81 BPLF1 knockout, and then we found that there is an increase in the sumo body formation as early as 1 hour post-infection, which is quite remarkable, which shows that this is an event that occurs very early in infection. And it's a phenotype that is recovered when complemented with the BPLF1 expression. And it's also something that we do find a noticeable increase in at 1 hour with just the VLPs. And this effect increases over time and peaks at 72 hours post-infection. And then we look at, in previous studies, they assert that SEM6 is a modulator of genomic stability. And in their experiments, when they silenced SEM6, they found that there is a formation of micronuclei in these cells. And another thing that they take notes of is that there is a reduction of the SEMP-A protein at the centromeres. And this demonstrates a disruption of the constitutive centromeric associated network. And these proteins are all targets of SEM6 and they are all targeted for either delocalization or degradation when SEM6 is depleted. And this is the model that demonstrates this. So the centromeric complex is maintained by the Mis18BP1, and through uncontrolled simulation of Mis18BP1, it leads to the degradation, and also uncontrolled simulation of the SEM proteins leads to the delocalization from the centromeric region of the host genome. And the BPLF1, so we looked at the B cells, and we looked at the centromeric protein A, and we found that as early as 24 hours post-infection, we found that there is a reduction in the foci of the centromeric protein A. And this is shown at 72 hours post-infection. And it's an effect that we see for the M81 and the VLPs. And it's an effect that's not apparent in the knockout and the control. And so in conclusion, we found that BPLF1 induces a suit of nuclear atypia. We identify SEM6 as a novel BPLF1 interactor. And the BPLF1 results in unrestricted simulation in host cells, which induces phenotypes that are associated with SEM6 depletion. And one of these phenotypes is the SEMP-A delocalization from the centromeres upon introduction of BPLF1. So I'd like to thank the following people who worked with me on this project, and I'd like to thank you all for your attention. So.
Thank you. So we for a couple of questions.
An obvious question is, what is the mechanism of action? Is it related to its DUB function?
Sorry, I can't hear you.
An obvious question is, what is the mechanism? And is it related to its DUB function, BPLF1?
BPLF1. And what function?
Deubiquitinase function.
Well, we demonstrate that this effect occurs with the catalytic mutant of BPLF1. And I haven't included it in this presentation, but there are domains that are independent of this that we have found that induce this effect. So the effect, we have several systems. We like to use the endogenous SEM6 first of all, but we haven't been able to demonstrate a degradation of SEM6 with the endogenous system. But through an artificial system where we express an isoform of SEM6, we found that there is a degradation of SEM6. We also, through immunofluorescence studies, we see there is an increase in the cytoplasmic presence of the SEM6. So there could be a delocalization of the SEM6 that could be happening. Or another alternative explanation, which is also being investigated with some interns that we have, they hypothesize that the catalytic region of SEM6 is being bound by BPLF1, which could be interrupting its activity. So there's many different things.
We'll make this the last question.
Thanks. That was really beautiful. Have you, so the BPLF1 overexpression with the H2B live cell microscopy was really cool, and it looked like you were initiating an abscission checkpoint and preventing the cells from dividing.
Yeah.
And I was wondering if, in the context of the knockout virus in the 293 producer cells, if you've ever done live cell microscopy with the H2B and you see cells dividing.
No, we haven't looked at.
Do you think they would?
That'll be quite interesting actually, with the producer cells, because we were just doing transient expression. Yeah, we haven't done that.
Do you think it could be that BPLF1 is
yeah.
keeping cells from dividing?
What I've done is I've looked at the abnormalities within the producer cells, and I do find a lot of these irregularities. If you look at producers, GP350 positive producer cells, wild type M81, they are just absolutely, they've exploded in size, and there's just all sorts of junk everywhere. And I'd like to see that. Yeah, that's a good question. Yeah, but no, I haven't seen it. I do think there will. I do suspect there will be something to see. Yeah. Okay.
Okay, and our last speaker of this session will be Francisca Manera, who will be talking about key epigenetic events during EBV-mediated cell transformation.
Hi, everyone, and thank you for the kind introduction, and also to the organizer for giving me the opportunity to present my ongoing PhD project. I would like to briefly start by recalling the principle according to identities defined by precise gene expression programs that depends on specific patterns of DNA methylation, histone modification, and overall chromatin organization that in a sort of epigenetic landscape define trajectories that are leading to specific cell fates. And however, the direction of a cell along these branching pathways can be modified upon exposure to specific environmental stressors, for example, upon EBV infection. And this can lead to molecular deviation in the cell identity maintenance pathways and increase epigenetically the susceptibility of the cells to malignant transformation and cancer development. And as we all know, EBV has the ability to hijack the host B cell maturation pathways to establish a lifelong persistent infection in the membrane B cell compartment. And to do so, the virus exploits the human epigenetic machinery in order to regulate its own transcriptional programs according to the requirement of a latent infection. And this may impact on the host epigenome itself as well. And there are already several evidences showing that this host B cell epigenome rewiring during EBV infection may be a mechanism of EBV-induced cell transformation. And there are already several evidences of EBV manipulation of different epigenetic regulators of the host. And in our previous works, we as well identified a common mechanism, epigenetic mechanism, through which EBV is promoting tumor suppressor genes transcriptional silencing, which involves the manipulation of the DNMT1 enzyme and promoter hypermethylation. But the main aspects of the host B cell epigenome rewiring still need to be dissected. And this is the main goal of this ongoing project, which aims to explore the broad spectrum of epigenetic events occurring during cell transformation. And the main objectives are the identification of epigenetic drivers and to explore in parallel the progressive chromatin landscape remodeling and the impact on the expression of cancer-related genes that may justify the progressive acquisition of a malignant cell phenotype. In order to identify epigenetic regulator genes that are essential for the EBV-mediated cell immortalization, we developed this large-scale screen loss of function, CRISPR loss of function screening, which involves the use of a recombinant EBV expressing Cas9 that will allow us to select the infected cells, the Cas9 positive cells as the only ones surviving in culture upon infection. And the idea is to transduce the infected cells with a lentiviral library, which includes guide RNA targeting 426 epigenetic regulators, a library that was previously developed in our lab. And those epigenetic regulators that are essential will be identified through a negative selection, and we will proceed by validating some of the interesting gene candidates. In parallel, to reveal some of these ERGs, epigenetic regulator genes that are transcriptionally altered during the infection, we analyzed publicly available RNA-seq data, and among the significantly deregulated genes, we screened for those belonging to this compendium of 426 epigenetic regulators. And as you can see from the heat map, most of those genes are transcriptionally altered, and we found 32 genes that are significantly deregulated, and 36% belong to the class of histone methyltransferases. And interestingly, we found 3 genes belonging to the family of the histone methyltransferases KMT2, respectively KMT2A, C, and D. And KMT2D is known to contain often putative driving mutation in the context of EBV-negative lymphomas, according to TCGA. Therefore, we hypothesize that EBV-mediated deregulation of expression of KMT2D may have a driver role in the context of EBV-mediated lymphomagenesis. So we investigated the mechanisms of KMT's deregulation, and just to be short, I will present only results of KMT2D, even though we found similar results for the other KMTs. So we confirmed that the KMTs are downregulated both in immortalized cells and in transformed cell lines, and this appears to be independent to the process of B-cell activation itself, while is a consequence of the infection, meaning of expression of viral genes, and notably we found LMP1 as the main driver of the KMT's downregulation through the NFKB pathway activation. We also found that this transcriptional downregulation is not a consequence of promoter hypermethylation, as the treatment with the demethylating agent ADZA didn't generate an evident rescue in the expression of the KMTs. And some in silico predictions suggested the involvement of the EBF1 transcription factor, as it was conferred by an overexpression experiment in which there is a rescue in the expression of the KMTs. And as I briefly mentioned, these are histone methyltransferases, and more specifically, they are involved in monomethylating glycine 4 on histone A3, mostly at enhancer regions, which is a histone modification that correlates with open chromatin and gene expression. And therefore, we are interested in exploring the impact on the chromatin. And we assessed the level of the histone mark H3K4 at different time points during the cell infection and cell immortalization in vitro. And we found that there is a decrease of the histone mark. And Therefore, now we are performing at a cut and tag sequencing to be able to map the genome-wide redistribution of the histone mark. And by performing integrative analysis with the RNA-seq data, and as well with ATAC sequencing data, we will be able to define the impact of the KMT's downregulation on cancer-related genes expression that may justify the progressive cell transformation upon EBV infection. So just in conclusion, we found that during EBV-mediated cell immortalization, there are several epigenetic regulators that are transcriptionally altered. And in particular, we identified a class of histone methyltransferases, KMT2, that are downregulated as a consequence of a reduced expression of EBF1, mediated by the LMP1-induced NFKB pathway activation. And by performing cut and tag RNA sequencing and ATAC-seq, we will be able to define, to better understand the impact of this downregulation on chromatin rearrangement and cancer-promoting genes expression. And as well, in conclusion, the CRISPR-Cas9 genome scale screening will allow us to identify other interesting epigenetic regulators that may be involved in cell transformation. So I would like to thank everyone that is contributing to this project, my supervisor, and especially Rosita, that she started with me this project, and that sadly she's not here anymore to see the progresses and the future developments. And I would like also to say a special thanks to Henry Gruffa from INS de Lyon, which is my co-supervisor, and to all of you for the attention.
We have time for a couple of questions.
Thank you for fantastic talk. Actually, decrease of K4 monomethylation in the Western blotting was really impressive.
Sorry, can you just maybe move the mask?
So the decrease of K4 monomethylation in Western blotting was really impressive. Then did you already analyze the targets of K4 monomethylation, like and the decrease of gene expression after?
So I don't hear so much. Guess you're asking if I analyzed for the. You're asking for the.
Oh yeah,
Yes,
this one,
yeah,
yeah,
so
K4
much.
methylation, yeah, was
Yeah.
decreased. And so just I'm wondering where K4 monomethylation, it's not the monometh. Yeah, monomethylation mark was lost in the genome. The targets of
Yes, no, for the targets we haven't identified yet because we want basically to do this cut and tag sequencing to see where is redistributed and which are the regions that are most affected by this progressive loss of histone monomethylation. Of course, as I said, we expect to find mostly enhancers, so we will then investigate those enhancers, which gene expression, I mean, they are involved in the regulation of which genes. And
okay, thank you.
Okay, I have a question actually. So you mentioned that you thought that the KMT2D downregulation was mediated by EBF1 downregulation. So what was the mechanism Is EBF1 a regulator of the transcription of KMT2D?
So basically it's already known that EBF1, the activity of EBF1 is modified by EBV, especially ABNA2. That is modifying the binding sites. So we are wondering if there could be another mechanism at another level of regulation, not post-transcriptionally, but also at the transcriptional level. And we just, for the moment, we only performed this overexpression experiment, and we found that there is a trend, as this was revealed, suggested as well by the RNA-seq data, as the curves are basically overlapping. And as well, we found that EBF1 has some binding sites on the promoter of KMT. So it would be nice, of course, to do maybe some chip as well to confirm. Yeah.
Thank you.
Thank you.
So now we'll go to a brief coffee break and be back for 4:30 for the next session.
There's a light when it goes on. I don't see it. Does the red light come on here if you push that? Yes. When that red light
Okay.
goes on, it's on.
Hello? Hello? You can close the door. What do you think? So, welcome to everybody to the last section of today. And first of all, I would like to thank Professor Leoncini and all the organizers of this very, very interesting meeting. I'm learning a lot of things concerning basic research on EBV. So it's a pleasure for me to co-chair this section with Professor Sattin, and now we are friends. Okay, and we start immediately with the first topical lecture on epithelial cell tumors given by Hironori Yoshiyama. He's working since many years, 30 years, more than 25 years on EBV, and he's from Japan. So, please.
Okay. Thank you for organizing committees to providing me a nice opportunity to give a talk. So today I'm going to introduce several talks about epithelial cell tumors. So first of all,
Sorry, could you speak close
close.
to the microphone?
Okay. Yes.
I can hear you very well.
EBV makes tumor for epithelial and lymphocytes, and several latency patterns depending on the tumor cells, latency 1, latency 2, latency 3. And today I'm going to focus on gastric cancer and nasopharyngeal cancer, and one other oral cancer. And so first topic is gastric cancer. And gastric cancer, as you know, is the fourth cancer incidence, and fourth mortality and fifth incidence. And it is divided to characterize four types of subgroups. Chromosomal unstable pattern, this is 50% of occupancy, and genomically stable pattern is 20%, and microsatellite unstable pattern is 20%. And EBV-associated gastric cancer, this also occupies 10% of gastric cancers. And its characteristic is PIK3CA mutation and PD-L1 overexpression, and EBV associated chromosomal methylation, and CDK2A silencing, and immune cell signaling, the characteristic of EBV-associated gastric cancers. And this is our case, and EBV-associated gastric cancer looks like this as endoscopic observations. And this hematoxylin eosin staining, it looks like this. This is the cancer cells, and this is epithelial morphologies. And these cancer cells are stained by EBVs. And 100% of gastric cancer tumors have EBV positives. And those are lymphocytes. infiltrating lymphocytes is characteristic of EBV-associated gastric cancer. And this many inflammatory cells is characteristic. And all those researchers found EBV-associated gastric cancer tumor cells arises from a single cell infected with EBV. If they examine the terminal repeats, number of terminal repeats, they are, depending on the tumors, they show the distinct numbers of terminal repeats. That means single infected cells became tumor in the end. And the distribution of this EBV-associated gastric cancer is this is 10% in Camargo et al. But this distribution is not really reflecting the real. This data is pretty old, and so still we won't know which percent. Yes, we know in Japan and East Asia, they are around 10% also. But other part, Europe and USA, we are not sure. But a certain number is African gastric cancer is EBV positive. And this is the first experience of my EBV-associated gastric cancer cases. And in this gastric cancer cases, the tumor is here. And this is the pyloric pyloric region. And this is a normal gastric mucosa. And we found EBV-associated gastric tumor is located the atrophic border of this is so not normal mucosa. And atrophic border, in the atrophic border, we found EBV-associated gastric cancer. And these are tumor cells. And tumor cells are stained by EBVs. And atrophic border means this gastric epithelium is duodenal type. So you can see the goblet cell is very bright, and it is so-called intestinal metaplasia and caused by H. pylori, characteristic to H. pylori infection. And very close to this intestinal metaplasia, we found tumors, and they are EBV positive. So in this case, it reminds us are there any correlation between EBV-associated cancer and bacterial infection? And if we look at the underneath submucosal region of this atrophic membrane, we can see many CD20 positive B cells. And among these B cells, EBV positive B cells also exist. So bacterial stimulation causes B cell proliferation, and among these B cells, EBV positive cell exists. And probably we are assuming that these B cells transmit EBV to epithelium. So, and coming from these cases, we thought, are there any correlation between H. pylori infection and EBV? So this is a cause of atrophic gastritis. H. pylori infection causes inflammation and reduces normal gastric epithelium and cause mucosal atrophy. And if we look at the endoscopic observation of these gastric mucosa, normal mucosa is like this, and atrophic gastritis reduces the height of the epithelium and become red, and it really proceeds to the atrophy, membrane looks like very red, and we can see the blood vessels. So, and these time course proceeding of the gastritis. So the inflammation causes this chronic gastritis. So in the submucosal region, B cells and T cells, lymphocytes. And atrophic region, there's no inflammation, less inflammation. But however, because of these inflammations, we are assuming epithelial cells have damages, get damages. So we examine EBV infection status according to the atrophic gastritis proceeds, and so from mild to moderate and severe. And dividing this classification, we took gastric biopsy specimens and examined take DNA and do PCR. And the target of primer is BAM-H1W. And we check the good linearity and examine the copy numbers of atrophic membrane, EBV copies to the epitheliums. And surprisingly, moderate inflammatory regions, there's many EBV copies. And severe atrophy, less EBV copies. So looks like the EBV is more, it's supposed to be transmitted to the inflammation cause moderate gastritis. So from this data, we made a in vitro infection system. And gastric epithelial AGS was exposed to H. pylori, and the 100%, MOI is 100. And after 7 hours exposure of H. pylori, antibiotics was added, and also GFP EBV, recombinant of EBV, thymidine kinase gene is changed to GFP. It was added, and after 48, MOI was 125%, 125. And after 48 hours, we examined the GFP positive EBV infected cells. So with H. pylori exposed or no H. pylori, there's a difference. With H. pylori treatment, we saw many EBV infected cells. And those are bacterial dose dependent. If bacterial MOI was 1 and 100 increased, EBV positive cells increased. And so those H. pylori exposure induces interferon beta to epitheliums. So what bacterial factor induces this enhancement of EBV infection? So bacterial virulence factors, BAC-A, CAG-A, or CAG-PAI, or flagella was examined. And CAG-A locates here, and CAG pathogenicity island is on this region, or this is bacterial genome. And pathogenicity islands includes type 4 segregation systems. And we made several knockouts of BAC-A and CAG-A and flagella knockouts. And wild type H. pylori induces bacterial EBV infection, and but CAG-A knockout and BAC-A knockout no change. And also, but CAG-PAI knockout reduces to the control level of EBV infection. And Fla-A knockout reduces somehow. So we thought bacterial attachment may be important because flagella is no bacterial moving. So we centrifuge bacteria and epithelial cell mixture and 300 G for 5 minutes and examine the bacteria attached to the epitheliums. So wild type pylori attached to the epithelial surface, and also Fly knockout H. pylori, also after centrifugation attached to the epithelium. And if we centrifuge bacterium, the bacteria, even Fly knockout, EBV infection increases. So why we saw two ways, viral replication or viral attachment or end entry. And to examine, we use BCL1 knockout EBV. And even BCL1 knockout EBV, bacterial exposure increased EBV transmission. And we also counted, washed out the surface of the bacterial exposure and EBV infection, and surface-bound virus membrane fraction was taken and did PCR and counted the EBV copy number. EBV was attached to the cell surface membrane. So we found probably receptors. These AGS cells are CD21 negative, so co-receptor may be upregulated. And the control sodium potassium ATPase is similar, but EPHA2 and NMHC2A was increased after bacterial exposure. And this increase of co-receptors was downregulated by SI, and also EBV infection was reduced by SI treatment. So there are several papers. EBV induces H. pylori mediated, H. pylori CAG mediated gastric cancer formation, because the H. pylori induces methylase, and methylase reduces SHP1 transcription, and SHP2 is an oncoprotein. And this story was published by Saggiator by Nature Microbiology. And also bacterial enhancement of EBV is published by us and also R. Robinson's groups. And so next story is we did primary nasopharyngeal epithelial culture, and they're using the same recombinant EBV system. And this primary nasopharyngeal epithelial was infected with EBV. And if we culture this primary culture with air-liquid interface culture, and this air-liquid interface culture makes pseudo-stratified epitheliums. And some epithelium makes cilia, and we can observe cilia bleeding. This differentiation system and EBV was infected. And so this is the result. EBV positive cell, this is RNA hybridization. EBV positive cells are more in suprabasal area than compared to the basal area. And the BRLF1 is also. And this is beta tubulin, so this is cilia is stained. So differentiated cells, more EBV replication. And also we examined cilia beat frequency does not change by EBV infection. However, EBV infection disrupts the cell-cell communication. Transepithelial electrical resistance was reduced by after EBV infection. So adhesion of cell was reduced by EBV infection. And the third story is oral cancer. Is some of the oral cancer is related to EBV. This is an old story by Twizoff et al. We know hairy leukoplakia is EBV positive. So there is a report oral cancer is many in Asia region, and EBV infection and oral squamous cell carcinoma is related to EBV and accumulated the 13 reports of oral this EBV infection and OSCC stories. Epidemiologically, EBV is related to OSCC, and odds ratio is 5.03. And from this analysis, we did some experimental experiment. And the former reports say the EVA positive and LME positive or OSCC exists, but as we look here, the staining is not good. So, but however, the Ati et al. Did nice EVA staining, and tumor cell was stained. Epithelial tumor cell was stained EVAs, and this lymphocyte stained by also EVA positive. So coming to this idea, we tried to infect in vitro infection system using skin squamous cell carcinoma and tongue squamous cell carcinoma. And two cells from skin HSC1 and tongue HCC25 cells become EBV positive after G418 selection. And they showed, this is a famous NPC model, the HSC1 was positive for LMP1, and very weak LMP2 also positive. HCC25 was positive for EBNA1, but no LMP1. And so we, how many EBV was infected to the cell, we examined the copy numbers of infected, very few, 2 or 3, and less than 10 each. And however, those HSC1 was positive for BGLF1 stained by immunostaining. CC25 was negative for BGLF1. And also, lytic protein was positive. But these cells proliferate faster than EBV negative cells. And E-cadherin was reduced, and ZO1 was reduced, but bimentin became positive, and several ZEB1 transcription factor was positive. So, and wound healing assay says EBV positive epithelial cells are more migrative and invasive. So, starting from my first story, EBV positive bucket lymphoma cells, if we make limiting dilation, some cells become EBV negative, EBV strip off, and those EBV negative bucket lymphoma cells can be infected with EBV again. And we don't have a good epithelial system, but some epithelial cells can acceptable EBV infection and become EBV positive. And they show the reflectable, this is the EBV associated gastric cancer model, they are type 1 infection. So I would show gratitude to my collaborators, Siena University, Hokkaido University, Yamaguchi University, and thank you very much for my talk, listening my talk. Thank you.
Thank you for your presentation. There is a time for a few questions, please.
So, your SCC and HSC cell lines.
Give it the microphone, please.
The two oral cell carcinoma cell lines that you infected with EBV in vitro, and you showed variable numbers of EBV genome. Do you keep those cell lines constantly under selection? I mean, what happens if you don't keep the selection? Do you lose EBV genomes? Because that's what is the problem.
Yes,
for example,
yes,
with NPCs cell lines.
It sustains EBV copies for a while, but we are adding G418 continuously and keep EBVs. But several passages, we don't lose EBV.
Is there any other questions? Please.
Thank you for a fantastic talk. Can I ask you about the combination effect of Helicobacter pylori and the EB virus? So how much does the Helicobacter pylori infection contributes to EBV infection? So usually the stomach needs pylori infection for EBV infection.
In Japanese case, most of the gastric cancer have H. pylori infection. So 95% of gastric cancer is H. pylori positive. So even though EBV is 10% of the total gastric cancer, so H. pylori infection is always doing something. That's what my thinking.
Thank you.
So thanks a lot.
Thank you very much.
And the next speaker is Shannon Kenney from University of Wisconsin, Madison. LMP1 promotes proliferation and inhibits differentiation in EBV-infected telomerase immortalized normal oral keratinocytes by activating the EPO pathway effectors, YAP and TAZ. Please.
Thanks for inviting me. The first author is Dio Singh, but he was unable to be here because of a visa problem. Today I'll be telling you how LMP1 reduces proliferation and inhibits differentiation of normal oral keratinocytes in the context of the intact viral genome. So we all know that EBV does something to promote undifferentiated nasopharyngeal carcinoma. It's essentially always there, but it's really hard to define exactly what it does. Because for one thing, EBV isn't transforming to normal primary epithelial cells by itself. And another thing is once NPC tumors are fully formed, if you put them in tissue culture, they no longer require EBV to get out, making it very difficult to study. So most studies have been performed using overexpression, massive overexpression of LMP1 or LMP2A in carcinoma lines that are already fully transformed and can't differentiate. So today, I'm going to tell you how we've been studying LMP1 in the context of the intact viral genome at normal levels in cells that can differentiate. So to do this, a few years ago, my lab infected normal oral keratinocytes. They're telomerase immortalized. We got them from the Carl Munger lab. Initially, we used a CADA virus obtained from Burkitt lymphoma cells that was made by Lindsay Huffledger, has the G418 resistance gene, and GFP inserted into the non-essential TK gene. Using this cell line, we can get stably infected NOx cells, and importantly, it's one of the few normal epithelial cells that retains EBV, and more importantly, it can still differentiate to some extent. So when we raft these cells, we can see that over on the left, that all the cells are EBV infected. They're all EBV positive. But if we look to see where lytic reactivation occurs, it only occurs in the differentiated cells. We've done a series of experiments, examine exactly why is that the case, and we'll talk about that tomorrow. But what I'm going to talk about today is that latent EBV infection inhibits differentiation of these NOx cells, and that's what we're going to be studying, who's responsible for that. So a few years ago, Mark Eichelberg and Eric Johansson's lab, in collaboration with us, showed that when he rafted the uninfected NOx cells, they expressed much more of the differentiation marker K10 compared to the EBV-infected cells. Furthermore, you can see, looking at MCM7, it's a proliferation marker. The EBV-positive cells have proliferation on top of the suprabasal layer, which is abnormal. This is a wonderful model, very physiologic, but very hard to do biochemical assays on. So fortunately, a couple of years ago, my lab discovered we could actually look at differentiation in monolayer culture over on the right. We discovered if we grow the EBV negative NOx cells without any growth factors, mainly you have to remove the EGF, and we plate them at subconfluent density, they spontaneously differentiate. So these are all differentiation markers, except for P63, which goes away with differentiation. And first, we showed that the type 1 Akata infection is extremely good at inhibiting this differentiation. And subsequently, we showed that a type 2 virus that we had inserted GFP and G418 into the exact same place as Akata can also do this. So this allowed us now to make mutants and do things like siRNA and find out which EBV genes are required for this effect. So we parked the viruses into HeLa cells where we were able to perform CRISPR-Cas9 mutagenesis. And Scott Nelson in my lab, in collaboration with Eric Johansson's lab, made an LMP1 mutant. It has essentially 12 amino acids before it goes out of frame, and then it ends at 25 amino acids. We put the mutant virus or wild-type virus into cord blood cells on a feeder layer just to show you that absolutely this virus makes no LMP1. Then we used the wild-type or mutant virus to infect at the exact same day, exact same time, the NOx cells. And one of the first things we asked is, if we remove all the EGF, can we finally see an ability of EBV to induce proliferation in semi-normal epithelial cells? And the answer is yes. So for the first time we saw, yes, wild-type EBV induces proliferation when we remove EGF. And guess what? LMP1 mutant cannot do this. So it is required for the effect. We also looked at the ability of the wild-type versus LMP1 mutant cells to inhibit spontaneous differentiation. And again, here you can see the uninfected cells differentiate. They stopped expressing P63. Wild-type EBV prevents this. The LMP1 mutant virus is unable to inhibit differentiation. It's also sufficient by itself to inhibit differentiation in uninfected NOx cells when expressed at biologically relevant levels. So we expressed LMP1 in a supposedly hematopoietic specific lentivirus that allowed us to express LMP1 at very low levels, much less than P-BABE, for example, that would just kill the cells. And the level is very similar to what we see with wild-type infection. I want to point out that although our mutant studies are done with type 2 EBV, this is a type 1 B958 LMP1 we're overexpressing here. And you can see that it is sufficient to inhibit spontaneous differentiation all by itself. So LMP1 is required and sufficient to inhibit differentiation of the NOx cells. So next we asked, does this effect require alteration of YAP and TAZ activity? Those are the downstream effectors of the hyposignaling pathway. And the reason we asked this is because the E7 protein in HPV was recently shown to activate YAP, and this is required for the ability of E7 to inhibit primary keratinocyte differentiation. In addition, another group showed that overexpressed LMP1 in the carcinoma line could increase TAZ activity, although they did not look at YAP. This is the hyposignaling pathway. So what happens is last kinases are induced that induce an inhibitory serine phosphorylation on YAP and TAZ that keeps them in the cytoplasm. If this phosphorylation is removed, YAP and TAZ can go to the nucleus, interact with DNA binding proteins called TEADs, and induce things like proliferation genes and genes that inhibit epithelial differentiation. Another major way to activate YAP is to induce Sarc family kinases and directly phosphorylate YAP on a tyrosine residue, allowing it to bypass the hyposignaling pathway. So when we looked at this inhibitory serine phosphorylation, we could see that this phosphorylation was definitely decreased by wild-type EBV, and this required LMP1. And to a lesser extent, we also see inhibition of YAP hypophosphorylation, and it's at least partially mediated by LMP1. Looking at the Sarc kinase activation, we could see that the wild-type cells have increased YAP tyrosine phosphorylation that requires LMP1, and we can see total Sarc kinase activity is increased by wild-type EBV, but not LMP1 mutant. A few years ago, the Ning lab showed that in B cells, LMP1 induces Sarc kinase activity by directly interacting with it. So it seems that it can also do this in epithelial cells. So the next question is activation in YAP and TAZ involved in the differentiation and proliferation effects of LMP1? And the answer is yes. So to look at this, we used siRNA, where we knock down either YAP alone or TAZ alone or both together. And here, looking at the proliferation marker PCNA, we can see when we get rid of both YAP and TAZ, EBV doesn't induce proliferation anymore. And remarkably, just getting rid of one or the other of YAP or TAZ restores differentiation, particularly when they're increased together, knocked down together. We can also see this when we look at the effects of LMP1 alone. So next we asked, if we were to inhibit YAP or TAZ activity, would we reverse the ability of LMP1 to inhibit differentiation or promote proliferation? And we took advantage of a drug, an off-target effect on ibrutinib, which is a first-generation BTK inhibitor, which was recently shown to be able to activate YAP puts it in the nucleus. The mechanisms weren't exactly clear, but it does it at clinically relevant doses. It's not seen with second-generation BTK inhibitors, and NOx cells don't express BTK. This is a fortuitous off-target effect, but it occurs at clinically relevant doses. So first we asked, if we added this drug to the wild-type EBV cells, would we increase Hippo-mediated phosphorylation? Remember that inhibits function, and we definitely increase hypophosphorylation of TAZ and hypophosphorylation of YAP. These are kind of high doses, though. So we asked, if we do a dose titration, and we think these two lower doses could be achieved in patients, and remember, you may not know this, CLL patients get this drug orally every day for the rest of their life. And here we see that even though we're increasing LMP1 with the BTK inhibitor, even the lower doses decrease proliferation and remarkably increase differentiation in these EBV-infected epithelial cells. They also decrease EMT induced by EBV, which we have shown is due to LMP1, requires LMP1, and YAP and TAZ are known to induce EMT in epithelial cells. Finally, we asked, well, what would happen in an actual NPC line? So we collaborated with KW Low and George Chow in Hong Kong, and they sent us the most authentic NPC cell line they have, NPC 43. It retains EBV because it's never been allowed to differentiate in culture. The thing is about this cell line is it stopped expressing LMP1. So we asked, might it still be dependent on YAP and TAZ? Could it have found another way to activate YAP and TAZ? And if we look at the amount of Hippo signaling, it's also turned off in the NPC 43. So is the TAZ Hippo signaling phosphorylation, and the amount of SARC-mediated tyrosine phosphorylation is at least as good. So it suggests that even if LMP1 isn't being expressed, this cell line still has active YAP and TAZ. So we asked, what will happen if we treat it with the BTK inhibitor? And we did remove other growth factors. And sure enough, its proliferation is reduced, and we can induce differentiation of the NPC line with this drug. So, in conclusion, when expressed at normal levels in the context of the intact virus, LMP1 does inhibit differentiation. It does induce proliferation of normal oral keratinocytes. We think this is likely to be important for how EBV contributes to NPC development. We suspect that the first step is that LMP1 increases SARC activity, something it's already known to do. SARC is already known to activate YAP by increasing tyrosine phosphorylation, allowing it to go to the nucleus, and by indirectly inhibiting Hippo signaling. And so we think that inhibition of YAP and TAZ may be a useful treatment for some NPCs. KW Low is collaboratively helping us to look at the amount of nuclear YAP and TAZ in NPC tumors, and it's finding TAZ in particular is often in the nucleus of NPCs. So these are the people who did the work. These are all the people, particularly Dio Singh and Scott, who did the work I showed you in NOx cells. Eric Johansson and Paul Lambert and George Chow and KW Low are very important collaborators. So I'm going to stop and take questions.
Thank you for this very interesting presentation. It's open to questions.
So thank you very much. It's an interesting talk. So do you have an idea by which proximal signaling events LMP1 induces SARC? So
I think I didn't show you, if we use drugs like dasinimab that inhibit SARC, it does something very similar to BTK inhibitor, but it's actually more toxic. And others have shown that LMP1 can interact directly with SARC in B cells and increase its activity. So I imagine it's doing something similar like that in NOx cells.
Please.
Shannon, here. Really interesting talk, thank you. I'm just looking for a clarification. So in this context, this is akin to a reactivation model, might you say?
I'm sorry, what?
Would you say that this is similar to a reactivation model in the epithelial background? And, or is it closer to an infection model?
Oh, I think this is important for latently infected cells that express LMP1. And I think this is important in how latent infection causes NPC. Now, does it have anything to do with our previously described ability that LMP1 also promotes lytic reactivation?
Right.
Well, it might, because we've shown that yapin has at some level required for lytic reactivation. And so we don't understand the paradox that the different things that yapin can do and LMP1 can do, but we suspect that when LMP1 is, that high levels of yapin inhibit lytic reactivation by inhibiting differentiation, and lower levels yet may be paradoxically required. We're looking into that.
Please.
Great talk, Shannon. Have you, I don't know if you showed it and I missed it, did you try to grow these cells, LMP1 transfected cells, NOx cells in mice, NSG or NODSCADE or anything? In vivo experiments?
Did I try to grow them what?
Did you inoculate them in mice and see if they grow as tumors?
Oh, you know, we have not been able to get EBV-infected NOx cells to form tumors in mice, and it's clear that what we need to do is combine it with some other oncogene or loss of some tumor suppressor that also occurs in NPC to see if we can get synergy.
Okay.
Okay, thank you.
So the next speaker, Salvatore Tornambè from University of Siena. So you are at home. EBV persistence in gastric cancer cases conventionally classified as either ISH negative.
Hello to everyone. Thank you for introducing me. Can you hear me over there? I think so, okay. So here is the plan of my first. I will do a brief introduction on EBV-associated gastric cancer and the hit-and-run hypothesis. And the main part of the talk is about the aim and methods used for our study. To date, EBV-associated gastric cancer represents about 9–10% of all gastric cancer worldwide. And among them, the percentage of association with EBV infection is ranging from 5 to 10 for adenocarcinoma cases, and from 22.5 up to 100 for gastric cancer with lymphoid stroma. In our lab, we previously published a paper where we actually found EBV traces in a large cohort of Hodgkin and non-Hodgkin lymphoma cases, as well as cell lines classified previously as EBV negative. Those data were in accordance with the hit-and-run hypothesis. This theory suggests that the viral oncogenes may be necessary as a cancer formation, to start the cancer formation, the so-called hit, and then for the non-imperfect replication of the EBV genome, it could be lost. During this process, the tumoral cells could acquire several kinds of new mutations, as well as epigenetic changes that could be inherited by daughter cells. Then the presence of the virus is not anymore needed for the tumor progression. Here we decide to investigate the presence of EBV traces in gastric cancer conventionally classified as EBV negative by applying higher sensitive technique, then to study the methylation pattern of genes known to be specifically methylated by EBV. The question that we asked ourselves was, what if EBV traces have specific biological and/or clinical feature? If so, it could have an impact on the decision-making for oncologists. We know that the gold standard technique for the detection of the virus is hybrid cytohybridization, as you can appreciate on the right side of the slide, for those cases diagnosed as EBV positive, and in the middle of the slide for those cases classified as EBV negative. For our study, we decided to select a cohort of 40 cases diagnosed in 2010 in order to obtain a longer follow-up. And among these, 7 were diagnosed as EBV positive and 33 as EBV negative. So we were wondering, what if EBV contributes more widely than currently acknowledged? First of all, we decided to apply to as a high-sensitive technique digital droplets PCR, targeting BAM-H1 and IMA1, known to be the most conserved region of EBV genome. The advantage of this technique is the generation of up to 20,000 droplets in one tube, and inside each droplet, a reaction of amplification could happen when compared with the real-time PCR, where one reaction of amplification could happen in one tube. Moreover, we decided to combine this technique with a enrichment method, laser microdissection, on each negative slide, in order to exclude even one small positive reactive lymphocyte that could affect the subsequent analysis. Actually, we found EBV traces by digital droplet PCR in 8 out of 26 adenocarcinoma cases, and in 6 out of 7 gastric cancer with lymphoid stroma. Those kind of data could be visualized in 2 different ways. On the right side of the slide as number of events, and on the left side of the slide as copies per microliter. Then we decided to apply another high-sensitive technique named RNA-scope. RNA-scope is an high-sensitive in situ hybridization, targeting the mRNA of IBNA1. The advantage of this technique is the particular design of the probe, called double Z. This probe consists of 2 parts that works in tandem, which means that both parts of the probe needs to recognize and bind the target for the subsequent reaction of amplification of the signal that could be then visualized by optical microscope as a typical single red dot. As you can appreciate on the right side of the slide, for adenocarcinoma case, classified, diagnosed as EBV positive, and on the left side of the slide as we found the RNA-scope signal in adenocarcinoma case classified as EBV negative. We found the RNA-scope positive signal in 4 out of 26 adenocarcinoma cases previously classified as EBV negative, and 3 out of 7 gastric cancer with lymphoid stroma. Moreover, we found RNA-scope signal in those cases where actually we found EBV traces by digital droplet PCR. Since the signal by RNA-scope could be visualized by epifluorescence microscope, we decided to combine this technique with immunofluorescence for cytokeratin in order to demonstrate the neoplastic nature of those cells, as you can appreciate here. We know that EBV is able to interact with the epigenetic machinery of those cells. So we decided to study the methylation pattern of genes known to be specifically methylated by EBV, EYA1 and TIMP2. What we found is that the methylation level of those cases previously diagnosed as EBV positive was quite similar to those cases classified as EBV negative, but where we found EBV traces by digital droplet PCR, when compared with those cases completely negative for the virus. We use this as indirect proof of the previous infection of EBV. Finally, we decided to study the impact of EBV with the overall survival. By applying Kaplan-Meier curves, we found, as expected, a significant better overall survival for cases classified previously as EBV positive when compared to cases completely negative. But actually, you can appreciate an intermediate overall survival for those cases where we found traces, EBV traces. In the end, we want also to study a correlation of EBV with the clinical pathological feature. By applying univariate cause survival analysis, we found that the viral state and the T stage significantly correlated with the overall survival, as we found by applying multivariate analysis. The take-home message of my talk is that the currently implemented methods may not be sufficient for detecting viral traces. Here I would like to share with you this paper provided by the Pan-Cancer Analysis of Whole Genome Consortium, where actually they studied the viral load of 2,600 cases among 38 types of cancer, and they found the more viral positive cases than previously reported. We also know that to test the hit-and-run hypothesis is difficult. Possible strategies may be to monitor the outcomes of national vaccination campaign against viruses in general, and to develop gene editing tools in vivo to delete viral oncogenes and evaluate the progression of tumor growth. Because if this theory is proven to be true, it will have implication in early detection, prophylaxis, and therapy of many cancer types. Thank you for your attention. I would like to thank all of my group of research, and a special thank to my colleague and first author with me, Maria Chiara Siciliano. Thank you. I went a bit faster.
Thanks for this interesting update presentation for discussion, for question and discussion.
Thanks so much for the wonderful presentation. I don't know if I missed it, but the genes that you saw that are methylated by EBV, the EYA1 and TIP2, did you also check if they are differentially expressed, or how the expression is impacted by EBV?
Sorry, I didn't get the last part of the sentence.
Did you check the expression? Because you saw that the methylation is impacted by EBV, but
No, we didn't study the expression level for those genes. We just focused our attention on the methylation.
But methylation also affects how the genes are expressed. So
that's actually true, but we didn't do that because we wanted to use it as an indirect proof on those genes. So this is why actually is an interesting question, but we didn't was out of our scope for this in this case. Thank you.
Okay.
Question?
Of course.
Please.
With your RNA-scope data, it's one cell among many that you detect as positive. So how does that relate to the tumor as a whole? Because it's effectively a clone, but it's positive, and all the others around you, around that one clone, are negative. So what do you think has been happening to give rise to that RNA-scope positive cell?
We are using those higher sensitive technique for the detection. Okay, what we know is that the virus could be lost by the cells. I think to answer to your question, one explanation may be we actually did it for our previous paper. We had the opportunity to analyze the same cancer as a primary tumor, and after, because the patient went on relapse, we actually studied the immunoglobulin rearrangement, and we found for those cases the co-expression of the same immunoglobulin rearrangement from the first primary tumor and the relapse, and the colonization with the RNA-scope signal.
So the previous infection maintained.
Exactly. To demonstrate the monoclonal nature of those cells.
Please.
Thank you. That was a nice talk. Did you find that in the RNA?
Excuse me to interrupt you. Could you please turn on the microphone? Cannot hear you.
Did you see that in the RNA scope positive cases that there was amplification of PD-L1 or other changes in the immune microenvironment that you might normally see in Iperish positive cells?
Oh, sorry. Could you repeat, please? Because I cannot hear you.
Did you see in the microenvironment that there were any changes in, for example, PD-L1 to see if there was amplification?
We know that before I showed you this analysis, okay, where we found the viral state and T stage significantly correlated with the overall survival. Our hypothesis is that the presence of the virus in those cases, even the cases where we found traces, EBV traces, the virus could have start change in the tumor microenvironment, able to avoid the expansion of the tumor growth. Thank you for the question.
Another question, please.
Thank you for a fantastic talk. In EBV negative gastric cancer, sometimes the surrounding background mucosa may contain EBV positive lymphocytes, right? So how did you exclude that kind of possibility?
In the case of digital droplet PCR, we decided to combine it with the laser microdissection.
Right. But even if you microdissect it carefully, but the epithelial cell may be just attached to lymphocyte, and that kind of lymphocyte may be contaminated.
Excuse me for stopping you. I don't know if you can hear the question, but I cannot hear you. I'm sorry. Could you repeat it? I think we have problem with the microphone.
Okay, sorry. Yes, probably you microdissected carefully, but sometimes epithelial cell or gastric cancer cells may be really exist next to lymphocyte. Then if the lymphocyte may be infected with the EB virus, so just I was wondering that kind of possibility.
In this case, to answer your question,
Sorry, how would you exclude the lymphocytes and just
because
you used the marker, no?
In this case, yes, of course we did it, of course.
So can you have the microphone?
Microphone, yes, of course, a microphone.
The contamination.
All lymphocyte just in the under the sections, in the thick of the section. That is quite unusual, but anyway, you cannot exclude. But when you do RNA-scope analysis, then you can really see that these are epithelial cells because we double stain with cytokeratin.
Cytokeratin, exactly.
So the last question, please.
I have two questions. First, you've achieved a technical achievement that's very impressive by finding these signals. And so I'm worried that you don't have any controls that tell us that this is really pertinent and relevant to the question you want to ask, which is extending the EBV relevance in these tumors. What kind of controls are you using to show us that this association is meaningful? And the second question has to do with the model, but I'll come back.
Okay, to answer first about your first question, we decided to use, now I didn't report the controls that we used for our experiments, but for digital PCR, we decided to use cell leukemia cells that have never been reported to be associated with the EBV.
So I'm talking about not controls, technical controls, but disease-associated controls that you would use to try to establish that the new associations you have are pertinent to extending the EBV relevance for these tumors?
I didn't get the question. Which kind of control? No, I would like to understand, if you don't mind.
Okay, we'll talk at the.
Okay, thank you.
Okay. So, thanks a lot.
Thank you.
So the next speaker?
She's arriving.
So Chair Leahy will talk to us about exploring gene and protein expression in the tumor and tumor microenvironment of NPC.
The microphone? You can, okay, perfect.
That better? Yeah. Thank you very much for that introduction, and thanks very much for inviting me here today to present my work. I can't read. Okay. So my name is Ciara Leahy, and I'm a PhD student under the supervision of Professor Paul Murray at the University of Limerick in Ireland. And today I'm going to present a project which is ongoing in our lab, which looks at exploring the gene and protein expression in the tumor and tumor microenvironment of nasopharyngeal carcinoma at a single cell level. So there are a lot of single cell RNA-seq papers in the last few years which have started to unravel what we know is a very complex and heterogeneous tumor microenvironment of EBV positive NPC. So these papers have shown things like that T cells are less activated, more exhausted, and overall less effective in the TME of these tumors, and that there is increased infiltration of immune cells like macrophages, and that the microenvironment is quite immunosuppressive due to the infiltration of things like Tregs. But because, and of course that's all very helpful and helps us to identify what kind of cell types are there, but the single cell RNA-seq papers lack crucial spatial information which would give us the relationship of these immune cells to each other and also to the tumor. Furthermore, we know that EBV protein expression is quite heterogeneous among EBV positive NPC tumors. For example, LMP1 expression varies a lot. So this is two EBR positive NPC cores which we have stained for LMP1. On the left, you can see florid LMP1 staining in yellow, and on the right, you can see very little, if any, LMP1 staining. The literature has reported that the tumors here on the left, so high LMP1 NPC, is more aggressive and has a worse prognosis. So we thought it was worth investigating whether this clinical consequence could be even partially explained by the tumor microenvironment that differs between these two types of tumor. So for the purpose of this presentation, we decided to ask what impact does the degree of LMP1 expression have on the immune cell infiltrate in the tumor and stroma of NPC. So to do this, we're looking at both viral and cellular gene protein expression using the phenocycler fusion system from Akoya Biosciences, which is based on codex or co-detection by index chemistry. So I'll briefly explain how this works for people who aren't familiar with it. We have a panel of antibodies, and each antibody is conjugated to a unique DNA barcode. So for example, if we have CD4, it's conjugated to barcode 1, CD8 conjugated to barcode 2, CD20 to barcode 3. Each barcode then has a unique and complementary reporter, which is attached to a fluorophore. And when these are exposed to each other, it fluoresces just kind of the same way as a fluorescent secondary antibody would fluoresce. So what you do is you stain all of your antibodies at the same time and you fix them to the tissue. Then we add in cycles of 3 reporters which target 3 proteins with spectrally different reporters. We image that, and then we wash with DMSO, which dehybridizes the DNA bond between the barcode and the reporter. And then we wash away the reporters. That brings us back to the start, and that allows us to continue to add reporters in cycles of 3 until all of our antibodies are imaged. So for this presentation, we had 10 NPC samples, which we later divided into LMP1 high and low expression, where there was 5 of each. The patients had tissue biopsies, which we made into a tissue microarray. We used a microtome to put the tissues onto slides, and then we used the phenocycler fusion system to stain and image the tissue in a 42-plex panel. So obviously don't have time to go through all 42 markers, but just to show there was 5 kind of core modules modules. The first module was kind of lineage markers. We had structural markers, immune module, immune activation module, and then for the EBV module, the protein which is most pertinent to this presentation is LMP1. So we know that EBV antibodies can be difficult to work with in the first place, and we have the added complication of that in Codex, once you conjugate your antibody to a barcode, it can lose some of its specificity. So we wanted to make sure that the LMP1 antibody that we were using, which is the clone CS1-4, still retained its specificity after conjugation. So on the top, we just have a typical chromogenic stain of LMP1. And then on the bottom, we have some of our Codex validations. So IM tonsil, we saw a nice membranous staining, which had a similar pattern of expression and localization to the chromogenic stain. Again, in MPC, we saw a nice membranous staining, which overlapped with the tumor. So we were confident that the antibody was retaining its specificity after conjugation. So the best thing about these kind of experiments is that we get these really lovely images. We can look at the tumor in red, LMP1 expression in yellow, and CD68 in green. Again, we can look at T cell subsets using 3, 4, and 8, or CD20 to look at B cells, but the issue with this is we can only ever turn on 5 to 7 markers at a time, and when you have a panel of 42 markers, you want to be able to look at them all simultaneously. So this data is only really useful if we can start to quantify it and analyze it efficiently. So to do that, we need to do some image pre-processing, and our segmentation can be a major obstacle for this type of technology. So our lab has settled on using a membrane-based method of segmentation. So we combine multiple lineage markers, which gives us a pseudomembrane, and then we run that in a tool called DeepCell. It's probably worth noting that we did try to optimize a membrane marker stain, but we haven't found one yet, which we've optimized enough to be able to segment efficiently. So once DeepCell is run, we then get a segmentation mask, and that allows us to extract the morphological, spatial, and expression features of each cell, and we save that in an FCS file. So it's essentially the same output as the flow cytometry data, but you have all of the spatial location of each cell as well. And then we import that FCS file into Missile, which is an OR library, which my colleague, Dr. Ina Fennell, has written to help us to analyze this high-dimensional data. So using unsupervised clustering, the cells divide into distinct populations. To be able to identify what those populations are, we look at the gene expression profiles of each cluster, and by comparing those to known cell types that are very well studied, we can assign then cell types to each of the clusters. And we were happy enough that we saw kind of the populations which we expected, like T cells, LMP1 positive tumor cells, lots of macrophages. And so this gives us then the phenotypic structure of the tumor. But because we have the spatial information of every cell, we're able to put those cells back into the tumor. So we can do that for the tumor cells, the M1 macrophages, the CD4 T cells. So this is the digital analysis. And then to validate this, we can turn on markers in our actual staining to see if markers that should mimic what we get here. So we can turn on pan-CK for the tumor and see that they look quite similar, CD68 for M1 macrophages, and CD4 for the T cells, just to validate what we've done. And this is just the same for CD8 T cells, M2 macrophages, and smooth muscle. And again, just making sure that we're happy with the segmentation and the cell type annotation. So again, we can just select the populations of interest and put them back on to get a really nice map of where our cells are in the tumor. So at this point, we're happy that we've segmented the cells, we have identified the cell types, but if I want to answer the question which I'd originally posed, which is comparing the immune cell infiltration, LMP1 high and low tumors, it doesn't really make sense to compare a full core to another full core because the architecture of each core is different. The amount of stroma, the amount of tumor nest. So we want to unbiasedly dictate the architecture of the cells. So for example, dividing them into specific compartments. So to do that, we do this thing we call cellular neighborhoods, where we take a cell and we look at it. We take every cell, but as an example, we'll take an epithelial cell here and we look at its 10 closest neighbors. And because they're all epithelial cells, the system will realize that it's probably in the tumor area. If you see a T cell and most of the cells around it are T cells, it'll dictate it as a T cell enriched region, and the same for macrophages. So that means that we get a map of the tissue architecture But instead of, now we have a tumor region, but that's not all just epithelial cells anymore. That also has all of the infiltrating lymphocytes or anything else which is in the boundary of that tumor nest. And again, for validation purposes, we can just turn on the markers which we, in the actual staining, which we know should kind of reflect it, just to validate that we're happy with that the neighborhoods are where they should be, and that we can use them for further analysis. So first of all, we looked at the cell frequencies in the immune stroma, and it's worth noting that for the immune stroma, what we decided was immune stroma was all the cells minus the tumor cells, but also minus some fibrotic bands, which we saw in some of the cores. So it looked like in LMP1 high NPC, there were higher amounts, or more abundant, there was a higher abundance of CD4 T cells and CD8 T cells, and the same for M2 macrophages and tumor-associated macrophages. But for M1 macrophages and B cells, there were these were more abundant in the LMP1 low, and this could be suggesting that there is a more tumor-promoting environment in the LMP1 high cases. When we looked at the tumor itself then, if you look at the scale on the axes, there didn't really seem to be much of a difference between either of the two. So this kind of suggested to us that while the literature reports that there are many more lymphocytes in LMP1 positive NPC, there may be a mechanism which prevents these lymphocytes from actually entering the tumor itself. They're just present in the stroma, and we don't know if they're as effective out there. So it's something that we really want to follow up on. So to conclude, for today, we've successfully stained for EBV antibodies using high-plex multi-immunohistochemistry. We can unbiasedly characterize both the cell types in the tumor and also the architecture of that tissue. In the immune stroma, we saw that most of the lymphocytes were more abundant in LMP1 high cases, apart from M1 macrophages and B cells. And in the tumor, there was no real substantial difference between those cell abundances of the immune cells. And that led us to believe that the immune cells may be excluded by some mechanism from infiltrating into the tumor in LMP1 high samples. So this was kind of an exploratory cohort. So we have lots of things that we want to follow up on. So to do that, we've just started a project with Resolve Biosciences, which is essentially RNA-ish, but for up to 100 markers. So we've designed a panel with 5 core modules looking at EBV gene expression, lineage markers, immune checkpoints, and cytokines and chemokines of interest, because we also have some RNA-seq data to inform this panel. So there, I'd like to say thank you for listening. Thanks for inviting me here. Thanks to my group, my supervisor, and to the funding bodies and our collaborators, and I'll take any questions.
Questions?
Thank you for the very cool talk over here.
Thank you.
So I had a question about related to the tumor neighborhoods.
Okay.
And I'm wondering, because you have a mask for cell segmentation,
Mhm.
and I'm wondering if it's possible to find centroids from those merged cells and then calculate a cell type median distance or distance distribution, because you can retroactively apply the identities of the cells to the mask.
Yeah, we actually have done that as well.
Okay, cool.
But we've just used the tumor neighborhoods in our recent analyses, just because we got really nice the tissue architecture from that. But we have also done the centroid method and the distance from the cells.
Great. And simple yes, no. Are there striking differences in cell type median distances across the cohorts?
Striking, no.
Okay. Thank you.
No hassle.
Hi, Kira. Thanks for a lovely talk. I know you said the project's ongoing, but I'm wondering if you've managed to stain for other EBV proteins apart from LMP1.
We have, but we've stained in IM tonsil, we've stained for EBNA1, EBNA2, LMP1. In NPC, we have stained for LMP1, EBNA1, and EBNA2, but it hasn't been as consistent across runs. But we have also have started optimizing some lytic cycle markers. It's definitely one of our goals, is to broaden the amount of EBV proteins that we can target on the codex. Thank you.
Can I ask one?
Yeah, of course.
So from what you said, NPC probably won't be very effectively treated by immunotherapy. Is that true?
Very preliminary data, maybe, but as I said, this is quite an exploratory cohort. We'd need to start looking at the immune checkpoint blockades in tumor itself and things like that. But we plan to do that.
Very, very interesting talk. Thank you.
I imagine these are fixed cells, right? Can you do it with frozen sections, and that might be amenable to, say, a multi-omic analysis or a single-cell multi-omic analysis of the nuclei?
So is this in relation to the multiplex immunohistochemistry?
Yes.
Yeah, you can.
You
So
can combine them.
Combine
the
RNA and the
multiplex
protein.
histochemistry with multiplex genomics or transcriptomics of
Yeah.
single cells.
Yeah, so the codex is definitely set up to work on FFPE and fresh frozen tissue. I don't like. Do you mean on the same? So when we
On the same slide.
on the same slide, once the slide is finished, its codex, you can't retrieve it afterwards. It's stuck in a flow cell, and once you take off the flow cell, the slide
see.
is likely to crack unless you wanted to try to do it
Okay,
beforehand.
so technically challenging.
Yeah.
And okay, yep. That's
Other questions?
Thank you very much.
So the talk will be given by Tushi Kaneda on Epstein-Barr virus infection rewires host chromatin structure and epigenetically contributes to tumorigenesis in a tissue-wide manner.
Thank you very much for this fantastic opportunity today to give a talk here in a fantastic symposium. My name is Atsushi Kaneda. I want to talk about epigenomic alteration in EBV positive gastric cancer. So we first conducted DNA methyl analysis in 2002 and again in 2011. Then we found a very unique and extensive DNA hypermethylation pattern in EBV positive gastric cancer. And in later, TCGA or other international group actually showed similar results. Actually, this is TCGA Nature report in 2014. So again, EBV positive gastric cancer showed a very unique hypermethylation phenotype. Then the question was, what was the cause of this hypermethylation pattern? Then we showed that the EBV infection itself was the cause to induce DNA methylation. So when EBV was infected in gastric epithelial cells in vitro, then a viral genome first methylated within 2 weeks. Then a host genome starts to be methylated, and it acquires extreme hypermethylation pattern in 4 weeks. And this is the distribution of DNA methylation. So in more than 7,000 genes, actually they are not methylation targets, but there are more than 3,000 methylation target genes in EBV infection. So it acquires complete DNA methylation in promoter CPG islands. And it also shows a loss of active marks, such as K4 trimethylation or K27 acetylation. And interestingly, there are a different group of more than 3,000 genes which acquire DNA methylation, but just in the surrounding region of promoter CPG islands. So in these cases, actually, they showed gain of active marks, such as K4 trimethylation and K27 acetylation. So these genes are not silenced, but when genes acquired complete methylation, actually, they were silenced. And this hypermethylation phenotype is not only in EBV positive gastric cancer, but also NPC, nasopharyngeal cancer. So actually, this is unpublished data, but when LMP1 was overexpressed, LMP1 seems to partially contribute to DNA methylation induction, and the methylation target genes include some tumor suppressor genes. So anyway, DNA hypermethylation is associated with EB virus infection. But today, I would like to rather focus on 3D chromatin dysregulation in EBV infection. In the first part, for gastric cancer. And in the second part, other malignancies like lymphoma and NPC. So first, we conducted Hi-C analysis to find out heterochromatin, B compartments, and the euchromatin in A compartment throughout the gastric epithelial cells and 14 gastric cancer cells. Then we found that here, B compartments in normal cells, so that is to say are inactivated in normal cells, but aberrantly activated in gastric cancer cells, or we found some A compartment active regions in normal cells, but aberrantly inactivated to show B compartments in some gastric cancer cell lines. We extracted these regions, then performed clustering, hierarchical clustering. Then we found, interestingly, that EBV positive gastric cancer showed some unique subcluster when using all the compartment shift regions, or even when using B to A regions only, or using A to B regions only. And the A to B regions are quite simple because this is inactive, aberrantly inactivated regions. So actually these regions are regions with a specific hypermethylation phenotype in EBV positive gastric cancer. So the question is, where are B to A aberrantly inactivated regions in gastric cancer? Before the answer, we conducted further analysis of high-C data by using interchromosomal reads to show interchromosomal interaction between different chromosomes or interactions with EB virus genome with human chromosomes. Then we found that the EB virus genome is interacted with some specific chromosomes like chromosome 2, 3, 4, 6, 7, or 13. And very interestingly, this pattern is not only in one EBV positive gastric cancer cell line, SCNU719, but they are common in other gastric cancer cell lines, or even in primary gastric cancer tissue. Then we validated these interactions by using 4C sequence technique by designing a bait primer in EBV genome. Then we confirmed that the EBV genome is actually interacted with some specific chromosomes, like chromosome 2, 3, 4, 6, 7, 13 again. And the 4C sequence also showed that the EBV interacting regions are common across cell lines or tissue samples. And more interestingly, these EBV IRs seems to be quite AT-rich or gene-poor regions. Then for further analysis, we used the in vitro EBV infection model to compare before and after EBV infection. Then again, we conducted high-C analysis and the 4C sequencing to detect EBV IRs. Then we confirmed that the EBV IRs are actually significantly AT-rich and gene-poor regions. And because these are the features of heterochromatin, we conducted chip sequencing for histone modification. Then we found that the EBV IRs are actually K9 trimethylation positive regions before EBV infection. So it's actually a heterochromatin, but it loses K9 trimethylation after EBV infection. Then it acquired K27 acetylation or K4 monomethylation. So this is the representative EBV IR. So it showed B to A conversion and the loss of K9 trimethylation and the gain of K27 acetylation. And let's look at the interaction between these EBV IRs and the surrounding regions. So actually this EBV IR used to be within a large heterochromatin. So it used to be interacted with the left part of heterochromatin. But after EBV infection, it loses interaction with the left part of heterochromatin. And can you see a small heterochromatin at the right side? EBV IR also loses the interaction with this heterochromatin. And you may also see the very small euchromatin at the neighboring regions of the EBV IR. So actually the EBV IR was not interacted with this euchromatin before EBV infection, but after EBV infection, it increases the aberrant interaction with the neighboring euchromatin. We confirmed this result by 4C sequencing again. So we designed the bait primer at the aberrantly activated enhancer regions in the heterochromatin. Then it showed the aberrant interaction within the neighboring euchromatin regions after EBV infection. And the genes in the neighboring euchromatin were actually upregulated by 2 or 5-fold. And when we knock down these genes, the cellular growth was significantly decreased. And instead of siRNA knockdown, we deleted enhancer regions by CRISPR technique. Then the gene expression level is decreased just 40 or 50%, but the cellular growth was significantly decreased. So this is a short summary for the gastric cancer part. So EBV binds to and rewires host heterochromatin, resulting in activation of enhancers and neighboring proto-oncogenes. We name this phenomena as enhancer infestation. And then we analyzed other malignancies like lymphoma and NPC. Then some EBV-IRs are actually commonly observed in gastric cancer as well as lymphoma or NPC, but other regions are quite tissue-specific. And the tissue-specific EBV-IRs were preferentially observed rather than common EBV-IRs. But how about epigenetic features in EBV-IRs? Then in bracketed lymphoma, again, B2A conversion was also observed. And if you may visit poster number 63, actually it was posted at the booth number 2. Dr. Okabe will present the data about bracketed lymphoma. So let's move to NPC. So we analyzed EBV positive NPC cell line or use in vitro EBV infection again. And we conducted high-C sequencing or 4C sequencing to detect EBV interacting regions in both cells. Then we got the 50 EBV-IRs in NPC positive, EBV positive NPC. Again, EBV-IRs are AT-rich, significantly gene poor, and canine trimethylation positive heterochromatin. Then we validated the result by using in vitro EBV infection again. Then we found EBV-IRs at quite similar regions with EBV positive NPC cell lines. And those regions are significantly AT-rich, gene poor, and canine trimethylation positive. And after EBV infection, these B compartments were actually converted to A compartment. And they are significantly enriched in EBV-IRs. And active histone marks are increased at enhancers like K4 monomethylation, K4 trimethylation, or K27 acetylation. And these regions include enhancer regions for some tumorigenic genes. So this is a short summary for the second part. So EBV-IRs are rather tissue-specific, but observed similarly in heterochromatin across cell types or tissue types. So EBV genome binding to and rewiring heterochromatin to activate silenced enhancers and the neighboring genes may be common in EBV-positive tumors. Thank you very much indeed. For the gastric cancer part or lymphoma part, Dr. Okabe conducted the study in the majority, and Dr. Mizokami analyzed NPC part. Thank you very much.
Questions? Paul? Yeah, great talk. So in some cases, you have the EBV genome inducing an enhancer, but you also have in the gastric cancers the induction of DNA methylation. So what's happening at the DNA methylation sites? Presumably the viral genomes are not going to those sites?
Right. Actually, DNA methylation is also important in gastric tumorigenesis. Actually, they silenced many tumor suppressor genes, and those regions show A to B conversion. So actually, by using A to B conversion, we analyzed hierarchical clustering. Then again, EBV positive gastric cancer showed some unique subcluster. So inactivated regions and activated regions are both very unique in EBV positive gastric cancer compared with other gastric
EBV
cancer.
genomes localize to the methylated regions?
Oh, right. Actually, hypomethylation was very rarely observed in the genome in EBV infection in gastric cancer. So basically, methylation pressure is really high, and hypermethylation is very easily observed in the genome widely.
Thank you.
Thank you again for the nice talk. I have a question regarding the similarities of the interacting regions. So you've shown that they are very similar. They're all AT-rich, they are gene-poor, they are heterochromatin. So my question is, have you looked further into how they are similar? Could there be, let's say, DNA motifs, for example, that are conserved in these regions that might explain why they are targeted by EBV?
Okay, thank you very much indeed. Actually, we analyzed, for example, motif analysis, but for example, activated regions in the heterochromatin, they include some specific motifs, but they are quite similar in the motifs enriched in genome-wide activated regions. So actually, we cannot find out any specific regions in those EBV-IRs. But interestingly, they are really commonly observed in specific cancer types. For example, very common in gastric cancer, and very common in NPC, very common in lymphoma.
Thank you.
I have a question. Hi. Yes. Very nice talk. About the EBV, the interaction between EBV, the human genome, I know that in your paper in gastric cancer, you find that OREP is the major regions that interact. I wonder if you had similar observation for other tumors, or maybe in different different tumors, you find other regions of EBV genome interacting with the human.
Yes, actually, we were interested in that part. And actually, we designed bait primer around the OREP in 4C sequencing. But for high C sequencing, we tried to analyze where in the EBV genome actually binds to human chromosomes. But the resolution was really low in high C technique. And you may remember today's presentation about high C data by extracting EBV genome only. Probably by those technique, resolution will become higher, like 5 kilobase or 10 kilobase or so. Yeah, by using such technique, probably we may find out where exactly the region in the EBV genome binds to or interacted with human chromosomes. Thank you very much.
Shannon.
Do you know if you need any specific EBV proteins in order to turn on these genes? For example, could EBNA1 be binding to the nearby EBV genome and activating, opening up chromatin? Do you know if you need any viral proteins?
Right. Actually, without EBNA1, actually, we don't have any idea. And we try to delete EBNA1 or EBNA1 binding to the between human chromosome and the episome by using decoy EBNA1. But after deletion of EBNA1, the altered epigenome are quite stable. So anyway, it's like a hit and run hypothesis, I think. So when epigenome is altered once, then after that, the epigenomic status was quite stable, even if EBNA1 is deleted. But I'm sorry, actually, I don't have exact answer how to activate by EBNA1 EBV protein.
One more question, the last.
Fascinating talk. So OREP seems to be in common between all the different types of cancers in EBV-IRs, but for the host genomes, anything in common between the different types, NPC, gastric cancer, and Burkitt lymphomas in the poster?
All right. Yeah. Actually, we don't. We have not found any specific features in host genome. Anyway, they are rich and gene poor, but just it's common features of heterochromatin. And actually, we are really interested in which heterochromatin is really preferentially detected and bound by EBV genome, but actually we don't have an exact answer yet. Thank you very much.
Thank you. So the next talk will be given by Joanna Wilson, and she's going to tell us about her mouse models of EBV-associated disease from understanding mechanism to preclinical model. Joanna.
Thank you for the opportunity to give this presentation. Oh, this is a bit like Zoom, isn't it? Except that I can see you all. Yes. If you want to keep to schedule, I've got 30 seconds to give my talk in.
You've got more. Don't worry.
Okay, so I'm going to talk to you today about some old data and some new data. And the aim is to showcase the resources, because what this is leading to is I really want to ask you, do you want any of these resources? And so I'm going to give you information, say old and new, and then ask you that question. And I'm showcasing two of our transgenic lines of mice, one that expresses EBNA1 in B cells. They develop lymphomas. We can take those primary tumor cells into culture and study them. We can inject those tumor cells back into fully immune competent syngeneic mice and watch tumor development in the recipients. I'm also going to tell you about the LMP1 series of mice that express LMP1 in the epidermis, and they're subject to chronic hyperplasia and chronic inflammation. And we can promote carcinoma formation in these mice, and again, take these carcinomas into tissue culture, and again, re-inject the tumor cells into fully immune competent mice. So to start off with the EBNA1 mice, this is EBNA1 from the B958 strain. It's not been modified in any way, but it's linked to the mouse immunoglobulin heavy chain enhancer to direct expression to B cells. These mice were made in the older traditional way of microinjection, where the transgene lands randomly in the genome. In this case, it's landed on chromosome 5 in a relatively gene-poor region. And the closest gene, which is not a known oncogene, is not deregulated in any way by the downstream insertion of the transgene. These mice all develop B-cell lymphoma. You can see it here developed in the spleen and in the liver. And while we can either speed up that lymphoma development by breeding these mice into a more susceptible strain, the BALB-C strain, or slow it down by crossbreeding them into a less susceptible strain of mice, the FEB strain, nevertheless, all of the mice develop B-cell lymphoma. And the C57 Black 6 strain is the strain of mice we typically use. So many years ago, we crossed these mice with mice that were transgenic for MYC, and nicely for a model of Burkitt lymphoma, there's a cooperation. So either NMYC or CMYC, that the bi-transgenic mice develop tumors faster than either of the single transgenic mice. Conversely, when we cross them with BCL2 overexpressing transgenic mice, there's a complete redundancy. There's no cooperation whatsoever. So when we published this, say, many moons ago, we suggested that this redundancy might suggest that EBNA1 performs a survival function in lymphomagenesis. And it's been really nice to see that in more recent years, some elegant publications that also indicate EBNA1 performing a survival function in lymphomagenesis. In these tumors, the tumors that develop in the mice, so this is a Western blot of tumor samples. They overexpress c-MYC, they overexpress E2F1. They also show higher expression of EBNA1 at the protein level. And I stress this is at the protein level. Here are 3 pre-tumor samples, and the RNA level of EBNA1 is the same, but in the tumor, you can now readily detect the protein. It's at very low level pre-tumor. We've looked at the change in expression of many genes, but one in particular I want to point out to you is that of MDM2, and all the tumors overexpress MDM2 and several isoforms of MDM2. We can use these mice as extending the model by taking the tumors from mice, and the tumors in the original mice will develop between 4 and 12 months of age, but we can take those tumors and then look at them in culture, do assays on them in culture, but also then re-inject them into mice so that the recipients will now develop the tumors in 4 to 8 weeks, and we can passage them indefinitely and also freeze down these tumor cells. And while each individual tumor from an original transgenic mice will have a different mutational profile, the thing they have in common is that they all express EBNA1. So using these primary tumor cells in culture, we inhibited them with, or we used an MDM2, series of MDM2 inhibitors, in fact, several inhibitors that inhibit MDM2 at different locations along the MDM2 protein. And all of the MDM2 inhibitors led to the death of these cells in culture compared to controls. And we can show that each of the inhibitors led to the loss of expression of MDM2. And in addition to that loss of expression of MDM2 compared to controls over here, we see a dramatic loss in E2F expression and also a slower loss of EBNA1 expression. And we've used a number of different inhibitors here, all used at equimolar levels, even though that's not necessarily their optimum dosage. And I want to point out one of these, which is the inhibitor called CAL101, which targets PI3K delta. And that also leads to the death of the cells in culture. And if you increase the dose, it leads to faster death. And the reason I point out that one is this is one that's approved in the clinic. And inhibiting with this PI3K delta, with this inhibitor, leads to the loss of MYC expression from the cells, the loss of E2F1 expression, again, compared to controls down here. So with MDM2 inhibition, I think we've shown there's an interplay between MYC and MDM2 and E2F1, and in the context of these EBNA1 tumors. And so I pointed out to you that MDM2 inhibition, as well as leading to loss of MDM2 expression, leads to loss of MYC and E2F1 expression, as well as EBNA1. And so does inhibition with a MYC inhibitor lead to loss of expression of these same proteins and the death of the cells. We've taken that system another step further to a quantifiable one where we've crossed the EBNA1 transgenic mice with red mice, so completely fluorescently red. They develop red tumors, and then we can take those tumor cells and transplant them into non-red mice so that we can then quantify the tumor development in the recipients. Just here is a sort of a red in vivo image of a red mouse versus a non-red mouse, and you can see that the splenocytes from these mice are very easily distinguishable by flow cytometry. Again, here's the non-red splenocytes from a non-red mouse, and the red splenocytes really quite easily distinguishable. And when we transplant them, as the tumor grows, we can see the increase in the red cells in the recipient spleen. So here at just over 2 weeks, there's 2.5% of the spleen is red, and at 3.5 weeks, it's now 22%. And so we can watch that over time as the tumor grows in the recipients. You get an increase in the fluorescent cells. And in this particular case, we've then treated the mice with those MDM2 inhibitors over 4 days and looked at the tumor burden after that treatment and see a reduction. These are just preliminary experiments, but see a reduction with that MDM2 inhibition. Conversely, when we do something very similar using the PI3K delta inhibitor CAL101, we actually don't see any reduction. Now, that might mean that we haven't got the dosing or the regimen right or optimal, but I would say that the MDM2 inhibitors are more promising. Moving on to our LMP1 mice, this is expressing, they express LMP1 of the cow strain, CAO strain, and expression is driven by the L2 promoter, which is actually the promoter that naturally lies downstream of LMP1 in the EBV genome. And this leads to expression in the epidermis, and particularly for peculiar unknown reasons, particularly the ears of the mice. And that leads to chronic inflammation and hyperplasia. And this mouse is shaved, it hasn't naturally dropped its hair out. But you can also then see that lesions begin to form. And so we've characterized this inflammation and the hyperplasia, and we just for convenience put it into 5 different categories of increasing hyperplasia and inflammation. And this can go on to lead to keratoacanthoma and in the older mice carcinoma. And this stage 4 ear is a section of the ear at the same magnification as a control ear, and you can see the extent of the hyperplasia, both of dermis and epidermis in that. And the inflammation is also characterized by the influx of immune cells, And this is showing that the immune cells are showing reactive oxygen species. And as the inflammation continues, we can see the induction of enzymes, for example, like INOS in these transgenic tissues compared to controls. Many moons ago, we did a proteomic analysis comparing the LMP1 expressing inflamed hyperplastic tissue compared to controls, and saw deregulation up and down of many, many proteins. And here's just one example. This is a member of the chitinase-like protein family increased in the transgenic tissues. We'd see that in the proteomic analysis. You can see it by in situ hybridization. You can see it by Western blotting. And actually, this has taken us off onto a sidetrack to explore these proteins. And with the chronic inflammation, you can use simple antioxidants such as here N-acetylcysteine, which feeds into the glutathione pathways, and treat the mice, in this case, simply adding it to the water. And you can show that that reduces the inflammation in the treated mice compared to the control mice. And we can quantify that inflammation. So here showing the inflammation, we can quantify it. This is with the increasing age of the mice, and these are N-acetylcysteine treated mice that have reduced, not disappeared, but reduced inflammation. Again, a long time ago, we showed in an older series of mice that when we treat the mice with chemical carcinogens, they have an increased burden of lesions, an increased number of papillomas and carcinomas compared to controls. And we've also shown that with our more recent series of mice, the LMP1 mice have an increased burden of lesions. We can take the carcinomas that form, grow them in culture, and several of those are then tumorigenic when they're re-injected into fully immune competent recipient mice. And we've got many of these cell lines that are LMP1 positive or negative, male, female, and with additional genetic modifications. So, for example, using these cell lines, we can explore LMP1 inhibition. So this is transfecting in a dominant negative LMP1, and we can show that the LMP1 positive, an LMP1 positive cell line in this case, was less able to form colonies in culture than the control, an LMP1 negative cell line, and the few colonies that did grow, they then grow more slowly than the controls, and the LMP1 negative cell line is unaffected. Similarly, with B cell lines that we have from emu LMP1 mice, we can show their dependency upon LMP1 transfecting in, again, a dominant negative of LMP1, and within a few days, the expression of that dominant negative disappears, either by shutting down expression or killing off those cells, and we can see it by flow cytometry and also by Western, showing again the dependency for survival of those cells on LMP1 expression. So I'm not sure I managed to do it in 30 seconds, but hopefully enough time. In summary, emu EBNA1 transgenic mice represent a valuable resource to continue to understand the mechanism of action of EBNA1 in vivo, and a valuable resource to test therapeutic approaches, both primary tumor cells and cells in culture and in vivo. For example, Christian Muntz has used this system in exploring anti-EBNA1 vaccination. The tumor cells are transplantable. They can be passaged indefinitely in vivo, and we have red fluorescent versions of these. The LMP1 mice also represent a useful model for studying LMP1 and for treating chronic inflammation, EBV aside, but also for exploring LMP1 therapeutic targeting. And the carcinoma cell lines can be used both in culture and again back in vivo. For example, Archisa has used these some of these cells. What next? If you would like any of these cell lines or primary tumor cells that I have described, just please email me. If you'd like the transgenic mice I have described, I hope to pass them on to the National Mouse Archive at Harwell. So contact me and I'll let you know what stage that's at. Here are a few of the people who did some of the work, and thank you.
questions? There's several down here.
That's remarkable work, but can you tell us exactly what kind of B-cell lymphoma, by inhuman lymphoma terminology, are you creating with EBNA1? Is it an activated DLBCL? Is it germinal center? Is it CLL? What's your lymphoma?
So the B-cell lymphomas are lymphoblastic. They have started to undergo immunoglobulin gene rearrangement. They don't have a Burkitt lymphoma starry sky type pattern, but I don't know that mice easily develop that type of pathology. What kind of thing do you want to know? They don't make immunoglobulins. They're centrocytic, I would say, centroblastic, centrocytic, both of those. What was the other one you said? IR4? I haven't looked at IR4.
Thank you. Really wonderful work and amazing resources. For the LMP1 transgenic mice, it's interesting they have an inflammatory phenotype because the potassium efflux is a known activator of the inflammasome. And LMP1 in latency is also associated with exosome formation. It's thought to modify the tumor microenvironment. Do these mice produce a lot of exosomes? Might they be an excellent model for packaging or studying exosomes?
We haven't looked at exosomes at all. I can tell you that the level of expression of LMP1 is really very low, and yet high enough to develop this dramatic phenotype. But very low expression, nevertheless, quite hard to see by Western blot. But we haven't looked at exosomes at all.
That makes sense because high expression of LMP1 appears to be toxic in cells in general. So
yeah, when we originally made the mice, we had several lines of mice that had different levels of LMP1 expression. And of course, as you go down, as you get lower and lower, you get to mice that have no phenotype. And as you go up, you just kill the mice. So this is sort of, was in the middle of the spectrum, but it's still nevertheless very, very low expression.
I think this might be the last question.
Do you think that the absence, I understand the EBNA1 transgenic mouse was not inflammatory. Do you think that the inhibition of inflammation in the human model with EBNA1 is the explanation for why you don't find it? And if you dropped out the GAGA repeat in the middle that seems to mediate this, that you would get an inflammatory response?
The EBNA1 mice, this is lymphoma, and we see the lymphoma develop in spleen and lymph nodes. The inflammation I was talking about was in the LMP1 expression in the epidermis.
I'm talking about the EBNA1 model.
Yeah.
And so I'd understood you did not find inflammation in that model. Is that right?
You don't see inflammation in the spleen or the lymph nodes, but.
And so EBNA1 has properties that inhibits inflammation in people, and that seems to come from the GAGA repeat in the center. If you drop that out, you think you might see an inflammatory response from that model too.
So the glycine alanine repeat in EBNA1, it's been well demonstrated that inhibits the expression of EBNA1. And as I say, in our transgenic mice, it's the full length EBNA1 that's not modified in any way. And I can talk to you more about this afterwards, but my hypothesis of what's contributing to the lymphoma in these mice is actually in part mediated by the translation suppression in these mice. So these mice, they express RNA at a constant level, and in the tumors we see a release of that translation suppression. And that is in part, I think, and there's several pieces of evidence to support this, in part responsible for the development of tumors in these mice.
Thank you. And I think we should thank all of the speakers. Thank you.